All right, and if you are participating via Zoom's online platform and you have any questions during any of the Q&A portions of the agenda today, we're asking that you type your question into the Q&A box on the Zoom webinar, and we will address as many of the online questions as we're able to. For in-person attendees, if you have a question during the Q&A portion, please raise your hand, and we will have a mic runner who will come around and you can speak your question into the microphone so that it's projected so everyone in the room can hear you, but also all the folks that are online then will be able to hear your question. All right, so that being said, I think we're ready to get going. First of all, I would like to introduce Campbell Ingram, who is the Executive Officer of the Sacramento-San Joaquin Delta Conservancy, to provide some welcoming words.
Good morning, everyone. It's my honor to welcome you all to the 5th Biennial Delta Invasive Species Symposium. I want to start by thanking the organizers and the Delta Interagency Invasive Species Coordination team for all their effort to bring us all here together today. I know this is going to be an informative and transformative event, and we really have them to thank, and of course, thank all of you for taking your time to be here and participate. We all know that the Delta is an incredibly important resource and a very special and beautiful place, but sadly, it's also characterized by its many stressors.
You're good. You're just kidding.
People can see my head now. You know, all the stressors from loss of over 90% of the habitat, fundamental changes in the hydrology and food web dynamics, and of course, it's one of the most invaded estuaries in the world. So it's truly daunting and uncertain work trying to control these invasive species, but we're making progress. One success story that we're very proud of is that our board approved a $10 million grant several years ago to California Department of Fish and Wildlife to help control nutria population that was centered in the San Joaquin Valley. You might wonder why the Conservancy is funding projects that far down in the system. Well, it was just out of recognition of the devastation that the nutria could cause if it actually got into the Delta. So I'm very happy to say that even with increased effort, there's been a reduction in number of animals taken, and though we've had some detects in the Delta, nutria have not gotten a foothold in the Delta. And so that's a great thing. And one of our first speakers is going to talk more in depth about that project. Sadly, some of the invasives that we face now and will face in the future won't be so easily controlled, but working in coordination to detect and respond rapidly, we significantly improve our odds. So the work is it takes perseverance, it takes optimism, it takes coordination, and coordination takes relationships. And I think that's really what today is all about. Really spending the time to get to know each other a little better, understanding our respective roles, getting an update on the science, and then really just figuring out how we can better coordinate to work together and achieve the greatest possible collective impact. So with that, I just want to say welcome again. It's great to see so many people here, and I'm going to turn it over to Henry DeBay, Deputy Executive Officer of the Delta Science Stewardship Council. Sorry, Henry. That's it.
Good morning, everybody. It's good to see you all this morning and see this great turnout in person and online. So I also have the fun job of welcoming you all to today's 5th Biennial Delta Interagency Invasive Species Coordination Team Symposium. Say that 5 times fast. Or DIS Team Symposium, and it is the 5th of its kind, but it's also the first back in person since the pandemic, so it's good to see you all here today. So as Campbell mentioned, my name is Henry DeBay. I'm the Deputy Executive Officer for Science at the Delta Stewardship Council, and we're really thrilled to co-host today's symposium because it really embodies the spirit of the DIS team and the goal of the DIS team, which is to foster communication and collaboration across state agencies that are working to detect, prevent, and manage invasive species and invaded habitats in the Delta. And we're strong believers that events like this today can really help to spark new ideas and to foster new collaborations or connections, if you will. And in addition to promoting information exchange, the DIS team really works hard to prioritize research needs, to fund those research needs, and to communicate the results of that research. And that's something the council also shares a deep interest in, which is the reason we prioritize research needs through the Science Action Agenda. We published the 2022 to 2026 science action agenda just last year. We also fund science, so we have a science funding program that funds projects and fellowships on everything from using remote sensing and LiDAR to more accurately and efficiently map invasive aquatic vegetation in the Delta, to exploring the human dimensions of invasions. And we're really excited to see some of the Council Award recipients on the agenda today: Dr. Matzek, Dr. Tanner, but in addition to that, we also communicate science. So just earlier this year, we published the 2022 State of Bay Delta Science, which focused on plants and algae and included a chapter on invasive aquatic vegetation, the state of science for the treatments. And we have those handy dandy Nutria pocket guides, which we developed in coordination with a number of you. And so the presentations and discussions today will not only help to further the information exchange that Campbell was talking about, but they'll help to hone those priority research needs, those funding efforts, and obviously the communication of the research that gets funded. So with that, I do want to just thank again the organizers, Rachel, the DISC team, all of the presenters today, everybody here in the room and online, and wish you all a fruitful symposium. So thank you very much.
Thanks so much, Henry and Campbell. Now we are going to have a brief video welcome from a legislator.
Good morning and welcome to the 2023 Delta Invasive Species Symposium. I'm Assemblymember Tim Grayson, and it's an honor to greet you today. As a policymaker and proud member of the Delta Caucus, it became priority for me to engage in the detection, prevention, and eradication of harmful invasive species that negatively impact our economy and environment. Invasive species do not stop at property lines, recognize political boundaries, or acknowledge interagency lines of authority. Communication, collaboration, and coordination are all paramount to our success in the fight to stop the spread and safeguard our unique environmental heritage. That's why, in 2018, I authored AB 2470 to establish the California Invasive Species Council and Invasive Species Advisory Committee, bringing together diverse stakeholders to coordinate around the common problem of invasive species. That's also why your involvement in today's symposium is essential to the progress in California's fight to stop the spread. Thank you for your time and coming together to find workable solutions in partnership with each other for the benefit of all California.
Excellent. So thanks so much to the Delta Stewardship Council staff that helped coordinate getting that legislative welcome together for us. All right, now that you all have been thoroughly welcomed to today's event, I am going to provide just a few opening words to introduce myself and also the Delta Interagency Invasive Species Coordination Team and give a little bit of background about today's symposium. So for those of you who don't know me, my name is Rachel Wigginton. I am a senior environmental scientist at the Sacramento-San Joaquin Delta Conservancy, and I am also a co-leader and the facilitator of the Delta Interagency Invasive Species Coordination Team, or DISC team. The DISC team is involved, as Henry and Campbell both said, in coordinating invasive species management in the Delta, and one of our big events is this 5th biennial Delta Invasive Species Symposium, which was first organized in 2015. So we really appreciate you all being here and being part of the ongoing work of the DISC team. The DISC team itself was formed in 2013 with the goal of fostering collaboration and communication among groups in California that detect, prevent, and manage invasive species, and also restore invaded habitats in the Sacramento and San Joaquin Delta. The group is co-led by staff from the Delta Conservancy, the Delta Stewardship Council, and the Department of Water Resources, but our team members come from a variety of federal, state, and local agencies, nonprofit groups, as well as academia. So it's a really interdisciplinary group of folks coming together to think about the complex problems of invasive species in the Delta. And we seek to achieve our main goal through a few different avenues. So first, we really focus on the exchange of information among the members in our group, who then communicate it out to the other folks at their agencies. So as we know, the Delta is a really complex region, both ecologically, socially, and politically. And our team comes together quarterly to provide updates to each other about the work that we're doing. And we also collectively try to problem solve any big issues that team members are having in their respective works. We also bring speakers to these quarterly meetings in an effort to strengthen the science, policy, and management interface. And then we know that that really impacts how invasive species work happens in the Delta. And finally, we organize events on important topics like the biennial symposium, which in the past has focused on things like early detection and rapid response, remote sensing, and other emerging topics and tools that are important for invasive species professionals. Next, the team works together to identify emerging management needs and highlight research and scientific questions that could need to be addressed in order to meet those management needs. In the past, we've done things like develop a research needs table, which helped us to give feedback to the last update to the Delta Science Action Agenda, which was developed by the Delta Stewardship Council. And we use that list and that group think about what our research and management needs are in an attempt to leverage funding to actually address some of those needs. So for example, this image shows an experimental replanting at a restored tidal marsh restoration in the Delta. And this experimental replanting was designed to understand how active revegetation might make restoration sites more resistant to invasion in the future. And this idea for this experiment came out of the development of that research needs table, was proposed by a set of DISC team members, and then was funded by the Delta Conservancy. So really that brought a lot of our team members together to put this experiment out in the ground and help us understand better how to control invasive species in these restored sites. So if you'd like to know more about the DISC team, any of our activities, please feel free to find me or any of the other facilitators today who are all part and members and leaders on the DISC team. Next, just at the top, I think it's always good to say our thank yous first. I'd like to take a moment to thank and recognize the symposium planning team. Caesar, Elizabeth, Gina, Lauren, and Rachel, we've all been working for over 6 months to plan this event, and I really appreciate all of their dedication in making today happen. I'd also like to recognize the support staff and volunteers that are contributing today: Beck, Emma, Eric, Hope, Mohammed, Annie, Nick, and Eddie in absentia. I believe he's ill. But all these folks at least offered to be here and help to put today's event on, and we really appreciate that group effort that makes these sorts of things happen. And last, the food and beverages, which you are enjoying in this morning and will also be getting at lunch, were provided through a generous support from the Alexander and Elizabeth Swantz Endowment, care of Ted Grosholz. So we really appreciate that support of today's event. All right, so I'm just going to give a quick overview of today's agenda. There are printed agendas out on the sign-in table if you didn't get one, but we're in our opening session today, right now. And then at 9:15, we'll hop into our first session, which will explore connectivity within and among estuaries and how that relates to invasion. At 10:50, our second session will really be an interdisciplinary session looking at how connections among research, management, and policy impact our work. At 12:30, we'll have lunch. And then at 1:30, we'll come back for a set of lightning talks, which will update us about invasive species work that's happening in the Delta or relevant to the Delta, before our last session at 2:45, which addresses how connectivity can create both challenges and advantages for the management of invasive species. So we have a really action-packed day today, so I really appreciate everyone being here and being ready to engage with these exciting speakers that we've brought together. So with that, I'd like to turn it over to Dr. Laurel Larson, who is the Delta lead scientist, and Laurel is going to provide some opening remarks for today's symposium.
Well, thank you, Rachel, and I'd like to further extend Henry DeBay's warm welcome on behalf of the Delta Science Program. It's great to see you all in person this year. So I just called my talk Opening Connections, Understanding Connectivity in a Degraded Estuary. I'm going to try to set the stage and provide some more context on how we might integrate these concepts of connectivity and invasive species management. But I'll also say that this is kind of a closing connection for me as well. I'm in my last few hours as the Delta lead scientist. Today is my last day. But it's a great way to spend my last day because doing science coordination with the wide variety of people with diverse interests and expertise has been one of the best parts of this job, hands down. And so let's keep the connection open. So I think, well, let's just start with a statement I think we could all agree with, which is that connectivity could either help or hinder the achievement of resilience objectives. And so this slide really shows the connectivity of habitats in the North Delta and how 10 years ago, back in 2013, it was really recognized that this connectivity of habitats in this region of the Delta was actually supporting kind of a hotspot for native fish species at a time when it was just thought that they were ubiquitously doing terribly within the Delta. On the other hand, we're also very familiar with how connectivity can hinder the achievement of resilience objectives when that connectivity promotes the spread of organisms that are undesirable, such as the COVID-19 virus, or in many cases, invasive species as well. But talking about connectivity gets a little more nuanced, especially when we go to the scientific literature. And so I just want to bring to light several different types of connectivity that we might be discussing when we bring up this issue of connectivity. And all of these, I think, are relevant to resource management and science. So at a very basic level, there's structural connectivity, which refers to the physical adjacency of landscape features like habitat within the North Delta Arc. There's also a slightly more nuanced concept called functional connectivity, which is based on how that physical adjacency or contiguity actually translates into fluxes of water, solutes, or organisms. And this is something I had the great privilege to be involved with in my previous work in the Everglades, where we were interested in how management actions to reconnect compartmentalized parts of this landscape via restoring flow and breaking down linear features such as levees and canals. You can see one of them down here. Oh, you can't see my cursor. But anyway, we were trying to understand how restoring flow through this landscape might impact biogeochemical processes and the contiguity of biogeochemical processes across the landscape. And so we designed some experiments before and after these pulsed flow releases and the breakdown of these levees in order, and accompanied those with sophisticated statistical analyses in order to figure out how the landscape was connected with respect to a wide variety of nutrients and other biogeochemical constituents. A type of connectivity that's often less familiar, I think, to ecologists is the concept of process connectivity. This is something that's really taking off in hydrology, but it refers to flows of information among physical and biological drivers in an ecosystem. And it's used as a concept that can help us understand what are the primary drivers in a complex system where you have lots of different processes and variables that are interacting. And this is actually something that we can map out now with high resolution time series. And one example comes from a paper that my postdoc, Adam Mojes, recently published, in which he was looking at flows of information from variables like precipitation and snowmelt to stream flow. So the stream flow arc is this, oh, I don't know if there's a pointer. The stream flow arc is the pink one that you see in the lower right, and you can see that there's about an equal amount of information from precipitation and snowmelt flowing to stream flow in this particular river, which is the Cedar River, Washington. There's a small amount of information from minimum and maximum temperature flowing to stream flow and soil moisture as well. So that's just something that can help us understand what might be the dominant processes or variables that we would need to control or understand in order to be able to forecast behavior, an aspect of ecosystems or the environment. So back in 2020, the Delta Stewardship Council and IMSC, the Institute for Marine and Coastal Sciences at UC Davis, put on a symposium on estuary and connectivity. And there were a lot of really great synthesis papers that came out of that symposium, including this one that I'm highlighting from Annika Keeley, who used to be Delta Science Program staff, that came out in Bioscience recently. And it's a really nice summary of different types of connectivity and how we should integrate those concepts into conservation, particularly in places like the Delta. So she describes ecological connectivity as emphasizing the movement of biota and materials as a process that influences population persistence, including the functional connections across landscapes that can be impeded by fragmentation and habitat modification. And she also references hydrological connectivity as emphasizing the functional role of water in these processes that create and maintain the geomorphic system and affect water quality and biogeochemistry in the embedded processes that shape ecological communities. And I think maybe most importantly, she emphasizes social connectivity, which is equally important to achieving conservation objectives. And this refers to the flow of information among institutions and knowledge across sectors to improve understanding and governance of cross-boundary processes. And all these different types of connectivity, as well as spatial dimensions of connectivity, can be integrated through this concept, which Annika Keeley refers to as the ecoscape concept, that recognizes these many types of connectivity across different scales. So in this graphic, she's showing global scales, regional scales, and local scales, and referring to different aspects of connectivity that refers to how organisms or constituents move. So you've got longitudinal connectivity connecting different parts of the estuary along the salinity gradient. You have lateral connectivity referring to exchanges between channels and marshes or floodplains. Vertical connectivity referring to exchanges between surface water and groundwater. Lattice connectivity, which relates to how organisms move in all directions. And then teleconnectivity, which is a concept that's most relevant to organisms that fly or swim quickly and can readily traverse inhospitable parts of the landscape. So in thinking through projects or examples that I could highlight that really integrate these different concepts of connectivity, one in particular came to mind, which is one of the projects that Henry mentioned we had funded through our 2021 proposal solicitation, and that you'll hear a little bit more about from Dr. Virginia Matzik later today. But this is a project that focuses on the spread of Phragmites in Suisun Marsh. This is arguably probably the most significant emergent vegetation invasive species we have within the Delta. And in fact, Suisun Marsh has one of the highest documented rates of Phragmites spread in the world, with an average rate of spread of about 1.32 meters per year, which is a number that they recently published in this paper that just came out a few months ago. And in particular, considering these different dimensions of connectivity is really important in understanding how we might more effectively control the spread of Phragmites. So structurally, Suisun Marsh is a somewhat disconnected landscape, or there's different degrees of connectivity among different elements of the landscape. And you'll see in this map that there are a lot of wetlands that are tidal wetlands in brown, but there's also a mosaic of managed wetlands. And the way that we control Phragmites in these different types of wetlands is different. We're not permitted, or we're not legally allowed to use certain chemical controls in parts of the landscape that experience that tidal exchange. Now, social connectivity is another really important dimension here, because in addition to this mosaic of tidal exchange and mosaic of connectivity that you see in this map, there's also a mosaic of land ownership that comes into play. And what this team is finding is that really tight coordination among these different landowners in terms of the strategies that are being used to control Phragmites is critically important here. So I hope that example and Henry's opening talk a little bit earlier really underscores that invasive species management is a priority that we value within the Delta Science program. There are 2 science actions in the 2022 to 2026 Science Action Agenda that feature invasive species management. One of them is about synthesis of knowledge and generation of new knowledge to evaluate the costs and benefits of different strategies for minimizing the introduction and spread of invasive species and informing early detection and rapid response strategies. And then the second one is about identifying and testing innovative methods for effective control or management of invasive aquatic vegetation, particularly in tidal parts of the Delta, under both current and projected climate conditions. And I'll just say that you too have an opportunity to get your work funded through the proposal solicitation, because early in the new year, the science program is getting ready to release a new solicitation. And one of the things that we're emphasizing in this solicitation is social science studies that integrate the social science dimension, that integrate social connectivity. And I think this is a really great opportunity for the invasive species management community. Now, Henry also mentioned that we play a role in synthesizing science. It's another core part of our science strategy. And invasive species management features prominently in our most recent edition of State of Bay Delta Science, where 3 out of the 6 science articles in that edition feature invasive species with a focus on ecology, invasive species management, and then remote sensing applications. And that chapter has a really interesting description of how we could apply the latest technology to move towards near real-time capabilities for mapping and then controlling invasive species spread within the Delta. So in my last few slides, I'd like to return to the literature. Oh, it's weird, these boxes got shifted around. I'd like to return to the literature. And when I was preparing for this talk, I did a literature search on studies that really integrate these concepts of connectivity with invasive species management. And I'd like to highlight this paper that does this really well. It's a review paper that's about 10 years old at this point, but I think it provides us with some really valuable food for thought as we consider the theme of the symposium today. And so this paper really discussed this type of interactions that result from integrating these two concepts. And there's a few visual examples here. So just to orient you to this graphic, the hatched area indicates a part of the landscape that's been treated for invasive species, whereas the solid green indicates the part of the landscape where that treatment is effective, where invasive species are actually eradicated. And so when we consider these two concepts together, a lot of times we have a situation where we treat a large area, but then there's movement of invasive species organisms in around the perimeter, which leads to a core effect. So a much smaller area benefiting from treatment than the area that was actually treated. However, the reverse is also true. And it all depends on the ecological processes and interactions between native and invasive species that are dominant in the landscape. So with the halo effect, the area that benefits from the treatment of invasive species is actually larger than the area treated for invasive species. And if we think about our treatment of invasive species at a landscape scale, something that might be quite beneficial would be a large halo effect where you have two isolated treated areas, but they collectively produce a connected corridor in which invasive species have been eradicated. And then, of course, the situation we would want to avoid is actually treating a corridor, but experiencing a large core effect that would lead to isolation of areas where that treatment was effective. And so these authors provide us with some conclusions that I think are quite motivating as we go through this symposium today. And one is that, you know, when we think about how these two processes integrate at a landscape scale, it really argues for spatially explicit planning approaches at the scale of the landscape. So by identifying areas vital to landscape connectivity and species persistence, conservation planning can guide optimal targeting of invasive species management and other conservation actions in space and time, potentially delivering far greater benefits with the same resources. So there's an issue of efficiency at play. I'm just kind of astounded by how the fonts have also changed in this presentation. Applying metapopulation theory in reverse to invasive species by reducing their connectivity and understanding the mechanisms behind local extinctions might help us to design networks that are well connected for desirable species, but minimally so for invasive species. And then targeting of invasive species management should be guided not only by current distributions of native taxa, but also by their potential distributions when we consider how they interact with invasive species and how they disperse on the landscape. And strategic targeting like this could really enhance biodiversity protection across landscapes. And so whenever I give an opening talk, I really like to leave the audience here with a challenge. And my challenge to you is to really think deliberately about what you want to come out of this symposium. And I think you all did a fantastic job in your 2021 symposium, which had a focus on early detection and rapid response. That led to a proposal that the Delta Science Program has funded that will pilot an early detection rapid response strategy for the Delta using the example of ribbonweed, which is a species that invaded the North Sacramento River back in 2018 and has spread since then. This pilot strategy is being led by Rachel Wigginton at the Delta Conservancy, as well as collaborators at CSU Long Beach. And so I think it's just a really great example of how a symposium like this can lead to action. And so my challenge to you is to really think deliberately about what you want to come out of this symposium today. If I were to give you an answer right now based on the preparation I did for this talk, I would really encourage you to think about ways to implement this integrative landscape scale planning with respect to invasive species management. It's something that's been called for with respect to planning marsh restoration, and I think it's also quite appropriate to think about this need for landscape scale planning in the context of invasive species management. And so with that, I will let you all go to your break.
I'm not a botanist, so if I use the wrong terminology, please forgive me. But do you find that the source for the spread of invasive vegetation in the Delta is in the Delta itself, or is it coming down the San Joaquin and the Sacramento rivers? And if you do an abatement in the Delta, should you, or is it recommended to go upstream if the sources are in the rivers? And how do you decide how far upstream to go?
I think that those are good questions, and they highlight considerations that should come into play that are specific to the species that you want to manage. In the Delta, when we're talking about invasive aquatic weeds, in most cases, these are species that are abundant within the Delta, and the origin of spread is indeed within the Delta. But the whole concept behind early detection and rapid response is that, you know, there are all these vectors from both upstream and from the oceanic environment. There might be things coming in marine ballast water, although there are regulations that try to restrict that. But there are many vectors for introduction of new invasive species within the Delta. And the idea behind EDRR is that we need to control the spread of those species as rapidly as possible in order to prevent an unmanageable situation where they become so abundant through the Delta that they're just really costly and difficult to control.
Awesome, thanks, Laurel. Do we have any questions from our Zoom participants?
There's one in the room, or two more.
Okay, so there's one more in the room. Yeah, go ahead.
What you just said about if the species is so prevalent that you're not even bothering is, I noticed that nowhere in the agenda was discussion of water primrose or red Suspania, which are two species that we work with in a small creek called Steelhead Creek, and I've seen the water primrose up as far as the Russian River. And I know that there is a stand of red Suspania in the city of Folsom that comes down through Lake Natoma into the American River. And it's very frustrating, you know, to see that. But it sounds like that's what the issue is that those two species are so prevalent that there's just they're not able to.
So Division of Boating and Waterways does implement control measures for those species that are abundant in order to help achieve boating objectives within the Delta. But yeah, I'm not an expert in invasive species myself, so I can't speak to those two species specifically. I know we have experts here in the room. But you know, in many cases and in many places, this isn't unique to the Delta, we're often faced with situations where invasive species are abundant. They're not going to be eliminated. And I think what's really important to do is to have processes of engagement with community members to identify what are the highest priority objectives for managing those invasive species and coexisting with them, and what are the priority locations for treatment where we want to try to promote habitat for certain target species or for recreational use. So there's really a need for, you know, strengthening social connectivity and establishing these processes of community engagement. And I think that's one of the things that some of the recent groups that we've funded through our proposal solicitations is trying to do.
Excellent, thank you. Was there one more question in the room? I think we have time for one more if there was one. No? All right, well, thank you so much to our welcoming speakers, and particularly to Laurel for joining us on her very last day as the Delta lead scientist. We now have a break until 9:15, so feel free to use the restroom, get another drink, and be back here at 9:15 for the first session. Thank you.
But we're going to try and keep on time, so I'm going to bring us back here. My name is Lauren Damon. I'm the Ecosystem Restoration and Climate Adaptation Project Supervisor at the Delta Conservancy, and I am happy to be facilitating our session 1, Connectivity Within and Among Estuaries. Our first speaker, I want to invite up is Tal Robinson. He's the Nutria Eradication Operations Supervisor at the California Department of Fish and Wildlife.
Okay, good morning. I am here to talk about eradicating the invasive nutria from the Delta and the rest of California. The arrow's not working. Oh, there we go. Okay. Okay, so as Lauren mentioned, my name is Talmadge Robinson. I'm the Senior Supervisor for the Stockton office, which is the Northern Operations Office for the Nutria Eradication Program of the Department of Fish and Wildlife. I have a staff of about 14 employees currently, with a few vacancies that I'm actively trying to fill. And our sister office down in Los Banos has a complement of close to 30 employees. So just to kind of give you an idea of the scale of the staff that we have and how much effort it actually takes on the ground. So for those of you not familiar with nutria, nutria is a large semi-aquatic rodent. They inhabit fresh and brackish water. We essentially found them in every type of water body that California has so far. They are native to South America. They were first introduced in the U.S. historically for the fur trade and farmed. I don't know how extensively, but they were present in several places in California. They were eradicated back in the 1970s and rediscovered in California in 2017 on a managed wetland down near Los Banos, California. And some preliminary genetic studies have indicated that this is a separate population source. That's not been released yet, but that's what we have come to find out. Since 2018, when the program really kind of started, gaining traction and garnering support from cooperators and the state internally, we found nutria present in Fresno, Mariposa, Madera, Stanislaus, San Joaquin, Sacramento, Solano, and Tuolumne counties. For the biology, they are reproductive by about 4 months of age. Average litter size in California, based on what we found for fetal presence in nutria that we've necropsied, is about 6. And they're able to have 3 litters a year, just about. So it's about a 13-month period, and they can have 3 litters on the ground. They do live in social groups. Home range size is really kind of unclear. We haven't studied it at all in California. You could say it's 25 acres. That's what some of the literature has suggested, but it's not really known here in California. And dispersal is also likewise unknown. The first indication of presence was down in Los Banos, and now we found several animals up in the Delta. So there's quite a bit of mileage between those two locations. And they can move several miles in a day, just based on camera evidence that we've returned. So some of the key identifying characteristics, I don't have a pointer, I'm sorry, but if you look at the images that we have here, you can see a really prominent golden crest around the ear. They also have a white muzzle with white whiskers. This is very typical. These pictures are very representative of what most nutria look like. And they do have that long, relatively hairless tail. It is lightly haired, but it's certainly a rat-looking tail. It is round and long. Why are they so invasive and destructive? Because they can consume up to about 25% of their body weight each day. And what's more is that they destroy the plant when they eat it. They typically take the basal portion of the plant and they eat the interior tuber and can uproot the entire plant. And they're basically able to feed in such a way that it can denude the habitat. So with that, there's potential for severe erosion and conversion to open water systems, like if any of you are familiar with the Chesapeake Bay project, where they had nutria, basically wiped out that entire vegetated system in Chesapeake Bay. So there's potential for it to exacerbate invasive plant infestations. That also has not been looked at, so we don't know if there is really any direct link to that, but certainly the opportunity exists. And what's really interesting is once you remove a population of nutria from an environment, going back into that environment again, seeing the natural regrowth that occurs, is remarkable. It's really outstanding to see, how much the vegetation actually returns and how quickly. But that also leads to us having limited access because it's just hard to get around in the environment when stuff grows back in. Another concern for nutria being is damage to levees and flood infrastructure. They are known to burrow, although it doesn't seem to be such an issue here in California. I think that's because they're not as well established as they have been in other places. And I just mean that in kind of the temporal sense. They haven't been here that long to really impact our levees, thankfully. They will inhabit just about any water body that has slow enough flows to hold animals. And we actually saw them in the San Joaquin River. We had a population living there back before 2022 when we had all that rain. So when we had drought conditions, the river flows were really low, and nutria were living right in the middle of the river. So that was kind of unexpected. Typically, we find them in side ponds, sloughs, backwater areas, that kind of thing. So to find them living in a river was kind of a interesting development. How do we find them? So our primary detection methods are just good old-fashioned hard work, getting out on the ground. When the program was stood up, and I wasn't involved in it at this time, but the program was faced with a challenge of how to actually break this down, find the animals, and put together a systematic way to cover the landscape, not knowing the full extent of the population. So they came up with a grid network of 40-acre cells and basically covered the entire state with these 40-acre grid cells. And the idea was to go in, classify the habitat, and then start to work your way through based on the quality of the habitat. It's not rocket science. We use boats, kayaks, we walk, and we drive in vehicles to get out in the habitat. And for those of you that have spent any time in marshy areas, you know how difficult it is to walk through them. And we look for scat tracks, dens, burrows, any sign that we think scat is the best indicator if we can find that, and vegetative cuttings. Vegetation cuttings, though, can be problematic because there's a lot of species overlap, damage that occurs from beavers, muskrats, and even otters can be confounding. And we use cameras. So we set up a lot of camera traps. We have them all over the place, and we use platforms to attract nutria. Typically when they feed, they establish a vegetative platform just because they're constantly hauling out of the water on the same location over and over again, and it beats down the vegetation. So the platforms that we either create or take out are designed to replicate that. So we use a 2x2 wooden platform that we build in the shop, or we can actually take grass and make a mat out in the field. And this is just to show you where the first detection was, again, down by Los Banos. And this is the current heat map based on the recent detections that we've had. Excuse me, I might have a drink of water here. So you can see that we have found them in many more locations, spread out across a large portion of the state. But thankfully, they are fairly isolated to their local areas. So you see this map and you see the circles, and that really is kind of how the population is distributed. It's not as though they are extent throughout that whole stretch, if that makes sense. Prior to the winter of 2022, like I said, we had drought conditions. They were living in rivers, and we really didn't see a whole lot of evidence of dispersal. But after that winter and all the water that we received, we believe that contributed to nutria expanding up into the delta. In response to that first detection that we got back in Sherman Island, we actually shifted all of our staff from Los Banos and Stockton to the Delta. So we had about 25 staff, I think, at that time, move up and blitz habitat classification and camera traps and just cover the ground as intensively as we could. Yeah, and since we've taken Nutria in Sacramento and Solano counties, waterfowl season has also been a problem for us because a lot of the landowners that we have agreements to enter their property, they've restricted access, obviously, for this time of year because they don't want us to disturb the waterfowl. But once waterfowl season ends, we will be busy. So as you can imagine, there are a lot of challenges faced in the Delta, varying water depths due to tidal influence and land management. There's constant fluctuation to manage species and to also flood out product season and drain product season. And there's a myriad of challenges, and each property is different, individually managed. Some properties are connected in such a way that you have to flood through them to get water to another property, which also creates problems. So again, it's just a challenge. There are a lot of remote locations that we have to get out to. Takes a long time to drive to them. It's hard to get equipment out to them. You can just imagine what we deal with. And remnant animals are always the hardest to capture. There's plenty of open habitat, fortunately, as you know, I've suggested the nutria aren't extant everywhere. So that means that there's a lot of, you know, available habitat out there. Containment is also a problem because we don't really have any containment out in the Delta. These animals are free to jump levees and cross property lines, and they can go anywhere. But there is some good news because we are successfully finding and trapping out known populations. And as you can imagine, the process is tedious, time consuming, and challenging. Just to show you the amount of effort and the returns that we've been getting recently, well, I should say through the years. The number of cells surveyed is the large gray bar on this chart. The number of nutria taken is the orange hash, and the number of cells with nutria taken is the black bar. So you can see that, except for this year, the trends are going in the right direction, and we have a reason why this year is an exception. Because in Fresno County, which has been put on the back burner until last year, really, has accounted for 568 of the animals that we've taken, which has caused that spike in the number of animals we've gotten. And you can see that here in this graphic as well. So just to give you an idea how much effort we've put into this, we've completed habitat assessments on over 1.85 million acres, executed entry permits with over 4,600 landowners in 14 counties. We've set up 9,300 camera stations. We have 1,239 currently active, and we've conducted 78,000 camera checks. Deployed 11,000 trap sets for a total of 130,440 trap nights. And that was as of Monday. So, you know, it's gone up since then. We've taken 4,001 nutria and 4,719 fetal nutria. So we are looking at many ways to improve our success. I'll just go over a couple briefly here. Citizen reports are definitely encouraged. We are using scent detector dogs with wildlife services. We are planning to deploy sentinel animals with satellite telemetry tracking. The use of drones is being considered. And then we're flexible. We're moving staff around on the landscape as needed to respond. Our detector dogs, you can see in this graphic, represent the blue line, and the handlers that go along with the dogs to make sure they're staying busy and being productive are the red line. So you can see the dog covers a lot more ground than the handler. But that's been very informative and very helpful to get out in some of these places and get confirming information based on what we think and then seeing the dog respond. It's been very good. So just to summarize, eradication trend lines are heading in the right direction. The program remains adaptable and responsive. Staff are dedicated and show great resolve. Collaborators and interagency support has been outstanding. And stakeholders, landowners, and the public are critical to our program success. We still have a lot of work ahead, but we're getting it done. And with that, I'll take questions, I guess.
Thanks for that talk. Can you say a little bit more about why the shift in focus from the Central Valley to the Delta, when it seemed like from your heat map there were more nutria in the Central Valley? So what kind of precipitated that focus in the Delta? Although I'm not mad at it.
Yeah, it's all about where we're finding them. So, you know, for a long time, my staff in Stockton were monitoring that central connected corridor between the Southern office and the Delta, and we just weren't finding them. And so when we did, we shifted. And so before we kind of had a skirmish line, we thought we had blockaded them from getting past us in the San Joaquin River, but then they showed up in the Delta, so we just moved.
I know that early on when the Genomic program was coming online, some of the necropsies were showing a lot of other issues within the nutria, like lesions on the liver. And is there any more information about that and how that might be affecting the population? And
we haven't seen anything that's affected the population as a whole. We've seen some localized stuff. We did have some pneumonia issues with a lot of those animals that we tried to capture and sterilize and collar. But no, we really haven't seen anything population-wide that's affected them.
Is putting a bounty on them, is that bad management practice? Does that just encourage artificial reproduction of them farms?
I don't know about the last part. I mean, it probably could, I guess, for folks. That model actually exists down in Louisiana, where they are paying a bounty. I think it's effective for controlling the population. It's not necessarily a strategy for eradicating, because then you're just targeting whatever animals you can recover in the field, and it's not looking at it from a population standpoint. So that would be an option, I think, for management, which is something potentially down the road if eradication doesn't go through. But at this point, I think eradication is going to work. So it's not a strategy we need to really consider, I think.
Are there any questions online?
Yeah. Yeah. How do nutria interact with other aquatic mammals such as beaver or otter?
So there is some anecdotal evidence based on the camera trap data that we have gotten. I mean, you guys, I can't even imagine how many actual photographs we've looked through as a program, but there is some anecdotal evidence that suggests some disturbance to other animals like beaver, especially, how they will be present on camera for a period of time. Then we see a nutria move in and the beaver disappears for a while. Nutria gone, the beaver probably comes back, you know, kind of thing. So it's not any hard science, but we do see some of that.
We can do one more in the room. And can you please state your name and affiliation for folks online?
Doug Johnson with the California Invasive Plant Council. I'm familiar with other large-scale eradication projects where it's difficult to say where is that end point and how much funding is going to be needed to get there, especially as maybe the numbers of nutria go down, but the cost to survey that full area remains large. So are you faced with a need to define an endpoint, like it'll take 5 more years or 10 more years at X amount of funding?
That fortunately is above my pay grade, so I don't have to worry about those issues at the moment. So Valerie Cook is a program manager, and she is for both of our offices, Los Banos and Stockton. That's more in her wheelhouse, but yeah, I mean, your question is a valid one. That will have to be a consideration at some point. But it's true of any eradication effort, especially for mammalian species. Your costs just go up exponentially all the way to the end.
Thank you so much, Tal. Next up, we have Drew Kerr, the treatment program manager for the invasive Spartina project.
Great, thank you very much. Well, it's a pleasure to join you today to talk about the San Francisco Estuary Invasive Spartina project, which is an excellent example of collaboration and partnerships. Anything involving wetland and aquatic invasive plants obviously requires coordination across boundaries. The weeds don't respect those boundaries whatsoever, and you need to leverage and harness the power of all the players in the area. Our project that I'll share with you today, a little bit of detail on various areas. Is a very complex project, very long-running project on a landscape scale, so I'm only able to touch on certain aspects today, but hopefully it'll give you a good overview. So the Invasive Spartina project began in 2000 as a partnership between the U.S. Fish and Wildlife Service and the State Coastal Conservancy. But it harnesses the power of over 200 different partners around the bay. This is a slide just representing a handful of those partners, some of the most active ones currently. And the project was started because we have lost so much of our tidal marsh around the San Francisco Bay. You can see in this time series from the historical to closer to the present day, losing about 85%. So it's essential to protect what remains. And the Goals project set out a goal of trying to restore up to 100,000 acres of the tidal marsh around the bay. And so the Spartina project is a foundation of that effort to restore tidal marsh in San Francisco Bay. And I think most of the folks in the audience here are very familiar with the benefits that tidal marshes provide. We have some of the special status species down there, such as the California ridgeways rail, saltmarsh harvest mouse, and of course just all the ecosystem services that tidal marshes play, from protecting urban areas from flooding and storm surge, to reducing erosion and sequestering carbon. And of course, a very important aspect is open space for recreation and for people to connect with these tidal marshes and enjoy them, understand them better, and build a political will to support them with funding and stewardship. We have a native Pacific cordgrass in the San Francisco estuary, Spartina foliosa, and it's a foundational species in the low marsh and pickleweed plain. It's an important early colonizer, and it is also an ecosystem engineer. We often refer to the non-native and stress its ecosystem engineer capabilities, but our native Pacific cordgrass is very much an ecosystem engineer as well. However, it plays well with others, our native, and it kind of grows in its own more narrow tidal regime. In the lower marsh, it's the lowest emergent plant in the tidal marsh, and it grows up into the pickleweed plain in mature marshes, but it kind of stays in its lane. Whereas the East Coast, the Spartina alterniflora that was introduced from the East Coast in the 1970s hybridized with our native Pacific cordgrass and produced this hybrid swarm that does not stay in its lane. You can see on the right-hand side of the diagram that shows how it grows not only down into the low marsh where Spartina foliosa grows, but way out into the mudflats, which are supposed to be unvegetated in our native system. It also grows throughout the entire pickleweed plain, dominating the native plants in those areas, colonizing the channels, changing the hydrology of the marshes, and well up into the transition zone as well. The UC Davis Strong Lab spent about 20 years researching the hybridity between these two, and has about 24 published papers on the topic. Why is hybrid Spartina a problem? Well, as I mentioned, it's a non-native ecosystem engineer. And of course, that means it hasn't evolved in this area with the different things that will keep it in balance with the system. So it dominates these mudflats. It's a very important aspect of the project that I think is often lost on folks, because they see that open mudflat and that absence of something there is kind of hard to understand. But that's extremely important for shorebird foraging. And there's an extensive UC Davis body of literature on that topic. It also displaces our native tidal marsh plant community and alters the hydrology. You can see on the right-hand side a diagram showing just a few clones in 1986 at a marsh at San Lorenzo Creek, and then how much it expanded into a monocultural meadow of hybrid Spartina by 2004. Some other aspects of why hybrid Spartina is a problem is undermining our native tidal marsh restoration, which I mentioned is extremely of what we're trying to protect and facilitate in San Francisco Bay. It threatens the rare plants and the biodiversity that throughout the wildlife that's provided when you have a diverse network of the native plant assemblage and the wildlife that rely on them. It reduces the flood control capacity and also can create mosquito breeding areas. The South Bay Salt Pond Restoration Project is probably the best example of this huge effort to restore tidal marshes around the bay. We also have the Napa Sonoma marshes closer to the delta here. The South Bay Salt Ponds is about 15,000 acres of decommissioned salt production ponds that are being converted to various types of marsh and pond habitat over several decades. So the Invasive Spartina project has 4 different programs. We have the inventory, treatment, restoration, and ridgeways rail monitoring. I'll kind of touch on a few of these. And you can see in the picture the kind of the scale of the photo is kind of to demonstrate the scale of a project. You can see little tiny biologists and this is just one marsh. We're covering 40 to 70,000 acres of San Francisco Bay Marsh and shoreline each year. OEI biologists surveying for the hybrid Spartina to facilitate treatment. The ISP also does extensive genetic testing. We're down to about 300 samples per year, but in many years it was up to about 500 samples to try to discern. It can be it's a hybrid swarm between the 2 species, and so it's fertile and back crosses. So the detections of some of the as we call them, cryptic hybrids can be very challenging even for trained biologists that are very experienced. And the diversity of the native cordgrass actually makes this very difficult as well. There are extreme regional differences in size and other morphological characteristics of the native Spartina that can make the hybrid ID challenging. And discerning that native often requires mature plants, which requires them to be later in the growing season, which further constricts the amount of time that we have to work on this each year. The definitive characteristics sometimes take several years to develop. During which time the plants could have been dispersing pollen that can pollen swamp the native plants and creating hybrid seed, creating little slaves that are sending out seeds around the bay and helping the infestation to move around the bay using the native plants as an agent, or it can just be dispersing seed that might take us for a while to track down all the progeny. Another partnership actually is an AI buddy system that one of our biologists, Simon Gunner, created, a linear discriminant analysis, or LINDA, we call her, computer program, which has memorized measurements of over 500 DNA verified plants. She's correct about 80% of the time on discerning between native and hybrid, but doesn't have eyes or hands. The ISP treatment program, which I manage, has a very narrow treatment window. But most of the sites around the bay where we have ridgeways rail, the endangered bird, we are coming in after breeding season for both often required for the proper detection of the hybrid, as I mentioned, because it might be later in the growing season that you can detect it accurately. But we have to come in, map it, to find all the occurrences of it, and then also accomplish treatment throughout a 9-county window of the bay, within those tight windows, to provide the utmost ridgeways rail protection, but also to get the highest accuracy. Our targeted spot treatment minimizes impacts to the habitat, and we use a lot of airboats. This happens to be one of the first days we ever used airboat back in 2008 to access marshes, and since then we've learned a lot about mud and how to move airboats effectively throughout the bay and safely. Majority of our sites, though, are treated by backpack, especially with all the reductions we've achieved over the years. Larger infestations are still treated by hauling hose out from airboat or truck. And this is just one example of the challenging environment that at low tide, and saying to the airboat pilot, we need to go back over there to this one occurrence that we need to treat. And he says, really? We need to go over there? And of course, this exact area we've known well, but when you first do these things, very, very nerve-wracking. The hybrid Spartina has been reduced by about 97.4% estuary-wide, starting with about 805 net acres. Back in the early part of the project, we didn't measure in what we call treatment acres nowadays. So that 805 net acres represents probably several thousand acres of pretty dense treatment acres of Spartina. We're now down to 20.7 net acres as of 2022, the last full year, and we're just finishing up our inventory for this year. Progress was slowed by some permit restrictions until very recently on treatment at a subset of sites that now contain about 80% of the entire remaining hybrid in the estuary. This is a time series of heat maps of every 5 years, showing kind of the progression, whereas you go from red in 2005 and through those first sets of years are the very dense infestations of over an acre, and you can see as they're moving into a lighter color, a less angry color, as I say, towards greens and yellows as we move through time. And you can see there's up in the delta, not as the North Bay especially has had smaller infestations. We've gotten on top of so many of those and reduced them to negligible levels or eradicated them, and that has really helped with the invasion pressure up into Suisun. So our 2022 invasive Spartina status, as of last year, we had 167 of our 221 ISP sites now contain under 10 square meters of invasive Spartina, with a total in all those sites of 200 square meters. 53 of those sites contain under 1 square meter. 60 sites were at zero detection. And a metric that I like to stress, or that I think is a good indication of continued progress, is that we've had an increase in these zero detections annually for 9 of the past 10 years, despite lots of things that are working against us, like drought and many of the other challenges that I've already mentioned. So we have phased treatment at a number of sites to protect the ridgeways around San Francisco Bay, as predicted in the ISP's EIR back in the beginning of the project, 2000, 2005. We saw that removing hybrid Spartina monocultures in this urbanized system would definitely reduce the artificially dense cover for rails in some areas. This is more problematic in fragmented marshes and those with low biodiversity. So treatment was restricted to a subset of sites in 2011, 2012 to proceed with caution and not put the rails at any risk of jeopardy. Unfortunately, the absence of management in those areas also allowed these infestations to spring back up to pre-treatment levels. But in 2018, our biological opinion allowed us to resume treatment at 7 sites. Of those, 7 of 10 sites already had 94% reductions by 2022, so we were able to make quick progress in some of those large monocultures without any native Spartina foliosa, I'll mention. And then we just received a new biological opinion in 2023 that is the phase 2 sites are now going to be resumed. We resumed treatment at 3 of those 6 marshes this year. I have a couple slides that are kind of Suisun specific here. So we had a we detected in 2016, and this is another good example of partnership. We had an infestation report from Point Buckler of some Spartina that were suspect. Biologists reported to us. We went out and investigated and did find that it was likely hybrid Spartina as well. It was confirmed by our DNA analysis. So that we expanded our surveys in 2017 and found that on Snag Island, one of the Motco Islands, we found fairly large infestation, and we found it radiating out from that area. About 90% of the entire Suisun infestation was on that Snag Island. Since then, we've gotten it down to about 275 square meters throughout that entire yellow area. Most of the occurrences are individual plants or small stands, and we continue to survey that each year and treat those. The nearest historical hybrid Spartina is about 9 miles east at Southampton Marsh, a very small infestation of just a few occurrences now. So this was likely an independent introduction, but it's certainly possible that it came in on the tides. And as far as the movement of propagules, the ISP did a small drift card study back in the early days of the Spartina project. Cards were released from 2007 and 2008, and then we did a second round in 2011-12. And within the estuary, the furthest dispersal distance ranges from about 2.5 miles to over 42 miles within the estuary. The cards from 10 of these sites actually left the Golden Gate and were found as far north as the Russian River, 80 miles from the release site, or Bean Hollow, 60 miles to the south from the release site. However, prevailing wind plays a huge role in dispersal of floating material. And since the South Bay begins to flood when San Pablo Bay is still ebbing, and you add the enormous amount of flow from the delta, dispersal from the South Bay up into the North Bay, and especially up into Suisun, appears to rely on much rarer events, and it was a much rarer in this case of our drift cards as well. Only a few cards from two North Bay release sites reach the Carquinez Strait, and it's very interesting. These are the very same areas that mark the easternmost infestations that had ever been found, just very tiny occurrences that were quickly eliminated by our treatment program in the early years of the project. So we've now reduced the North Bay propagule pressure so dramatically, but of course, with the movement of the tidal prism up into the delta over the course of with sea level rise, and continued vigilance is certainly warranted, and we still do need to remove the rest of the hybrid Spartina from the Suisun infestation itself. I'm going to skip over that because I'm running low on time. I just want to show a few things of our restoration project. We planted about 40 different sites with over 600,000 Pacific cordgrass, marsh gum plant, and transition zone species, mostly to benefit the Ridgway's rail. We partnered with the Watershed Nursery to learn how to grow these, to harvest small amounts of Spartina foliosa, the native, grow it out in beds, genetically tested Spartina foliosa, and transplant it out into the marsh to reduce impacts to the marsh from borrowing these species. And we've also we partnered with H.D. Harvey and Associates to create high tide refuge islands that are the only things really above the tide at extreme high tides. There's an example of a couple of them here in some of these marshes. So another sea level rise adaptation to try to benefit the rails. And finally, this is a good example of Eden Land and Ecological Reserve in 2011, us planting native cordgrass throughout, and the transformation that happened by 2019 with the first Ridgway's rail in this marsh detected in 2018 and 13 rails detected by 2021. Certainly likely much accelerated by our active planting with no propagule sources nearby to repopulate those areas. And that was a new pond that had just been opened. That was not from Spartina treatment itself, that it was a bare mudflat. And finally, the Alameda Flood Control Channel, a good example, probably our best example of, in the old days, I wish we had drones when we started the project to take better time series, but you can see from 2003 to 2005, the incredible acceleration of hybrid Spartina across this mudflat at the Alameda Flood Control Channel. And then years later, by 2019, after removing the hybrid Spartina and reintroducing the native cordgrass, we have miles of native Spartina foliosa fringing the channel, and that's basically the site has been completely transformed. So most tidal marsh restoration in San Francisco Bay relies on passive native vegetation establishment, and you really need the watchful protection of an entity to make sure with the hybrid present and it growing in this matrix of the native, the watchful protection of the ISP has facilitated these marshes to develop on their own. They're very vulnerable in the early stages when they're open mud flat, when they're open mud, and can be quickly invaded and dominated by the hybrid Spartina. So ongoing stewardship monitoring for hybrid Spartina invasion is really important so we can respond rapidly. And finally, basically, we just want to have a goal of healthy, resilient marshes with thriving native biodiversity, and that requires ongoing stewardship to maintain the health of the systems that are extant, to facilitate restoration, and to look out for new threats that may arise. And that's our website. It's right now hosted on the CALIPSI, California Invasive Plant Council website. We are in the process of redoing our website, so that's a good place to go for information at this time. Thank you.
Okay, we're not going to have any time for questions for that one, but Drew, I hope you're going to stay for lunch so people can come ask you then. Awesome. Okay, next up we have Dr. Virginia Matzek from Santa Clara University.
All right, well, thank you very much. I'm pleased to be talking in this session because it's about connectivity, and this project is really closely related to the idea of connectivity, because it's about whether our treatments of Phragmites and Suisun marsh are changing the potential for further spread within the marsh. And of course, as Laurel mentioned earlier, it's Phragmites invasion is really bad, and not necessarily getting much better, but we're trying to kind of get at what the sources of the continued spread are. And so this is a part of a larger project that was funded by Delta Stewardship Council. Rochelle Tanner is going to speak, I think, about some of it later. And there are other aspects of the project, including follow-up after herbicide treatment with outplanting of native species. And there's a whole social science component where we're trying to figure out the kind of barriers to participation and strategies for communication and coordination in the marsh. But this part is it's actually a student project, an undergraduate project of Santa Clara students that was trying to get kind of a narrower piece of the reasons behind the continued spread. So Phragmites is the common name is common reed, and it was originally native to Eurasia. It's now kind of everywhere. It's famously in Chesapeake Bay. It's actually in Great Salt Lake and a whole bunch of places in the U.S. In Suisun Marsh, our concerns about it are that it impedes navigation in the channels and recreational access, especially in these managed wetlands, the managed ponds. So a lot of Suisun, as you probably know, is managed for duck hunting, and so that duck hunters like open water and kind of fringing vegetation or little islands of vegetation in which to create blinds. And so when Phragmites comes in, it forms these very dense stands, and it also has an effect on the wildlife by crowding out species that provide better food availability for waterfowl. And it really spreads fast when it's not being treated. So these are some older images from Goodyear Slough, which the Phragmites cover is classed in red there. And you can see that just over a period of less than 15 years, it's like almost entirely taken over that site. So it runs away with great speed. In terms of what people are doing for treatment in the marsh, so Suisun Resource Conservation District has been coordinating a cost share program for a couple of decades that engages with the private landowners there to do mainly herbicide treatment. They also do some burning and disking. And keep in mind that these managed wetlands are they're dewatered in the summer, so that it's dry and they can go in and they can burn, and then they reflood them for waterfowl season. So there's a period of time when they can get in there and do vegetation management during the summer. And so this has been going on for quite a while. But marsh-wide, what we see is that is not sufficient to halt the spread or reverse the trend in Phragmites. So this is from the paper that Laurel also showed a little of earlier from Jason Hagani's work. And you can see the tidal wetlands in blue and the managed ponds in orange. And it's just been a steady rise in the total amount of acreage that we see invaded by Phragmites. It looks a little promising there in 2020. Psych, that's actually probably a classification error from the drought. So we don't actually think that it's being reversed there. And so, and as Laurel mentioned, the ability to treat in the tidal marshes is hampered by restrictions on what kinds of chemicals can be used, but even in the managed ponds, it's still going up. Now, on a local scale, Jason's work suggests that the spread of Phragmites is kind of out from clumps. So you can see here that the green is the oldest kind of center of the clump, and then the subsequent years in which Phragmites is detected shows this kind of outward growth from probably from the spread of underground rhizomes. And so that kind of highlights the necessity of really doing a good top, or better than a top kill, but really some kind of control method that gets at the underground part of the plant so it doesn't just resprout. But our interest was actually in spread sort of across the marsh by seed. So we think that a lot of the Phragmites spread to new areas of the marsh comes from seed production, and it has this great big fluffy seed head inflorescence in late summer. And so we're interested in whether any aspect of the current control regime in the marsh is bringing down that aspect of spread. If it's starting to impede the production of seed or the viability of seed, and we were interested in that from any kind of aspect of propagule pressure, whether it's the size of the patch or the physiological response of the plant. Was there any evidence that we were getting lower amounts of seed production viability, or to hamper the continuation? And then our other interest was in the possibility of herbicide resistance. So some of these have been treated continuously for 20 years, and we would certainly expect that over time you could develop herbicide resistance. And so what I'm showing you here is a patch that had been hit by the sprayer it's got some green leaves in it. And I'm not saying that at all that that's evidence of herbicide resistance. It's possible they were just missed by the sprayer, but we see this pretty commonly in the patches that they still have green after the herbicide treatment has come through. So I had a couple of students, Gabe Rodkey and Michael Weatherford, seniors at Santa Clara University, who were interested in taking on this project. They were working on the outplanting, which is what you see here. And they did some really nice work, very difficult work to get at the answers to these questions. So our first was, in the places where there's been intensive control of Phragmites, is there a diminution of the potential for spread by seed? And conversely, is there any evidence that that's starting to create herbicide resistance? And so our experimental approach was to use data from Suisun RCD and the cost share program to try to divide the privately owned parcels, and actually the publicly owned ones as well, into to be characterized as either very low intensity of management, so generally no management at all in the last 10 years, or high intensity management, so continued strong attempts at herbicide or other kinds of control methods. And I have to say that this is probably the trickiest part of the whole project, and the possible weakness in it is that we didn't necessarily have great information for this. So we had information from SRCD, but if landowners were controlling Phragmites but were not participating in the cost share program, then we didn't necessarily know that. And we also had trouble when we were out looking for Phragmites patches that had been heavily treated. What we mainly knew was that the parcel had been treated, but we didn't necessarily know if the treatment had been over the entire parcel, and that every possible patch that we were looking at had in fact been, a target of control for 10 or more years. So some difficulty there that we couldn't find a methodological way around. So when we characterized the parcels as either high or low management intensity, we located 5 patches of Phragmites, and each of those had inflorescences, and then we collected the inflorescences when they were ripe in late August of 2022 under an blistering heat. Oh my God, I'm not a wetlands person to begin with, and there's no cover out there. There's no shade. It's really, really hard to do. I will not be joining the Nutria project, just want to let you know. But anyway, went out and got these nice ripe inflorescences. We also measured the patch size and the density of inflorescences because we're interested in whether the way that treatment was affecting it was actually to make the patches smaller or to make them sparser in terms of how many inflorescences they produced. And then we brought these inflorescences back to the lab and we started counting. Oh, the seeds are really, 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 really tiny. So stripping, weighing, counting the spikelets is quite tedious. You can see a couple of pictures there of Phragmites seeds for scale. You can also see that whole weigh boat only weighs about 1 gram. So it's very tedious work, which is why you get undergrads to do it, because they will work for minimum wage and pizza. And so we counted all the things. We counted seeds per spikelet. We counted the spikelets on the inflorescence, or aliquots of them. We counted inflorescences per meter squared, and then the meter squared of Phragmites in the high and low intensity areas. And the reason for doing all of these things was we wanted to multiply them together to get this measure of propagule pressure. So by doing that, and what I mean by propagule pressure is sort of a measure of the threat of invasion from seed in the marsh. So we're sort of multiplying up. And then these are our principal results. So what you see here is that in the places that have not had very much Phragmites control are in teal or late blue-green, whatever that color is. And the places where there has been a long history of control are in dark blue. And so you can see that our sites where there's been no or little management are producing quite a bit more seed per meter squared of marsh. And so some other kind of diving into why this is, we looked at whether the inflorescences were smaller, and they were in the high treatment area, and there were also fewer of them per patch. So inflorescences were sparser and smaller, and there were fewer patches because of the treatment. So the major driver of the trend was actually the fact that just the control has diminished the number of patches, but even within the patches, fewer and smaller inflorescences. We also wanted to look at germinability. So we're thinking here about more the physiology of the plant and whether the control had any effect on viability of the seed. And so we took the seeds and we cold treated them to break dormancy and then germinated them in growth chambers to test that. And here is this is a very close-up picture of somebody's hand with a couple of germinating Phragmites seeds. Kind of amazing, isn't it, that such a large plant grows from such a small beginning. We could not, in fact, detect any statistical difference in the germination rate as a result of the history of control. And then we took those germinable seeds and we planted them out in the greenhouse and we grew them up tenderly and caringly as baby Phragmites plants, and then we tried to kill them. So we used herbicide on them for our herbicide resistance test. And we visually assessed them on a 0 to 5, so a 6-point damage scale, which is pretty common in the literature for just sort of trying to be as quantitative as possible about how well the herbicide acts. And so we took the highest emergent blade from each tiller and cut it or applied herbicide there, cut the rest of the plant, and then we looked at whether it had completely knocked it out or only partially killed it, so on. And the good news is that again, we could not find any statistical evidence that the heavily treated Phragmites is developing or any induced resistance. So I'm going to finish up there with our conclusions that generally the Phragmites control is working. It's the propagule pressure is diminished in the areas of the marsh where it's being heavily treated. This has less to do with the physiologic response of the plant and more to do with the fact that the patches are just smaller and less dense. But that's kind of not the best news, because what it also highlights is that there's a lot of places where people aren't treating Phragmites at all, and that's probably the source of the continued spread of the continued increase in size in the invaded area. A long history of treatment also did not have any effect on germinability and was not seeming to induce herbicide resistance. So I would like to thank very much, first of all, Delta Stewardship Council for funding this work, and Suisun RCD for our major partner on the ground there, Jason Hagani for his work in helping us work with maps. John Takakawa did all of the interaction with landowners to get us out on these privately owned parcels. Fish and Wildlife was helpful in giving us access, and all of the private landowners who let us ramble around their land and ask nosy questions were also gratefully acknowledging. And of course, Michael and Gabe, who did their sampling in the hottest part of the year and toiled many hours in the lab and greenhouse to get you these answers, which I'd be happy to answer some questions about if there's time. Thank you.
And state name and affiliation for people online.
Roland Brady, Sacramento Area Creeks Council. What do the landowners think of Phragmites?
So I would say the ones who are really into stewardship and vegetation management are trying to get rid of it. They see it as a pest, and they also, they have a lot of other vegetation management issues to come up. So for instance, Russian thistle is now kind of a big deal in the marsh too. And then there are, parcel owners that just they're just not as actively managing their land. So I wouldn't say that it was universal that the Suisun landowners are engaged with control and, seeing it as a pest, but the ones who are maybe most active in stewardship.
Hi, thank you for the presentation. Amy Williams from Yola County RCD. I'm wondering what herbicide you used to test for herbicide resistance.
Glyphosate.
Okay.
Hi, Ted Gross holds snacks and lunch for actually University of California, Davis. I'm wanting to know whether either from your work or others, whether there's anything known about how far the seeds may go. I mean, genetic data that sort of looked at neighborhoods of this. Do you have a sense of how far these things may get transported?
That's a good question that I don't know the answer to. I do know that the work that's been done on genetic variability within, invasions has found that where there is more genetic variability in patches, they seem to be more robust and possibly to spread farther, but I don't know.
Are there questions online? Nothing? Anybody else in the room? Oh, somebody came in.
Yeah, Wes Schwang had a question. Why is glyphosate still used? Are there any other alternatives?
So we use glyphosate because that's what is being used, and I have to say that I don't know what that decision-making process is. It's what the SRCD has been recommending to landowners and what the pesticide applicators use, given the I think maybe the dryness of the and the timing of the control. But sorry, again, a question I can't answer.
We still have like 2 minutes for questions, so anybody else?
Yeah, but this time you have to ask one that I can answer. This is getting kind of embarrassing.
Chad Ockery, Westervelt Ecological Services. Was just wondering if maybe ecologically the amount of seed being put out isn't limiting because there's enough seed to occupy any areas that could be colonized. So there's still enough seed out there. I don't know. I'm just wondering about that.
Yeah, that seems entirely possible. That even though we can detect a pretty significant reduction in the total amount, whether that's biologically significant, whether that's actually helping or not, is a question beyond the scope of this study and would be really interesting to try to find out.
Hi, this is Drew Kerr, Spartana Project. I just wondered what the literature says on seed viability length, the length of time the seed's viable for Phragmites.
Yeah, so I think only for a couple of years. I don't think it has a very persistent seed bank.
Hi, I'm Louise Conrad from Department of Water Resources. Thanks for your talk. I am wondering if your research suggests any critical patch sizes that should be prioritized for treatment, given that patch size seems like it played a role in some of your in the results about propagule pressure.
Yeah, that's a really good question, because I think one strategy might be to say let's try to have the most local eradication. If we have something that is kind of on its last legs, try to knock out that patch, as opposed to tackling areas that are more densely infested. But I would also say that the landowners are getting, the ones who have been really vigilant are getting to the point of it's almost hard to find Phragmites on their land anymore. And yet then there's, Grizzly Island Wildlife Area that's just got vast quantities of it. The tidal marshes are full of it. And so I would say that patch size might be less important than trying to tackle whatever policy and permitting barriers there are to working with those denser areas. That would probably yield more. Thank you so much.
Okay, our next speaker is virtual, Carolyn Teepolt from the Woods Hole Oceanographic Institute. Give us just 1 sec to get the technology right.
Recording again, so I'm assuming things are good.
All right, thank you so much. I'm really excited to virtually be there. Wish I could be there in person. And I'm going to zoom us out to that kind of highest level of sort of global connectivity in Laurel's intro slide, and talk about genomic tools and some of the work my lab is doing to use them to look at connectivity and spread of invasive species from San Francisco Bay and beyond, up and down the coast. Next. So as I said, I work a lot with genomic data and population genomic data, which is just differences, looking at differences between individuals within a species and how they're distributed across the landscape. And this can give us some interesting information about the recent history of species. Next. So in terms of invasions, it can help us trace new populations to their sources. Next. It can help us estimate dispersal among regions by looking at how genetic data is shared among populations. Next. And it can tell us something about genetic diversity in populations. Next. And finally, it can tell us something about adaptation across the landscape. And this last question isn't something I'm going to talk about today, but it's something that my lab thinks a lot about. So if you have thoughts, I'd love to talk to you about it later. Next. And a very, very brief history of population genetics methods effectively over the past couple of decades. Things have been getting more and more interesting with population genetics. We're going from being able to look at just one place in the genome with a great deal of effort to being able to look across entire genomes. Next. And that's kind of similar to me to this progression of video game graphics, right? So, even with these single gene approaches, you're still, you're always seeing underlying structure, you're seeing what's going on, but the higher and higher resolution the approaches get, the better and better a view you have of what's happening. So you can go from, asking questions like, does Mario have a mustache in the 80s to, how many little hairs are in Mario's mustache? Next. And just very briefly, all the data I'm going to show you today are from single nucleotide polymorphisms, or SNPs. And these are just regions of the genome where individuals vary by a single base pair, an A, T, C, or a G. So it's the smallest unit of genetic differentiation, which on its own doesn't give you a lot of information, but when you look at hundreds, thousands, or even millions of them across the genome, it actually gives you a really beautiful high-resolution view of what's going on. Next. And of course, you probably don't need to show you guys the slides, but San Francisco Bay is really considered to be a hotspot for marine invasions and kind of a beachhead for further spread up and down the West Coast. So on the left, I have the classic Cohen and Carleton figure showing accelerating invasions in San Francisco Bay up until about 1990. And on the right, from a paper by Greg Ruiz, just showing that of non-Indigenous species, marine species on the West Coast, the first record for many of them is San Francisco Bay, which may partly be some observation bias, but is likely also because the bay is such a hub for all kinds of connectivity with other shores, and it's such a large and diverse set of ecosystems. So a good place for species to be introduced in and for them to do well in, and to kind of give them a foothold to expand outward. Next. So I want to talk today about what genomics can tell us about marine non-Indigenous species spread and dispersal from San Francisco Bay and connectivity with the bay and other regions. Next. And I'm going to do this by telling you about some of the work in my lab on two different invasive species, both of which have spread from San Francisco Bay, but which have very different likely dispersal mechanisms. So on one end of the spectrum, we have a tunicate, a biofouler, Bacillus schlosseri, and on the other have the European green crab. So if you think about some of the many ways marine invasive species can get around, two of the big ones are larval dispersal and anthropogenic transport. So in terms of larval dispersal, Bacillus does a terrible job. These little tadpole larvae settle after about 4 hours max, so they're not going very far at all on their own. Whereas green crabs are fantastic at this, with about 2 months in the plankton. And larval dispersal of currents, particularly in El Nino years, appears to have been a major vector for green crab spread up the coast. Of course, there's also anthropogenic transport for green crabs that works well from one region to another on a large scale, but it's likely not likely to be a major factor in moving green crabs up and down within the West Coast. By contrast, Bacillus, of course, once it settles, loses that cute little tail. It just basically grows on anything you leave in the ocean for too long: the bottoms of boats, rocks, ropes, aquaculture gear, other marine infrastructure. And so adult colonies are very easy to be moved around with that kind of a vector. Oh, next. Sorry, I forgot that I don't have the power over my slides. So the first vignette I'm going to tell you about is this golden star tunicate, Bacillus schlosseri, our very minimally larvally dispersing species. And all of this work was led by my grad student, Zach Tobias, as part of his PhD. Next. And Petrellus has a long history of introduction and spread to the point where we know where it's invasive to some extent, but we don't actually know where it's native. But it is definitely invasive on the West Coast. Next. And generally with population genetics, we start at the largest spatial scale and then we drill down. And so that's how I'm going to present the data to you, is to show you kind of all of our sites first, and then focus more and more in on what's going on in California and San Francisco Bay. So for this project, Zach actually has sites on both coasts, 12 on each coast. Next. And the West Coast, where we're really interested, here are the sites again, with a little bit of the history of spread. The first record was from the Mare Island Naval Shipyard in 1947. After that, Petrellus was found in the 60s, down in Southern California, the 70s in Oregon, 80s and 90s up in Washington and British Columbia, and in the last 20 years up in Southeast Alaska. Next. And so without further ado, let's dive into some of the data. Most of the data I'm going to show you today look like this. They're principal components analyses. So each dot is one individual animal, and it's colored based on the site where it was collected. And, you know, like any PCA, the closer two dots are in two-dimensional space, the more closely related they are, in this case, genetically. And so the big thing, I hope that jumps out to you here, is that we have 2 coasts, but 3 major groups of populations. Next. And those correspond to the East Coast, the West Coast from San Francisco Bay up to Alaska, and Southern California. Next. So genetics is already telling us that there were multiple introductions to California, and those well, even though I'm picking on the Bay as a source of West Coast invasions, it's not to blame for those Southern California populations. They're very clearly derived from a different introduction. But San Francisco probably is to blame for the more northern spread of the species. So now we're going to refocus, and I'm going to show you some analysis focusing just on that central northern Pacific, northern West Coast cluster. Next. And when we do that, when we drill down further, we see more structure, right? Now we're picking up this somewhat finer scale structure in the West Coast, but it's still pretty distinct. And you can see there's kind of 4 major groups. Next. And those correspond to Central California, which really is San Francisco Bay in this case, and Oregon. Oh, previous, please. Seattle, all the British Columbia sites, and the 2 Alaska sites. And one thing I want to just note quickly here is that with Petrellus, which is likely moved mostly anthropogenically, you can see some of the impact of potential differences in those movement patterns by looking at those British Columbia sites. And you notice that we have 2 way up in the north of British Columbia, 2 way in the south, but yet genetically, those populations are most closely related to each other than they are to the American populations just on either side of the border, which is likely linked to a lot more transport of gear within British Columbia than outside of it. So now we're going to zoom in on that Central California, Oregon site next, or cluster, I should say. And when we do this and we reanalyze those data, you can see that there's even more separation in the data. So even Richmond and San Francisco, just 15 kilometers apart, have really distinct and different Petrellus population patterns, which we found really fascinating and kind of promising for future work. Next. So with Petrellus, what we're finding is that there were at least 2 distinct introductions to the West Coast. And I say at least because, of course, we didn't sample everywhere that Petrellus is found on the West Coast, so I can only speak to the sites that we were able to look at. And that populations are really distinct on very small spatial scales, at least 15 kilometers apart, maybe less. We just haven't looked yet. And we find this really exciting because it suggests that we might be able to use this system or systems like it to trace introductions to a really small source area potentially, which might be helpful for targeting management and understanding spread more generally. We're kind of toying with the idea that you could pull some Petrellus off a boat and actually figure out where it came from, which I think would be really neat. Next. So moving on to the green crab with its robust 60-day larval duration. Next. Green crabs have actually been a real challenge, green crabs on the West Coast, for traditional genetic approaches, because this population is really not very genetically diverse. It's passed through two bottlenecks. We know this introduction pathway through prior genetic work by Joe Roman, from the native Europe to the East Coast, losing genetic diversity in that transfer, and then from the East to the West Coast, losing genetic diversity again. So CO1 sequencing doesn't give us anything because everyone on the West Coast has the same sequence. But luckily, when we use some of these higher resolution approaches, looking across more of the genome, we can start to get some clarity. Next. And in fact, the first record of green crabs on the West Coast is not in San Francisco Bay, but it's believed to be a bit of a one-off and not the source of the rest of the populations, which are believed to derive from that second report from Redwood City, introduced in 1990. Next. Or I should say, not introduced, first recorded in 1990. When we look at spread from that start in the 80s or 90s, you can see that green crabs have been extremely effective at moving their way up the coast. They made it to some neighboring bayments in California in the early 90s, 97 to Oregon, and 97, 98, they really spread very far up into Vancouver Island with the help of a big El Nino Southern Oscillation event, which kind of increases or boosts this nearshore northward current and makes kind of a nice conveyor belt, unfortunately, for green crab larvae. And I should say that they are now found as far north as of last year as Metlakatla, Alaska, but we're going to focus a little bit further south for this talk. Next. So what do we see with genetic data with green crabs? Well, despite this really severe bottlenecking, we still see some genetic structure, but you might notice that this is a little bit different than what we saw with Botrytis, because we basically see all of our sites, except the ones I have hopefully helpfully colored in gray, piling up right on top of each other. And we're going to zoom in a little while, but unlike Botrytis, when we zoom in on this, we're not going to see any more structure. This is generally representative of really high connectivity among sites up and down the coast. The exceptions are these two sites in gray. One is Souk Basin, which is in southeastern Vancouver Island, and the other is Seadrift Lagoon, which is not that far outside of San Francisco Bay. And I want to point out that unlike that Southern California Botrytis introduction, these were not caused by a new introduction from outside the range. In fact, next. If we look at genetic diversity, both of these sites contain a subset of the diversity we see in the West Coast as a whole. So effectively, they have now gone through a third bottleneck, losing even more genetic diversity and giving them those distinct population signatures. And I don't have time to go into this in too much detail, but looking at the oceanography, both of these sites are likely really isolated from the influx of larvae from outside. So we think that essentially crabs got in initially, there's been no inflow of new larvae, and so these populations have bottlenecked and diverged. Next. Like I said, other than these sites, we really don't see any structure, and that's indicative of these kind of large-scale dispersal of larvae from one embayment to another. There just doesn't seem to be any genetic diversity or any genetic differentiation, rather, to pick up from these sites. And that tracks with some of what we know from the spread of green crabs. We know that they spread hundreds of kilometers in that 97, 98 year in just that 1 year, indicating really strong dispersal to the north. And next slide. We also have, I anticipated myself. These two sites are isolated. I already talked about that. Next. But when we look at Souk, while larvae don't seem to be coming in, which leads to that distinct signature, they are getting out. So we can use that population as a tracer of how far individual larvae may be traveling, and as an indication that these larvae are also not just moving north, but also moving south down the coast. So the individuals I've circled, which were found in embayments in Washington and Oregon, have that Souk genetic signature. So likely traveled out of Souk, down the coast, about 400 kilometers coastwide at the coast, along the coast at the maximum to get there. Next. And just, this is evidence. There's some hybrids that have one parent from this larger outer coast cluster and one from Souk. So this is evidence of them settling down and reproducing with local crabs. And while I realize that this group is very interested in the bay, and obviously this is much further north, this distinct signature lets us trace this population in a way that we can't necessarily from other sites. And it's just a general indication of a lot of exchange of larvae to the north and the south regularly, which I realize is kind of a bummer for management because it means that if you're interested in managing your local green crab population, you really have to think at a much larger spatial scale. Next. So just zooming in quickly, when we do this and look just at California sites, Seadrift Lagoon remains really distinct. And that's true even if you look at multiple generations. So this is a stable signature over time. But like I said, there's no more structure in the data to be pulled out. All of these California sites fall in the same cluster. Next. Next. And interestingly, I have some samples from Vallisneria Lagoon and from Seadrift Lagoon, which is that weird artificial kind of frontage lagoon. And despite the fact that there's literally about maybe 50 feet separating these in places, there doesn't seem to be much genetic exchange between them. I'm not picking up any signatures that would suggest that there's a lot of exchange. We think this is because Seadrift is basically connected with Vallisneria via a pipe, and the water level can be controlled. And in talking to folks there, it sounds like essentially it just happens that when the larvae are in the water, they tend to close the pipe. And amazingly, that seems to be enough to maintain this really distinct genetic signature. Next. So the green crabs have spread over 2,000 kilometers in under 35 years from a single San Francisco Bay source. And they're traveling extensively up and down the coast into and out of San Francisco Bay. But currents are really important to this. You notice we didn't see a separate pattern with British Columbia. That's because ocean currents are not big respecters of national boundaries. But these isolated populations can diverge really quickly, which tells us something about connectivity itself. Next. I realize I'm running a little long. Broadly, I'm really interested in using these genomic data as part of broader understanding of a system. So I've been lucky enough to work a lot with managers and ecologists and other folks working on the ground. And this is something I'd love to continue. So if you think that this type of data could be interesting or useful to your management, I'd love to talk more. Next. And a very quick pitch, if you know of any interesting jobs coming up in the San Diego market in the next little while, Zach is going to be defending in March. He's fantastic, and he's looking to move back to his home, his kind of birthplace in San Diego. Next. And finally, a very quick thank you to everyone involved in this, especially all the folks who've collaborated for sample collection and our major funders, NSF, Washington Department of Fish and Wildlife, and the North Pacific Research Board. Next. And if I have time, I'm happy to take questions. If I don't have time, I'm happy to field questions, via email or anything like that later. Thank you very much.
Is there any natural predators for the green crab, like otters or any other thing that might also help to stem the tide of them?
Yeah, so there's some really interesting research that Ricky Jeppesen at the Elkhorn Slough NER has been doing, where they're looking at the kind of increase in the otter population down there, and they think that the otters are keeping the numbers down pretty considerably in Elkhorn Slough. So that's one thing. Other than that, pretty much I think anything that can eat a crab that size will be happy to take it, like fish and birds. We often find them pretty damaged. But they do reach some pretty astonishing numbers in some places, so it doesn't seem to be enough, unfortunately.
Based on the pattern of longshore drift from sediment dispersion, it's pretty consistently along the west coast of North America, north to south, yet we're seeing this progressive migration of the green crab from south to north. Can you comment on that?
Yeah, so it tends to, so the initial spread was pretty episodic and corresponded pretty nicely with El Nino years. The larvae are in the water column during sort of the winter, generally speaking. And so the going assumption is that the Davidson current is enhanced in those years and is transporting those larvae north. Of course, that's not universal. We do see that southern transport. That's a different phenomenon with a relaxation of that kind of outflow of water from the Strait of Juan de Fuca. And so that kind of reverses the flow and lets the further to the north allows for some more dispersal towards the south. I should say I am not an oceanographer, but I'm working with some folks more on the more up in the Salish Sea area. And it is really interesting how the oceanography and the genomics complement each other really well.
That looks like it, Carolyn. Thank you so much. And
Right.
we'll see everyone else up here at 10:50.
Thank you so much. Bye.
Everybody, welcome back. Great. Sorry, that was a short one. My name is Rachel Klopfenstein. I'm a program manager at the Delta Science program. Really excited to introduce this next session, session 2, which is all about connectivity between research, management, and policy. And we have a really great lineup of presenters. I'm really excited to introduce all of them. And we also, this session is going to be followed by a panel discussion. So all the speakers are going to come up for each of their presentations. There'll be time for questions. And then we're going to invite all of them up and have a panel discussion up here so we can ask more questions of them and have the panel's perspective on this topic of connectivity between, sorry, something popped up here. Again, research, management, and policy. So our first presenter is Dr. Ted Groschultz, who is the Distinguished Professor and Alexander and Elizabeth Swan Specialist in Cooperative Extension at the University of California, Davis. And Ted's going to be sharing how to better collaborate to see more successful invasive species management outcomes and kind of give a little reflection on a amazing career. And so come on up, Ted.
So just to start, I'd like to thank the two Rachels for being invited here. I think they just wanted me to provide the meals. That's the only reason I'm participating here, but it's great. Just a note about the title. This is a terrible title. I actually, if I was running the meeting, I would have rejected this title. We'll skip past it. So thank you for not. I want to start with a paper that I wrote with my unfortunately deceased colleague, Susan Williams, wonderful colleague, really tragic that her career was ended early in an accident. And just some reflections on where we were about 15 years ago, thinking about the science and management of invasive species. So sort of looking backwards. So this talk will be a bit of an historical look back and a look forward to where we are. And then some ideas for where to go in the future and some suggestions about what I think we can do that's positive in bringing science and management together. So looking back at that paper, a lot of it was about sort of basic gaps in science. So scientists needed to provide everything from life histories to how things got around. So there's a lot of need for that at that point. However, we also need to be a little bit more cognizant of the regulatory framework. A lot of my colleagues really didn't understand who was managing and who had authorities back then. At that point, there was really a lack of federal agency support, and the Aquatic Nuisance Species Task Force, well, which was still poorly funded and slow to act, was there with their 6 regional panels. So there was sort of this federal overstructure that just didn't have the resources to do what they needed to do. NISP was very overextended and really not really well represented with aquatic systems. They were very terrestrially focused. Now, of course, we had evolving databases from USGS, CNAS, CERCS, Nemesis, et cetera. A lot of things were getting going, but that's kind of where we were at that point 15 years ago. Whoops, hit the wrong button. Don't do that. So recommendations for research at that point were things like, let's start focusing on community and ecosystems. Let's not just look at species-specific impacts. So more towards thinking about trait-based prevention programs. So can we predict new invaders based on a suite of characteristics? Use of species distribution models, which of course are now environmental species models. So we've gone back and forth with how to predict where things may end up based on environmental characteristics. We focus on early detection. Interestingly, not really rapid response with it, but still using genetic methods of the type that Carolyn just talked about for finding out when things are showing up. Better methods for risk assessment, understanding connectivity. Yay, that's part of what we're doing here. Possible biocontrol where feasible. We can talk about that more and focus of economic models, and then climate change and introduction. So these are the kinds of things that we thought about 15 years ago. Yes, many of these are still on the list. So what are the obstacles now? Okay, well, this is not meant to be a session of complaints. I will talk about these and then move forward with positive ways to think about these and make progress. There's still a lack of focus. The Aquatic Nuisance Species Task Force, which I work with very closely, trying to rewrite outdated aquatic nuisance species management plans. I'll take full responsibility for the new green crab one, so I know that that's ineffective. There's still regional panels that are still dividing up the same small pot of money. NISP was absolutely been pretty absent for the last several years, are now back on track. So we've still struggled with that sort of federal level of oversight at the state level. So what we need really is more progress on addressing non-ship-based pathways. So Chris and others can talk about ballast water. We've made progress on hull fouling quite a bit. We still have not made any progress on live bait, seafood, aquarium trade. Where did caulerpa come from? Lots of things that we know. That's where green crabs came from. So we still have much to do in this area. There's very little coordination at the state level. Who remembers CASE? Is anyone old enough to remember that? The 2009 California Aquatic Invasive Species Management Plan, written by CDFW, which actually we wrote first and then handed off to Karen. But that actually tasked the California agency's Aquatic Invasive Species team, CASE. Throughout all of that, CASE never came to existence. It was sort of mandated, but there was no money put forth, so it never happened. So we never got this statewide organization that could have done that. Now, you can see the number of agencies, everyone's here, CDFW, DPW. You know, what we're missing, of course, is that. Now, let's look at two. We have CISAC, which is the California Invasive Species Advisory Committee, and I'm the current chair, so I can apologize for that. We have very little aquatic focus at all. Basically it's a CDFA focused organization. But there is DIST, and this is a wonderful organization, but imagine this was statewide focus, and I actually have an acronym ready for this, CISCO, the California Interagency Invasive Species Collaborative Organization. Imagine what this does statewide in Southern California and Humboldt. So let's take that and make that a statewide, but we don't have that at this point. So a few other things that we're missing are basically the inability to sort of mandate or prohibit things like a whole genus of caulerpa or spartina, because we have now a new bill which basically did just that. But prior to that, we'd eradicated, Taxifolia mediterranean strain, and suddenly prolifera shows up. I mean, so it's just going to be one after the other. And I can point to several other examples. So we really need to do that if we can. Okay, current other obstacles are basically standardization of regulations, even high-profile species. Green crabs, which are actually prohibited in Washington, are actually for sale live in places like Long Beach. You can go down there and there's big bins of crawling green crabs, and the Long Beach market for just to buy and take home and stick in a bag. So there's no regulations regarding these kinds of situations. So I think this is certainly something that we can make progress in the future. Okay, connecting science to management. It's a duality. It's not just scientists needing to sort of provide information from managers. Managers need to provide information to scientists because, we sit in our, areas and don't pay attention. So there really needs to be a lot of communication about, for instance, management levers. I mean, so it's very easy for the academics to say, okay, this is what you should do. It's like, well, we can't do that. We only can do certain numbers of things. We can perhaps do things like change, harvest. We can maybe change the timing of the pumps. We could possibly stick out, Dicotinia weevils instead of spraying. We could open and close the salinity gates. I mean, there's a certain number of things that can be done. But academics, my colleagues, don't really know what those are. Do I sound like Ted Summer yet? So I think it's an important thing to be able to do is to communicate what those levers are. So there can be much more clever thinking about that. They need to bring scientists and managers need to work together in the early discussions of proposals and where to go. Obviously, everyone in the room knows this, but I think that's going to not only determine how we do, but what the questions are. And I know a lot of my colleagues need to be kicked in the shins and say, well, gee, that's not the kind of work that I do. But it's like, geez, learn how to do it. Get involved, like get a collaborator, get an agency partner, start doing that work that's needed. I started working on freshwater floodplains. I was a benthic marine ecologist. I didn't know anything about it, but I learned, and I was valuable to some degree in that. So I think part of that is getting people to move out of what they typically do in order to answer these kinds of questions. Okay, and lastly, of course, understand the form of information. So don't just assume a published paper is going to fix things. You need to understand what the decision process is and how to aid those decisions. Is a decision tree needed? Is some sort of numerical model where you can plug in information needed? Let's talk about what will actually aid the decision process rather than just dumping off information. Okay, so that's my last thought about that, but I think those are some of the things that we need to make progress on. So 15 years in the past, we're still struggling with those issues today. Okay, so some modeling approaches or new approaches to managing AIS. So for some of you have heard me rant on about this, so I'm kind of still continue to push this idea. Some approach that Steph Green and myself published in 2021 addresses sort of this dilemma that we all have is like, well, what if eradication doesn't work? Well, as you can see in the top things, eradication is likely to succeed if you have something that's geographically limited, small population size, easily and readily accessible, the invaders easily identified. You can imagine that being a little nutria stuck on the top of the fence or something like that. But however, the systems that we work in, especially in the Delta, are challenging. I mean, it's really difficult to even get into the place, let alone see what's under the water, see where things are. So eradication is very challenging. So what we've been advocating, and we'll take some questions on this at the end, is moving away from eradication. Yes, moving away from eradication. Now, the idea is that management and science need to come together to develop alternatives if you declare that, well, we just can't do it realistically. So what we've done is work with managers, ANS managers, to figure out, well, what is it that you want to accomplish and provide some tools to allow them to do that. So the idea is to create useful quantitative targets for reduction that will provide positive benefits for the species of interest, the threatened endangered species, the habitats, whatever it is that the invader is actually negatively impacting. So in the course of our work, what we did was we surveyed a number of ANS managers, almost 250 across the US and Canada. And you can see these little colorful graphs. They're a little hard to see, but the yellow bars are eradication, the red and orange are control or other sorts of containment. Basically, most managers, especially in these estuary marine systems, are not looking to eradicate. Most are saying we're past that, we need to do something else. And what we also found that most of these managers have two kinds of data that are critical for developing these guidelines. They have monitoring data, so they know how much the invader is there. That seems pretty obvious. But many of them, in most of the marine systems, have data on what's being affected, what's at risk, what's going down while the invader is going up. That's all you need. So many managers actually have these data in hand. Well, the problem is that with this goal of suppression, we're often being undertaken with no quantitative goals, like more is better, where we're just going to go get rid of as many as we think we can undertake with the resources we have. And what we're suggesting is just a basic plot of abundance and impact can help develop a specific target, a useful target in the future. And the point of this is most of these abundance impact relationships are nonlinear. So they look like this. I hope this can be seen. I don't have a pointer, but I will point out that this is a very general relationship. Oh boy, okay. So what I'm going to say is that it's this non-linear relationship. Oh, for the question period, yeah. So the point is, you see the blue box. What it means is you don't need to harvest all the green crabs to get the benefit. So the shellfish biomass is basically recovered at that 10 to 20 number, and that could be any invader abundance and any response variable. Look at that paper by Bethany Bradley and PNAS to convince yourself that that's a very general result. We did this for green crabs. We did this for rusty crayfish. It could be done for red swamp crayfish, lionfish. So it's basically setting goals, finding this relationship, and then using these thresholds as a way of coming out of having a good target to get back what it is that you want. Carolyn just covered this. So what I'd like to sort of let you finish with is actually this is being used by managers. So Washington State spending many millions of dollars to manage and mitigate green crab impacts. So both WDFW, DFO now have functional eradication, which means you're eradicating the impact of the function. You're not getting rid of all the green crabs. This is what they use now. This is their guidepost. And I suggest this is a useful guidepost for many species in the Delta also. All I'm going to say here is we've been able to maintain the population in this one site, sea drift, which Carolyn told you about. I can answer questions. But this is the point that I'd really like to make, is maintaining this kind of functional eradication requires ongoing work. That's everyone's saying, well, you got to continue in the future. You may be doing this anyway. If it's a high profile species in a high profile location, you can get volunteers to do this. And what I'm going to suggest is this is a way forward for bringing science and management together. Community scientists and volunteers. I have a question. Has anyone have heard of community management? No, we hear community science or citizen science. I'm going to suggest that community management, which should be a community science and management. This is how you increase capacity. This is how you get the public involved. So let's think about community science and community management together. Okay, I've got some conclusions, which are basically just saying that you can't really get the last one. It's really expensive. And then if you eradicate it, they're going to show up again. We've seen this with Caulerpa. We've seen this with Ascophyllum. We see, you know, Oriental fruit fly gets introduced every year on time, right? It gets happened. So eradication may not be the best way forward anyway. And this provides you a way of getting to goals at a reasonable cost and effort. Okay, just acknowledge lots of folks and I'll stop there. Thanks.
Thank you so much for that. I'm still struggling to formulate what I want to ask because you've just talked about this functional eradication, but I think the essence of what I want to know is for something like when you have a functional eradication goal of let's get down to 10 or fewer green crabs on catch per unit effort, then I see how the monitoring of the functional eradication goal and the eradication itself or the control is the same thing, right? You're catching them out.
Yes, yeah.
Right. But can you think a little bit about how this would work with other species where I could imagine the idea would be that like monitoring for whether you're at a low enough, you know, controlled population would be itself an expense. It's not the same thing as doing the work of control.
Well, see, you're measuring Phragmites, right? So you're already getting Phragmites abundance. You probably have that x-axis already, right? You know what the changing abundance is of Phragmites. Aren't you also measuring the abundance of native plants too at the same time? Isn't that part and parcel of what people are doing? Because that's what's among the things being impacted, right? Native plants by Phragmites.
I guess what I'm saying is the remote sensing work that is getting that x-axis is not actually controlling the plant, right? So
no, that's right. No, you don't need to control it. You can just get that x-axis and the y-axis. You can create that plot and then make the decision to control based on that. Yeah, you don't have to do it in the same action. Yep. Yes. Person in gray.
Do we have any? Oh yeah, one more question in the room. Get my steps in. Do you think that with the nutria in the delta that could have been what happened and why we're seeing them again since 2017, that last one wasn't actually the last one and that's why they came back?
I hope no one in the room is responsible for that 1976 eradication. I'll say, I think so,
yeah.
But I think that's the issue. So the question would be, do we need to get every last one? And I think if you plotted nutria abundance versus, you know, the sort of destruction based on, say, you know, Chesapeake Bay data, those probably data are already there. You probably can figure out, well, we need to get nutria down to some level that's affordable. We may not need to spend the entire CDFW budget getting the last one, but that's one view. Sorry.
All right, thanks all. I think we'll hop on to our next talks. One sec. Okay, great. So our next presenter is Brad Henderson from the Cutting the Green Tape Environmental he's a program manager at the Cutting the Green Tape program for the California Department of Fish and Wildlife. And I think this will be exciting for folks who have done any invasive species work on the ground. Permitting regulations can always be a challenge. So this will be all about how to improve what's going on to help improve those processes for restoration and for invasive species control.
Hi, everybody. It's great to be here. Yeah, regulations and permitting. All this great work goes into designing these projects, doing the science, getting these projects on deck to be implemented, and then there's the wait, right? So really what this talk is about are how the Department of Fish and Wildlife, along with many other agencies, are working really hard to increase pace and scale by working through improvements in both approaches and kind of our outlook, really, as well as the actual tools to permit invasive species projects, which either are standalone and will lead to enhanced, recovered, restored habitat, or invasive species activities as part of a larger project for restoration. So let's get started. I'm Brad Henderson. I have the pleasure of leading our statewide Cutting the Green Tape team with the California Department of Fish and Wildlife. We just celebrated our 2-year anniversary on November 1st. And so we're still pretty new. And so what are we about? Our Cutting the Green Tape program is really designed at its core to increase the pace and scale of habitat restoration. And we do that in 3 different ways. It's through grant funding. I'm actually within our watershed restoration grants branch. That's a part of the department that hands out money via Prop 1, Prop 68, and recently a bunch of general fund money that we got. And we're trying to improve those processes because those were getting pretty bureaucratic over time as well. So that's one piece. Another is the permitting, as well as the California Environmental Quality Act, aka CEQA, and we administer a couple of new permitting solutions as well as a CEQA exemption. And then really the most fun part is just coordinating with restoration practitioners, scientists, volunteers, local governments, federal government staff, all those kinds of things. It's so much fun. Love working on restoration projects. So we call ourselves the Cutting the Green Tape Strike Team, and it's kind of a little bit of a silly name, but I think it actually makes sense. We really view our work as urgent. It's urgently needed. We need to move quickly, and really increasing pace and scale is about doing things faster and doing them cheaper. And that's really kind of what it boils down to. Everything we do, every action we take, we ask ourselves, will doing this thing help support improvements in terms of pace and scale? And if the answer is no, then we stop doing it or try something else. And so we have a team. We actually have funding. We've heard a little bit today about how lack of agency focus or lack of agency staff can get in the way of doing good work. We actually have a team. I have a small staff in headquarters, a couple of scientists, and an administrative person. And then we have 9 staff across much of the state, not all the state, unfortunately. Specifically for you all here in this region, we have a supervisor and 2 staff in our Bay Delta region. That's our region 3. It's the green region there. And we have 1 environmental scientist in North Central region. And many of you may know Pete or Kaylee already, but whenever in doubt, we also maintain an inbox. Restoration permitting at Wildlife, we staff that inbox, and we will put you in touch with the right person to help strategize how to get your project done, as well as on our webpage, we've got all of our staff listed. So here's a little preview of what the department's doing. Really, our activities in terms of permitting fall into a couple baskets. We haven't addressed all of them yet. We are a work in progress, but we are really looking and focusing on take authorizations. So those are permits to pursue, catch, capture, or kill some organism, usually a protected one, as well as CEQA, and then we have some other kind of work to do in the future. So just really quickly, one permit type I'm going to talk about is something called the Restoration Management Permit. That word management is really important. It's a consolidated take authorization permit for state-listed species and a category called fully protected species. We also have something called the Restoration Consistency Determination, which leverages federal Endangered Species Act permits and allows, in certain cases, the state to waive the need to write a permit. And I always say the most efficient permit is the one you don't write. So we'll talk about that. Kind of an older tool that many of our staff that we fund are working on is something called the Habitat Restoration and Enhancement Act. And this is a permit for small areas. Might be something really important for early detection and quick eradication of a new outbreak of something. So something to keep in mind. It's a very efficient tool. And then finally, something that we didn't bargain on because it came about because of a budget trailer bill, but the CEQA exemption for restoration projects, otherwise known as SERP. And we'll talk about all these things here as we move through. So the restoration management permit has a very intentional name. This permit is for restoration, enhancement, activities, management activities, all these things, really instead of a permit that really merely treats protected species as something in the way of your project, this kind of switches it up, and really this is intentional. You are intentionally managing protected species for their recovery. And so really important concept here is just to keep that word in mind. And really what this permit is all about is really a permit that will result in net benefits and for projects that contribute to recovery. And it's also an umbrella permit. We were able to take existing law and not really have to go pursue a regulatory change or anything like that, and develop a new permit using our existing statutes. And so we've combined California Endangered Species Act authorization as well as fully protected species authorization. In the future, we are looking at adding additional authorizations to this permit, including common species take and species of special concern, and maybe even lake and stream alteration. This would create a permit that is a one-stop shop for restoration. So that's really our ultimate goal, and that is going to take a legal change. So it's not going to happen overnight, but we are working on concepts and have some ideas about how to do that. The current permit we have is free, doesn't cost anything. So that helps in terms of saving dollars to put into project work rather than into permitting fees. I didn't mention this before, but our team is actually general funded out of the California budget, so we're not dependent on permit application fees either. One thing about the RMP is that it's been designed to cover projects, large or small, and even enormous projects. And we've actually done this in a couple of places, tens of thousands of acres worth of work in certain areas have been permitted via the RMP, and even phased projects where we might not even know all the steps. So we've been able to incorporate those. And another thing that we're really thinking about with this permit is the fact that many projects need lots of different permits from different agencies. So we really work hard to incorporate other permit requirements by reference into our permits so that you don't end up with 5 different permits with 5 conflicting sets of conditions. So that's really important as well. This permit also is really focused on the whole life of the project, really. And so what I mean by that is, there's the initial beginnings of the project where you're implementing. You might be constructing something, depending on what kind of restoration it is, and then moving into kind of an implementation and management phase, and also monitoring. When we can, we are focused on trying to permit all that take in one permit, and really having it serve the whole project rather than the project team having to come back to the department 2 or 3 times to get different permits over time. We really want to try to get that buttoned up early. What is take? And again, that's pursue, catch, capture, or kill. And so the RMP can cover both non-lethal take, which usually means moving something. In a few cases, it might be a conservation or recovery translocation type project, but we also can cover lethal take, and sometimes that's unavoidable. But again, the focus of this permit is recovery. And we're really working hard to adjust our risk tolerance in the department for these restoration and recovery projects, and to recognize that they can be really messy, and we have to make hard decisions about what to do where there are listed species, protected species right there, and that's where we can do the most good is by working directly in their habitat. And so that's a really big thing is adjusting our risk tolerance so that we can have the most bang for our buck and get these species on the road to recovery. Another tool that we've developed is something called the Restoration Consistency Determination, and this is for dually listed species that are covered by a federal take authorization. So this can be like a biological opinion or habitat conservation plan. And when you have that situation, you can request something from the department called a consistency determination. And that determination is basically a letter that says, we have determined that your federal take authorization is consistent with the State Endangered Species Act. Therefore, you do not need a permit from us. Again, this is free, doesn't cost anything, and this is really quick. If we have dually listed species only that need take authorization, this can cover the whole project. It's a simple request, and it's a 30-day process for the department to make its consistency determination. We can do this on a project-specific level. We can also use federal programmatic biological opinions. And some of you may know there's a new one that Fish and Wildlife Service has covering the entire state of California, 72 species under the ESA. And there are also regional programmatic biological opinions for salmonids that we work with a lot. So we figured out a way to work with those. And this restoration consistency determination uses the same legal authority as the restoration management permit, and that is that the take is for restoration management purposes, and really focusing on recovery. So this programmatic biological opinion option is something that that is new, and we always said we could not do programmatic consistency determinations, but we figured out how to do that. And really, the way that these things typically work is that there's an overarching biological opinion that has a process built in where a project proponent can request enrollment, so to speak, into that program, and they submit an application form that talks about their specific project. Then the federal entity would go ahead and make some sort of written approval and say, yes, your project qualifies for the programmatic. And so this is a way that that we can take that collection of documents and deem that whole stack of paperwork as consistent with the California Endangered Species Act. And again, I already mentioned this, but there are existing PBOs out there for restoration, with the Fish and Wildlife Service one being the newest and most exciting. This is just an example of what that form looks like. And you'll see under information to include that it requires a project description. And that's really how we can then figure out the nuts and bolts of the specific project and deem it consistent with CISA. So the last kind of take authorization I'm going to mention is something called the Habitat Restoration and Enhancement Act. This is an umbrella type permit. You can get coverage from the water boards, potentially, as well as the department, for a number of different types of permits all at once. This does only pertain to small projects up to 5 acres or 500 linear feet of stream or shoreline. But you know, thinking about the topics here today and kind of this idea of early detection and treatment of new outbreaks of invasive species, this could be a tool for that. It could also be a great tool to pilot a new treatment idea somewhere in the field and do it on a small scale and get it permitted really quickly. So SHRP stands for Small Habitat Restoration Program. That's a water boards program. HREA is the Habitat Restoration Enhancement Act. That's a CDFW program. Those two things work beautifully together for small projects. And the last CDFW tool I'm going to mention is this CEQA statutory exemption for restoration projects. This was part of a budget trailer bill that passed in 2021. It's a complete exemption from CEQA for qualifying projects. We've had a number of projects that either focused on invasive species or invasive species control as part of the project. And so just keep this in mind. CEQA can be a big barrier to restoration. I've come across a couple of projects lately where they've budgeted $1 million plus for CEQA. That's a lot of money. We estimate that going through SERP might cost somewhere around 10% of what would normally be required of CEQA. We'll help you navigate this thing. It's fairly complicated, but basically the project needs to meet a set of criteria that the project is exclusively restoration, although it can have some incidental public benefits. It needs to address long-term net benefits to climate resiliency, biodiversity, and help recover species again, there's that word again, and include procedures for ongoing management, as well as management to protect the environment during the initial phase of the project. So we'll help you with that. And the very last thing I want to go through is just really hopeful stuff, because we have done everything we can do so far at the department, but other agencies have done a lot too. So a really great resource for all of you who are practitioners is an NGO called Sustainable Conservation. They have a new website that's just gone up. They do free permitting consultations to help walk you through the plethora of federal, state, and sometimes even local permits that a project might need to obtain. And so go talk with them. And then I'm just going to blast through some examples from some of our agency partners and what they're doing to cut green tape. I mentioned this already, the new statewide programmatic biological opinion. Guess what? They have a project category for removal of non-native terrestrial and aquatic invasive species with re-veg. Cool. NOAA, and I'm just using the Central Valley programmatic here. There's others in different regions of California. Project category, invasive plant removal and re-veg to improve fish and wildlife habitat. Fantastic. There's even a little synergy multi-agency process here with NOAA and the Coastal Commission, whereby if a project qualifies for the NOAA programmatic, it may also qualify to get a pass or at least a very expedited process for a coastal development permit. Good old Nationwide 27 with the Army Corps has been around a long time, has its own reference to invasive species control. The water boards. The really exciting thing that I just don't have time for is the new statewide restoration general order. I mentioned the small habitat restoration 401, but both of these tools are fantastic. In particular, SRGO, when you really want to do things on a large scale, that's going to get you an expedited 401 and waste discharge permit. And then there's CEQA. There's lots of options with CEQA. I talked about SERP, but there's other ways to get through CEQA in a quicker way. And one of the things that myself and my strike team are here to help with is to help navigate that, come up with some ideas, and see about maybe cutting some of the red tape out of CEQA. And so I'm just going to end with a couple parting thoughts. Use these tools. These restoration permitting tools are going to really, really make a difference as we gear up to really speed up what we need to do in California. Get help, talk with us. We're here to help. Sustainable conservation is another great resource. This stuff is complicated, but there's more than one way to skin a nutria, and that's actually a benefit that we have all these different options, and that's going to get us where we need to be. Thanks so much.
We only have about 1 minute per question. So we have one quick one right here.
Thanks for the presentation and thanks for all your work in this space. It's really fantastic. We love the SERP process. We've gotten a couple of projects through SERP. I'm curious what sort of pushback the whole process, particularly SERP, is getting, and what that portends for January 25 when we get this thing to be permanent.
Right. So what Campbell's referencing is that SERP does have a sunset date. It was an experiment, really. It was a budget trailer bill experiment to kind of see how it works, and we're in the mid-30s in terms of how many SERP concurrences have gone through, and I wouldn't be surprised if we have 50 or more by the end of next year. So we've heard concerns from people in various places about this being a shortcut, an end run around CEQA. However, the criteria that are built into SERP are really, really stringent. And I think that's part of kind of demonstrating the power of this tool and being really mindful about how it's used. We have definitely had conversations with lead agencies advising them that this might not be the best tool for your project. In particular, projects that might be inherently controversial. Sometimes it's just better to engage through a traditional public review process. And so that's every project's different, but we've definitely been mindful about which projects use that tool, and then we hope there will be some good news early in the new year about getting another few years out of SERP, if not making it permanent. We certainly would love to see that. Thanks.
Thanks, Matt. Thanks all. And I'm going to do a better job reminding folks to introduce themselves before they ask the question. Reminding myself here. Okay, fantastic. So our next presenter is Dr. Rochelle Tanner. Rochelle is an assistant professor of environmental science and policy at Chapman University and is going to be speaking to us. We're zooming back to Suisun She'll be speaking about the role of communication and challenges with communication for invasive species management. So come on up.
Great, thanks Rachel for the invite and the introduction. It's great to be here. Chapman University is in Southern California, so I came up for the day, so it's already been quite a long day. But today I'm excited to talk to you about some brand new data that we've been collecting in Suisun Marsh for the last, I guess, like 3 or 4 years now. And I'm going to tell you about why my title here is not how you should communicate with anyone outside of this room. So before I start, I just want to thank everyone who's been involved in this project, which is actually 2 projects funded by the Delta Council over the last few years. I was a Delta Science Fellow, postdoctoral fellow at UC Davis, and now I'm part of a large collaborative project with, you heard from Virginia earlier, and then 3 other institutions. Also, not on this slide, a lot of the data that I'm going to present about our newest project was collected by my postdoc, Gabby Keeler-May, who's probably watching. So I just want to thank her for doing that. So I think all of us understand that invasive species control requires a lot of coordination. And in the social science spaces, a lot of times we refer to this as collective action. And this is an idea that it's not enough to mandate something. There has to be some intrinsic motivation of stakeholders to work together. And easier said than done in somewhere like the Delta or somewhere like Suisun Marsh. And with invasive species, there are processes in place, obviously, that are meant to facilitate this, but they don't really work all the time. And what that leads to is situations like this, where all of us, and as Virginia said, I'm not really a wetland person, or I wasn't before this, and so it was like quite harsh to be put out there in the Phragmites patch, mowing it down with a aquatic mower. And so when we don't have successful invasive species mitigation or eradication, this is what happens. And so over the last few years, we've been looking at both the ecological consequences of community, like invertebrate communities and plant communities and bird communities. But we've also been looking at how landowners work together to manage this. And this is my slightly controversial stand on what we should do with Phragmites based on the ecological data that I'm not going to present today, so you can ask me about that later. But we've basically determined that there's no path forward where we're going to eradicate it. And so we determined from the invertebrate side that there is a specific patch density that promotes abundance and biodiversity that is similar to native plant stands. And so it kind of goes along with what Virginia was saying as well. And so in order to implement this, this requires a lot more complex communication and coordination than just telling everyone, okay, we need to get rid of as much of this as you can see. And so we tackled this in Suisun Marsh, which is a mixed public-private ownership area. And there are about 170 landowners in this area, as shown as figured by our colleague. And what this means is that every individual parcel is managed differently. And the people who live there have, or manage it, have different values and opinions about what should be done and what is working. And so we use this study system to kind of understand, is it possible to motivate collective action? And if not, how do we move forward with the spatial prioritization of this? I'm not going to talk about the spatial prioritization, mainly because it ties in all of our 5 institutional data, and we're just not there yet. But I'm going to talk about how you communicate out to the landowners once you have that spatial prioritization. So as a recovering biologist, I've learned to say this at the sort of meeting that I went to a couple weeks ago. Recovering biologist who is now working a lot in social science, I work a lot with this thing called framing, which is part of strategic communications, and it leverages psychology, cognitive science, and linguistics. And it's this idea that if you say something the right way, you can get buy-in from almost anyone. I know that it sounds like kind of a magic ticket, or maybe something that politicians use, because they do use it. But I think it's really more than that. It's a set of concepts and theoretical perspectives on how individuals and groups and societies perceive and communicate about reality. And I think this has pushed me personally more to a moderate view on how we all work together. It's not necessary always to see eye to eye on every issue, but it is necessary to see eye to eye on what the solution is. And so we tackle strategic communications in my lab by looking at 3 distinct things. We look at trusted messengers, cultural values, and how to get people to buy into a community level solution. And so in Suisun Marsh, this means engaging with duck hunters, the public, and indirectly policymakers. I just wanted to give you an example of what this looks like in practice first. So this kind of graphic is the way that we structure our strategic communications. It's actually quite formulaic, and we evaluate each step of the communications piece. So we start with a cultural value, which in the US is protection of our natural resources. This has been proven time and again by national surveys and all of the surveys that I've done in the Delta, so that's great. We use metaphoric language to describe common scientific topics that are really nuanced and difficult for stakeholders to approach from the same direction. And then we lead everyone through an understanding of how the issue that's sitting in front of us that we're discussing can lead to a community level solution. And it's all avoiding this, we used to call it the swamp, but now we can't say that anymore with the current, with the past administration, but we used to call it the thing in the middle of the swamp. And it's this area of unproductive cultural models that people just acquire through being in society. And so here, the unproductive cultural model, or one of them in invasive species management, is that this is just the environment doing its thing. We're not a part of it. It's not our problem. So we want to make sure that our language is intentionally dodging that. So I'm going to give you 2 of our projects that we're working on in Suisun that have distinct stakeholder audiences, and they also have distinct goals, but some overlap there in the middle. So we worked with landowners and managers in Suisun Marsh, but we also did a study looking at public and scientist perceptions of wetland restoration. And the common goals in here are that we're looking to identify underlying cultural values that bring us all together under a common goal. This is the first step towards collective action. We're also really interested in determining trusted messengers, because we see a lot of great communication coming from all sorts of agencies and nonprofits, but we don't really understand how that's being taken up or being perceived. We want to improve science literacy, because even though we all talk about very complex scientific topics, it's also not clear that we're all speaking the same language. Something that was really eye-opening to me when I started doing this work is that every word is actually a metaphor if you use it enough times. So we have some recommendations for some of the words that have become really charged in invasive species control, and maybe ones that we shouldn't be using. And then I think the last big goal is to use this as an opportunity to build community. Communication is a way to bring people together. And we don't want to just do this to strong arm people into doing the things that we want them to do. We want to make sure that everyone has a say in what the solutions are. So we used a series of quantitative and qualitative approaches, which was new to me when we started doing this. But they follow a very similar format depending, or across all the studies that we're doing. We start with expert elicitations. We want to see what people are trying and what is not working, so that we can make maps of, psychologically, whether we're going to target certain phrases. we run surveys about value and trust statements. We evaluate literacy without explicitly asking about literacy. And then we do interviews to get at emotional responses. And I think something that Gabby told me when she came back from doing interviews a couple weeks ago was that a lot of the feedback from her interviewees for that, well, this felt like a therapy session. I feel really good now. And I think that really speaks to, no one has really asked anyone how they feel about anything. We've asked a lot of questions about what people are doing and how they're working together, but we haven't asked people how they feel about it. So that's what we're going to present here. So I'll give you a little insight into the 3 things that we're going to put into this communications toolkit, but keep in mind, we're not done with it yet. And depending on what agency you work with, you might be receiving part of this as an email, part of the study. So we ask questions about expert knowledge a lot because some of the foundational literature in this area shows that scientists are not perceived as trustworthy messengers of climate science or environmental science because they're not perceived as experts. And so we looked at this in our landowner audience and found that scientists and managers are perceived as more of an expert than duck hunters, which is most of our audience, and indigenous people. We always test indigenous people as like our baseline because a lot of these people don't think about that when they're thinking about expertise. And in our public audience, we tested a community member who was the main audience, indigenous person and scientists. And I know that these all overlap a little bit. So social science data, the error bars are a lot bigger. But what I take away from this, and it is statistically significant, but the audience always ranks themselves the lowest. So even if someone lives in an area and spends all of their time in that area, they don't see themselves as having expertise, which is a problem for thinking about empowering people to make decisions on their own land. In the landowner audience, luckily they do see scientists and managers as having a little bit more expertise, but when we compare all of these values to just the regular cultural values that we test, like it's important to protect the environment, it's important to responsibly manage our environment, they're all significantly lower. And so the takeaway from this is that expertise is really not the way to communicate about anything. You can't start your conversation with, well, I'm this, and you have to listen to me. So 3 minutes left. All right, I'll rush through this. So in our investigation, we found that the Suisun Resource Conservation District, which is created and managed by the landowners themselves, is the most trusted messenger, and it's the single node in our network. There are no other communication strategies going on. There are some agencies around the edge here. Not only did we find that they don't participate in this trusted messenger network, there's one in particular that we've had a lot of negative emotional responses to. You know, that's just really showing that a trusted messenger has some of those shared cultural values. They live in the same place. They don't have a ton of power, but they can disperse resources. I'll skip this because I talked about it already. But basically, we went on to continue to look at cultural values that were shared among the public and the landowners, and they come up the same as all of our national studies too, which is really interesting. So if you communicate up front that you want to protect the environment, stewardship is important, and we want to responsibly manage land, then people are more likely to listen to you, and they're more likely to feel positively about it. That's that green bar. So they're more likely to feel like they're hopeful and they want to participate. We also did some work looking at some language that is already used by agencies like the Delta Council to see how it tested against some other cultural values that we were looking at. And we found that generally, again, those 3 top values come out the same. There are some other values that are used like interconnectedness and safety. They don't test very well. And then anything economics related tests pretty low. And this holds with national surveys that show that economic incentivization, even if it's personal or community level, is not a good way to motivate people to work together, which is not what you would think. And we also tested management value statements, and none of the ones used by agencies are working with this particular population. They just care about waterfowl hunting and waterfowl habitat. Okay, so this is further confirmation. We looked at literacy about what invasive species are, and basically all of the landowners in this space are just trusting blindly what the Resource Conservation District is telling them. So they don't really come up with their own definitions of what invasive species are. And so this is really an opportunity for us to have conversations about how do you identify an invasive species, and why does it matter that it's invasive species? And this is what I promise. Restoration is one of the most highly charged terms that we're seeing in our interviews, no matter what kind of restoration it is, vegetative restoration, tidal restoration. And so we're recommending that we just not use that in certain contexts when you're trying to motivate people towards collective action on invasive species control. So our next steps, we're gonna deploy this, and you might see it in your email, and we're gonna do this beyond Phragmites as well. Okay, I'm at the end. Thanks, everyone.
Thank you, Rochelle. That was great. We have time for one quick question, and I assume we have questions here. So anyone in the audience have a quick question?
A million questions, but
Sorry, could you introduce yourself, please?
yes, Doug Johnson, California Invasive Plant Council. So you're recommending not to use restoration. Does that mean stewardship and responsible management are preferred? Are there other recommendations?
Yeah, so that was something that my postdoc just told me as I was like about to leave for this. Yeah, the cultural values test really highly. I think it's a mixed bag when you use the word restoration. So if you're in front of the right audience, and if you have the right trusted messenger, it could be successful. But the problem is that people have their own understandings of what restoration is. And so until we do a better job of improving literacy around it, you're better off describing the specific action that you're trying to take.
Excellent. Thanks for that question. And thank you so much, Rochelle. Here we go. Okay, so we have one more talk for this session, and then we have a panel afterwards. And I'm excited for our next presenter, Dr. Lisa Wanger, who's a research professor at the University of Maryland's Center for Environmental Science, part of the Chesapeake Bay Biological Laboratory. And Lisa is also the chair of the Delta Independent Science Board. And this talk is going to be all about the importance of economic valuations to help inform invasive species management strategies. So Lisa.
Is the time relative to 20 minutes or 15? Okay. Thanks. I'm so glad to be part of this conversation. And what I'm going to be talking about is how we bring public values and institutional missions into decisions about how to treat invasives, and to hopefully use that information to get the highest benefits we can out of these programs. So this is a slide that really shows the whole stream of benefits of how we connect what's going on the ground with invasive species management to these social benefits, which I'm going to call economic benefits, in contrast to the previous speaker who used them to mean financial benefits. We economists actually define economic benefits as anything that makes people happy or gives them satisfaction. So it's a very broad definition of what we consider benefits. So this is a complicated slide here, but this is the connectivity that has to happen before you can get to showing the social benefits of actions. So you start with a management activity. I'm going to go remove nutria from the landscape. What's the first ecological outcome? Well, the first thing is I have fewer nutria. That's usually how people measure a lot of program outcomes. But hopefully then you get to some functional thing that has meaning to people. You know, the marsh is staying intact. It's providing habitat for birds and fish. We have these other ecological outcomes that get closer to things you can easily explain to non-scientists about why this is beneficial. And then to get between the boxes, first we have to say how well is that management activity working. And similar to a previous speaker, we do find a lot of non-linearities in these relationships. So first, you're putting in all this effort. What level of outcome are you getting in terms of bird habitat and things like that? But then we still need to make the connection to, okay, I know how the ecosystem is changing, but I need to get to an endpoint that people value. If you're going to do monetary valuation, I'm not saying we're able to monetize all the good stuff, so it is a reductionist approach in some ways, but we look for, what is it about the ecosystem that people are using? And so usually the middle function is something about number of users in response to changes in the ecosystem, or number of beneficiaries. It can be what we call non-use values too. And then finally, we get into this, how does that affect the value? So I'm going to talk about fishing, so I'll just use that example here because it's easiest to understand. If the invasive species treatment increases the number of fish or the size of the fish, that might increase the number of anglers, and then they will have a more satisfactory experience if they get more fish or bigger fish. So that's what's going on here in terms of creating all those relationships, and it is quite challenging in most cases. So but we enjoy the challenge. Okay, and then another sort of big picture thing I want to give you before I delve into the case study that I'm going to talk about is, I'm not sure everyone understands that the mathematics of economic analysis are rooted in human behavior and perspectives. And so this kind of complicated figure is rooted in the observation that you've heard the saying, a bird in the hand is worth two in the bush. And this is what this is representing. People value sure things more than they value risky things. And we can mathematically quantify that if we have the right kind of data, where basically what happens is the square is a decision point. I'm either going to spend a lot on invasive control or a little, and then that leads to a circle, which is a chance event. And that effort results in success. I realize we can define success multiple ways, and we have second chances sometimes, but let's just pretend we have a one-shot deal. You either get success with some probability. I'm showing for the high cost effort, I'm showing a 70% probability of success and a 30% probability of failure. And then associated with success and failure is some measure of the value of the outcome. Just for illustration, 110. And then you just multiply the 0.7 by 100, the 0.3 by 10, and you sum those to get the 73. And this is what economists mean when they say expected benefits. So if you try to pick up one of these economic studies of invasive species, you'll often see this term expected benefits. And it has this very specific meaning, not like, oh, we expect to get this, So we're using common words in uncommon ways. So I've only, again, I'm not going to, this isn't the main case study, but I do just want to talk about how important I think this information can be. I've only had one case study where I've done valuation of invasive species where I've been able to gather the right kind of data for these expected benefits. And we looked at a variety of variables that were affecting the ability to restore sites. And the restoration going on here is a revegetation to prevent invasion by cheatgrass. I don't know if you guys are familiar with that. Rangeland invasive, very invasive grassland species. And I guess maybe in some ways this is a little depressing, but so here's the 50-50 probability line. So you've got probability on the y-axis, proportion of the neighborhood in cheatgrass turned out to be the variable that explained the most variability of outcomes. So it had the highest explanatory power. And so we were able to show that the blue line is representing sites that recovered on their own. So if you have very low cheatgrass in your neighborhood, you don't need to restore because it's gonna go to native vegetation without human intervention. And then the red line is, how restorable is it with this revegetation program? And what you're looking for, ideally, to maximize your management dollars is where those two curves diverge the most. And then also recognizing that you have a very low probability of success once you have really high propagule pressure in the landscape. And so I think this is just a useful concept to keep in mind of thinking about these expected benefits and how you can use your resources to avoid spending money where you're not going to be successful or where the system would have recovered on its own. So things vary a lot by situation. All right, now finally to the case study. And I do just want to acknowledge that I'm going to go pretty fast through this case study. I didn't have enough time to make the talk shorter. And there is a paper if you really want the details. I'm going to rush through a lot of the details. So the case study is about research into Hydrilla verticulata floridome resistance in Florida. And I had to show you this map because congratulations, according to the USGS in 2016, you have eradicated Hydrilla. So I don't think that's true, but you can tell me later if that is. Anyway, but the important thing I wanted to show here is that in Florida, they're dealing with the dioecious form, the asexually reproducing form of Hydrilla. So that's a key plot point that we'll get to in a second. So the problem that they experienced was they were treating, they have these lake systems where they're treating Hydrilla with fluoridone, and suddenly it just stopped working. Not stopped, but was a lot less efficacious. And they responded by increasing the concentration, more applications, and it was costly and it was having off-target harms to other plant species. And so they set out to do research on what was going on. And the reason it wasn't obvious is because it was asexually reproducing Hydrilla, they just didn't expect it to evolve fluoridone resistance. They didn't think that could happen. So it really did take a lot of people coming together to agree that that's what was happening. So the system we looked at is the Kissimmee Chain of Lakes, or KCAL, I'm going to call it. These are hydrologically connected lakes, and the ecosystem services of them are recreational fishing, lakeside fishing. They're very well loved for those two activities, and also they serve flood mitigation and endangered species habitat. They spend a lot of money on hydrilla control. Half their hydrilla budget goes to KCAL, and half their invasive control program goes to hydrilla. So it was important to get this right. And then this just really shows the smoking gun here. So what this graph is showing over time, the blue line is how much money they were spending on hydrilla control, and the orange line is how many acres of hydrilla were surveyed. And so prior to the first black line, what you're seeing is the control levels were fairly constant, but hydrilla was creeping up. And then between the two vertical black lines, you really see a huge increase in spending. And they were able to maintain the hydrilla levels, but only with tremendous effort and tremendous cost. And so that was the period that the research was initiated. And then finally, the second black line is when the research was applied. And you see the costs drop precipitously, and the hydrilla control was even better than they had before the resistance developed. So what the research brought to the table was, first of all, several agencies funded this research. They had different priorities. Like EPA wanted to see less chemical herbicide used. Florida Wildlife Commission wanted more fishing. USACE wanted to use their research lab. So everybody had their reasons for chipping in. But they were able to show that endothol would be an acceptable substitute, even though it had been rejected for use in the past, because they thought it needed to be applied during peak growing season. So the thought was, if we kill all the plants when they have maximum biomass, you get a lot of biological oxygen demand, and you get anoxia in the lake. So they didn't want to do that. So the research was able to find a way to use endothol, change the timing, change the concentration in a way that prevented that problem. And they also were able to find a cheaper way to use it so that the cost became comparable to the fluoridone. Okay, now this messy slide is where all the good stories are. I don't have time to tell them all, but this is the connectivity story, where trying to put the pieces of this puzzle together were extremely challenging because it is such a joint effort to do hydrilla control in Florida. There's multiple agencies out there treating. There's multiple people keeping different sets of records with different protocols. And we had to be data detectives and talk to lots of people and rectify different stories coming from different databases and different people. And so I appreciate the previous speaker who laid out all those qualitative and quantitative techniques. We used almost every technique possible we did interviews, we did expert elicitation, we did literature reviews, and then finally ended up with simulation modeling to give us what we needed. And just, we had to talk to everybody and bring in all their perspectives to really understand what was going on, why they were treating hydrilla, and what the benefits were to everybody. So, and something that happened during this process was that we discovered there were different stories about what worked. The data were very messy. I'll show you some data later. And so we ended up actually having to simplify our models in a lot of ways because there was almost too much messiness to represent with high fidelity, and there were conflicting stories. So it's kind of an interesting story that the multiplayer, it's a benefit to the treatment of hydrilla, but it made it very hard to do the analysis. Okay, now I'd like you, it may sound like I'm getting into the weeds of economic analysis, and you all don't do economic analysis, so why should you care? But I do want to share some of these details with you because it really plays a big role in how I'd like you all to think about collecting data, saving data, taking pictures. And first of all, I just want to acknowledge, of all the invasive species groups I've worked with all around the country, you guys seem the most sophisticated in terms of doing controlled experiments and really very thorough monitoring. So I just want to grant you that first of all. But the actual scenario is what we were able to show with data. So what happened? So I need to back up for 1 second. We were valuing the research investment. So what we need to do is say, okay, what happened when you applied the research? And so we got this hydrilla stabilizing, we got this new treatment protocol. But then the really key thing is to figure out what would have happened if you hadn't invested in the research. And so this is what we call the counterfactual. And this is where a lot of the effort went into. Really had to depend on some of the old timers to tell us how they used to treat. And basically what they did were these spot treatments of endothal, which didn't result in a lot of boat access, which was the main reason they treat. And the costs were higher. And so this is where we're trying to get to show the benefits. Thank you. You've got, and ideally we're modeling this with stochasticity. So, you have things like weather and other things changing the effectiveness of your treatment over time. So you have your actual, you have your counterfactual that includes both the ecosystem service benefits and the treatment costs, subtracting off the treatment costs because they're going to differ. And then, the benefits are this area between the curves. And later on, I'm going to show you some of those if I don't run out of time. Okay, so this is what we're headed toward. And these are some of the data inputs that we were able to gather. This now we're going back to that flow chart I showed you. I need all those relationships to say, how does the ecological change the fish is the top figure here. And an interesting finding was that the game fish, the fishers actually preferred the lakes to have about 40% hydrilla cover. They thought that generated the highest game fish population. But then there were folks more interested in the non-game fish who said, no, it needs to be lower, more like 30%. I'm not sure this graph is actually quite right in terms of the final numbers, but, and then the lower graph is the fishing effort. And you will notice a strong similarity between the fish abundance and the angler effort. And that is because we have what we call an endogeneity problem here of the data on the fish population comes from what the fishers catch. So we can't get around that. There's no other surveys, although for the non-game fish, we did have some site-specific studies that enabled us to change that figure. But anyway, yeah, so we're kind of stuck with the data we have. And then just quickly, some other key model elements are, you get this huge reduction in effort once you get above this 80% threshold of hydrilla. It basically goes to zero. We modeled this logistic growth function for hydrilla in the absence of effective control. And under that, it only took 1 year to get to 80% cover. So effectively, you only get 1 year of fishing if you don't treat more effectively. And then we transferred values using economic benefit transfer from other studies of how much people are willing to pay to keep fishing. And also this non-use value of, I just like knowing the lakes are healthy and I'm willing to pay a certain amount for that. And we looked at things by lake. There was a big spatial analysis component too. And then there's different cost per acre treated. Thank you, that we used. And there's what you see here is the big lakes tend to be lower cost, the small lakes tend to be higher cost. There's other variables going on here, but basically that's showing that there are some economies of scale to treatment. And then here's the final results, where this green curve here is showing you that area between the curves, and it's made up of the ecosystem service benefits or losses avoided really with treatment, and then the treatment costs, the net treatment costs, which are higher than in the counterfactual. Okay, and then we also had to do a lot of work figuring out who spent what on research. We got a number, and then we were able to generate the net benefits of research. And this as a sensitivity analysis we did showing some of our biggest uncertainties. How long did it take for the hydrilla to reach the certain threshold, because the data were super messy. And what area would you have treated in the counterfactual? But the good news is blue represents positive net benefits, and over the majority of our state space, you see positive net benefits. Okay, so I'm just going to conclude on some of these things. That's some of the messy data. So this is what we were dealing with. Different lakes had different responses. They were treated by different groups in different amounts. And so being able to use I had a very excellent statistical modeler working with me who was ready to pull apart these different factors, but we just couldn't make enough sense of the data to do this in a very thorough way. So that's another story. Okay, so the great thing about these retrospective benefit cost analysis is they really help you see the cost of inaction. If we hadn't done this, what would have been the harms? And so that can be a very powerful management perspective. But building up this sequence of models, I've glossed over tons of detail of the modeling we had to do, really requires good record keeping. And in particular, it would be super helpful if people would take more pictures of the before conditions. So when I'm working with stakeholders, I can show that. People don't do that a lot. And of course, before after control designs are ideal, and I see you are doing some of that. These collaborative approaches across agencies are great for the ecological outcome, but they're super challenging for modelers. So the more record keeping can be standardized across agencies or even combined would really enable these kinds of economic analyses. And then just one little connectivity thing that I just can't help but mention is that, I showed you that cheatgrass thing where the amount of the neighborhood in cheatgrass affected your restoration success. Similarly, that same thing happens within all other systems, but there's also these opportunities because that connectivity changes over time. And so some key things we found were for hydrilla, hurricanes uprooted them and created a low population. That's an opportunity to really get in there and see some progress. And then in our systems in other states, we see cold winters can also have a big effect. So I'll get to the end here. Just want to acknowledge lots of folks helped with this. And there's a paper if you want to know more. And these are invasives I have known. So thank you.
Okay, so I don't think we have time for specific questions, but Lisa, will you be here during lunch? Yes. But you can ask Lisa specific questions during lunch. So, but before that, we are going to have a panel discussion before the lunch with all of our presenters from this last session. So, Michelle, can you come on up? Instead, Brad, thank you. And I'm going to ask that for folks that are adding questions virtually, you can add it into the Q&A on the Zoom. But if you have questions that are specific to one individual or to the panel at large, please specify that if you can. I also want to give everybody in the audience and those that are at home maybe a second to just stand up as well. We have 20 minutes. I know we're getting up to the lunch, but yeah, take the chance to wiggle a little bit. Yes, there we go. Stretch it out. Okay. So for this panel discussion, we're just going to have an opportunity to dive in more to this theme around connectivity with research, with management, and with policy. And just a couple of reminders for our panelists up here. You can turn your mics on and off, and I encourage you to keep them off. If you're not speaking, turn them on when you are. I'll help remind you. And for anyone who's asking a question in the audience, just introduce yourselves before. So I'll start with a kind of just a softball question. But, one of the, I think, threads that came through all the talks in this last session were emphasizing either actual projects where you worked with individuals, worked with community members, or on very interdisciplinary teams.
And so I just love if you could share either specifically or more generally, that's fine, why it's so important for this interdisciplinary work in invasive species management. And if you have a, if you want to, just feel free to unmic or raise your hand too, panel members. Brad, if you were going to start, you can go ahead.
Yeah, I can attempt this a little bit. I think from my perspective in my role, it's really focusing on really ramping up all types of enhancement, recovery, restoration for the right audience. We just need a lot more people excited about it and engaged in it, and different non-traditional advocates for it. And so one of the trends that's exciting, and I have a statewide perspective. I actually don't know that much about the Delta. It's one place I haven't worked in my previous jobs here in California. But just seeing more urban groups, more groups of color getting involved in stewardship, restoration, invasives control, and I think that's one thing that we as scientists or policy wonks or regulatory wonks really need to learn how to communicate with these folks who ultimately are absolutely necessary. And we also need, I think, to let go some. We've worked in this field a lot. We have a lot of knowledge that I think sometimes just the act of interacting with nature, treating oneself as part of nature and not separate from nature, loving nature. I think under that model, some folks are going to do some stewardship that we might regret a little bit later. But I think it's more important to get folks involved than it is to get everything 100% right. And so that's one thing that I'm really focused on in my job.
Just because it's getting close to lunch, I'm gonna pick a different story. I could reiterate things from the case study, but I just want to say that we did eradicate nutria in the Chesapeake Bay, and I think one of the key elements I got from that was how valuable it was to bring in the people with local knowledge. That we ended up using trappers. There's an economically viable trapping industry on our Delmarva Peninsula, and we just engaged them in trapping. And it wasn't the only thing we did, but just that there was so much power in bringing in the local knowledge and getting efficient treatment. So, and of course, on the other end is finding, when you have to live with an invasive, finding what level of control meets different stakeholder needs, because I'm sure you've all experienced that some stakeholders like invasives, and finding that middle ground is also so critical to getting acceptance.
Yeah, I mean, it's sort of, you're not here? Yeah, repeating really just that, our work, we rely pretty heavily on, community managers, scientists, volunteers, and also restoration projects as well. So I think two things. One is the capacity to undertake what we're doing, whether it's managing invasive species or restoring habitats. We're never going to have anywhere near the resources that we're going to need for this kind of work. So engaging the public both to educate them and bring them in as part of the process, but also to take advantage of their efforts. I mean, we need this dramatically. So I think we really, in order to sort of solve the big problems, we need to figure out how we're going to do this without the resources to actually do it. And I think this is one area where we really need to think broadly about making the public part of whatever it is we're doing.
Yeah, I think I've had really similar experiences to Ted and Brad. It's the reason that all of my other projects, except for the one in the Delta, run in multiple languages. So we really focus on minoritized communities participating in restoration. But I also want to make the pitch for interdisciplinary science teams, because we wanted to talk about that too. And I think that's been a real strength of the project that Virginia and I are currently working on, is you get these perspectives across fields and we all approach the problem differently, and all of us have a different history with
the species that we're working with, and I think that's been really valuable. The reason that I've gone from biology to social science and part of my work is because we know a lot about the biology of a lot of things, a lot about the ecology, but we don't really understand how to motivate people to work together. And so I think we really need social scientists on our team.
Thanks all, that was great. Well, I actually want to start because we haven't heard from our online audience in a minute. I'll check first if there are any questions. And if not, then I encourage anybody who is virtual to please add your questions to the Q&A. But right now we'll turn to folks in the room. So does anyone have a question here? Dylan is our mic runner, so Dylan's going to run over.
Hi, I'm Michael Ariano. I'm basically like a delegate from our high school right now, at West Lucera Collegiate Academy. I was wondering if there's any programs or ways that we're trying to reach youth right now, because there's a lot of young people who really care about the issue of invasive species and environmental issues. And it's hard to find ways to get involved at younger ages. And I was just wondering if any of these different agencies have programs to help reach youth and solve this issue.
We can add yeah, bonus panel member. Yeah. Can we pass the mic up here? 1 second. Yeah.
Well, there's, I think a lot of the nonprofits and resource conservation districts work with the public more. Amy might have some ideas too. She's over there. But I work with the Consumers Preserve, and they have a great environmental education program. Usually they're working with 6th grade and below, but there are programs to get involved at the Consumers Preserve. It's often limited by the people that need to go out with folks. So, it's difficult sometimes, but I really feel like that's a good place. That's in the Northeast Delta. And does anyone else have some ideas?
Well, just in my little area, I know Save the Bay Watershed Project, there's a number of, as you said, NGOs around these areas that involve education programs and bring people in. So I'll pass the microphone to the person back there.
So the Center for Hang on 1 sec, if you can use the mic, it helps our online folks here. It's for the online audience.
The Center for Land-Based Learning, which is based in Woodland, but has programs throughout the valley. They have programs called SLEWS, and I can't remember what SLEWS stands for. It's SLEWS if you're Googling it. And they engage high school students, and it's a really great program. And Mai Vang, who's one of the city council members here in Sacramento, she went through the program in high school, and she's a huge advocate for the kind of work we're doing because of that. So it pays dividends even if they don't go into the field.
I appreciate that question and collaborative response. So thank you. And yeah, the SLEWS program is a really great program here in the Delta region. Okay, so another question for folks in the room.
Yes.
Thanks.
Okay, so I'm Tal Robinson with CEFW. My question is really for Ted, I think. How would you define or differentiate functional eradication versus just ongoing management?
So the idea of functional eradication is you're trying to identify a target that gets your invasive population down to a level with the impact on whatever it is that you're trying to support, endangered species, your special, the habitat. So you're getting your population down to a point where their impact is really minimal. So we've eradicated their function. We've gotten them out of the system enough where they're not really having much of an impact. And so that's different than just sort of managing, saying, well, gee, we're going to get as many as we possibly can, but we don't know where that trade-off point is. We may not necessarily need to go beyond that level that we identify for functional eradication to recover any real more additional ecosystem function. But if you're just kind of doing more is better, you may be wasting resources you may not actually need to get to that point, or you may have to work a little harder to get to sort of that sort of threshold, if you will. So it's having a threshold that's really useful, that's cost effective, that's
you know, worth achieving rather than just saying somewhere more is better. That's the difference.
Yeah, go ahead, Lisa.
So, yeah, I think we were really talking about the same thing, Ted, when I was showing how the fishers liked 40% cover of hydrilla in the lakes. And then, and so that's basically the agency does have to kind of adjudicate the fact that some people want 40 and some people want 30, and they have to work that out either by treating different lakes differently or picking something in between, or, you know, so that's how that, I think, is a perfect example where they pick 30% cover as like, we're not going to try to go below that.
I want to just add one thing, because I understand where you're coming from, and I get this discussion all the time. I think we sort of struggle with sort of a CDFA, and I'll take a lot of heat for this sort of view that if we don't eradicate it, we fail. And you can look at, you know, Oriole and fruit fly. I just talked to this guy saying, hey, yesterday, it's eradicated every year, and it's back every year. Eradicate back. It's like, you know, it's like serial eradication. Is that really any different or any better? Okay, and I'm not saying this. And I understand there's real mandates for that, because if something gets established, then there's all sorts of international issues that sort of pop up in terms of exporting fruit. So I'm not, you know, addressing that issue. But I think from a practical view, just as this was pointed out, if we can sort of reduce what we need to the point that we're getting the benefits back, I think that's really the critical area. So I think we just have to sort of get away from having to reach something that we may not be able to. I hope you guys can eradicate, but if you can't, then what? That's the issue.
Yeah, but from my perspective, management is more about maintaining and hitting a target, a threshold, which is exactly what you're talking about. That's not eradication.
That's correct, right? I mean, it's a long-term process, and that can only be undertaken where there's will and resources. Absolutely, you can't do that unless you have that. So you may be looking at the long term. And the issue that I try to point out is you're saying, well, eradication, we get it done, and then we can walk away. But there's so many examples of that species or similar species getting reintroduced. So unless we can eliminate the pathways for future introduction, we may not actually succeed that way.
Virginia Medza from Santa Clara University. I was really struck, Lisa, by what you were saying about the data needs from practitioners. It would make your job easier, and I certainly feel your pain in trying to like get cost data for anything on restoration. I was just wondering, for the rest of the panel, are there any kind of data needs that you can see, or things that people who are working in this field could do differently that would make your jobs or the research that you're trying to do more effective, better integrated, et cetera?
So I think this might be a little controversial, but I think bringing the people side of science back into the science is something that would make my job a lot easier. You know, asking people what they're, you know, everything that we've tried to get away with in science with mitigating bias, actually bringing that into the conversation and making it a part of why we make decisions is really important, because people don't make decisions rationally, and it's really important to understand where people are coming from, the cultural context, and I think it's going to make it easier for us to work together across diverse stakeholders if we actually understand more than the nuts and bolts of what people want, but why they want it.
Okay, I think we have time for probably one more question, and we're gonna turn to Dylan, if you wouldn't mind. I do want to take an audience question from online real quick, and then we'll go back to one more in the room.
Okay, so when communicating with the community, have any of the panelists experienced community burnout from ongoing messaging? And if so, do they have any suggestions for how to keep community members engaged and keep the message fresh?
Great question.
We're just both agreeing, yes. I mean, you know, I talked about that sort of 5 or 6 year community program. It's not the same people 6 years later. I mean, it's continually getting new people. So one of the jobs of that public involvement is continually recruiting
And from the cutting the green tape person, getting some on the ground results can be really energizing for community groups. I think sometimes some of these smaller projects, there's some overthinking and over planning that can go on, and sometimes just doing something even on a very small scale can really help.
Any additions? Good. Okay. One more question from gentleman in the room. You want to ask a question, Dylan? No question. Okay. Are there any other questions in the room? Sorry.
Was inspired from Ted's talk, so he probably has the best answer for this.
Introduce yourself, please.
Nick Rash with the Department of Water Resources. I think a lot about invasive weeds, and almost all of our invasive weeds are from horticulture or the aquarium trade. How do we put more effective policies in place for that? Because it seems that if there is any even tiny economic benefit considered for some of these plants, we don't touch them. Policymakers don't want to do anything about it, and so even if the potential cost of managing them if they get introduced is incredibly high. That's the struggle I feel like there is.
So could you just restate in a second what you think the struggle is? It's basically getting rid of...
Policymakers don't want to impact the economy negatively by banning anything, even if the cost of managing invasives is very high.
Right. So, I mean, of course, we all know there's lots of programs of, say, taking an aquatic horticulture, which, as everyone knows, is like the largest, fastest growing segment of horticulture. So trying to replace non-native species in aquatic horticulture with reasonable native substitutes is, one way of sort of keeping some of that pathway there. So I think we need to think cleverly about ways in which we can reduce the likelihood of invasion at the same time not nuking the economy at the same time. All I will leave you with, everyone, is ways in which maybe either it's at the border or something. There's lots of ways we can, increase border protection. I mean, at ports and harbors, to try to manage that. So I think there's a lot that we can do to keep things from coming in that way. But I'll turn it off at that point.
Well, and I'll just add, I think I have a two-part answer. One is I do think economic analyses can be your friend here in terms of, I think it is important, though, to get to the actual impacts, not just costs of control, because that assumes you already know it's bad. And so I think really spending time to figure out how it's bad, it can be very helpful in quantifying that. And then the second thing is, someone threw up a FICMANU part of their slide. So we used to be very involved with FICMANU, and this issue came up all the time. This is the federal interagency working group that works on invasive species. And so APHIS would like to ban more stuff, but the problem is they don't get support from politicians. So I think we have to change some incentives here, where you need to have incentives that really highlight people preventing problems instead of responding to problems. And I don't know exactly what that solution looks like, but probably liability insurance and other things could create a better incentive structure for being more proactive.
Anyone have any final quick thoughts here on the panel? If not, we'll wrap up.
I just have one quick thought. I mean, one of the issues, and this will be very controversial, is that in other countries, if you bring something in for commercial value, you have to demonstrate it's not invasive. That's a whitelisting thing. So we can flip the whole narrative and say, well, instead of having to prove it's going to have an impact, if you're going to bring it in for sale, you better show that within reason it's going to be safe. So there are ways of doing that. Other countries, Australia and others, would have sort of phytosanitary regulations we could learn a lot from. So I think we just have to rethink what we're willing to do.
All right, well, in a second, we will thank our wonderful panel members, but I have a few quick announcements because I know that it's time for lunch. That's the main announcement. And I just want to give folks some instructions. So lunch is available outside at the registration table. If you signed up to be here in person, you were able to order a lunch. And if you have a dot on your name tag, that's for dietary restrictions. And so you'll actually come find me at the table to help find your specific meal. If you don't have a dot, you still have a meal, but please make sure that your name tags are available for us to see as you're grabbing your lunches.
that's the main reminder. Also, if you're here for the end of the day, we're gonna have a little Mentimeter quiz to gather some information about today's meeting. So we encourage you to stick around for that. Also, come back after lunch. You can eat your lunch in here, but also we'll start with our afternoon sessions at 1:30. And last but not least, at the very end of the day, we're also gonna have a little social gathering, a no-host social gathering over at Lucid Wine. And the address is, yeah, where Emma was just standing. It's on the whiteboard in the back. So if folks wanna come and talk a little bit more about what we covered today, we'll be hanging out over at Lucid Wine, which isn't too far. And that's it. So enjoy lunch and we'll see you all back at 1:30. And thank you to everyone online.
All right, everyone, welcome back from lunch and thank you for coming back for our afternoon session. We will now be starting the lightning talks. So this is going to be a series of short talks covering a variety of invasive species topics, followed by a group question and answer session. So we will be holding all the questions until all the talks are finished. And I will ask, when you are asking questions, whether you're in the room or online, please indicate which speaker your question is for to help us direct them. And let's see, and then I believe we have another break after this session, and then we will have the final session. So we will now get started. And our first speaker is going to be Nick Rasmussen, substituting for JT Casby, who couldn't be here this afternoon. And he is from the Department of Water Resources.
All right, this is a project we did to test UAVs as a tool for monitoring ribbonweed. The species was first detected in the Delta in 2017 by the folks at Boating and Waterways, and they found it at Long Island, which you can see here on the map of the Delta. And over the course of the next few years, they found patches spread across a much wider area of the Delta, which you can see here all as red points. They sent samples to CDFA in 2021, where they used DNA sequencing to determine that the species is Vallisneria australis, commonly known as ribbonweed. The species is native to Australia, but has been introduced in a variety of countries around the world. However, this is the first documented case of the species being found in wildlands on the North American continent. The species is a submerged aquatic plant that roots in the sediment, and it primarily spreads vegetatively through creeping stolons and rhizomes. The species tolerates very low light levels. It is distinctive looking in that it has these strap-like leaves, which grow up to 10 feet long. And importantly, the species forms dense monocultures wherever it becomes established. And also importantly, the species tolerates higher levels of salinity than most of the other invasive submerged aquatic plants we have in the Delta. So we tested out UAVs as a monitoring tool during 2022 and early 2023. This was a project that I led, and I worked with several DWR's drone pilots, including JT Casby, Anthony Elias Linares, and Brian Armstrong. So we went out with the drones and imaged all the known ribbonweed patches in the Delta. And then we measured those patches using ArcGIS. And fortunately, even though this is a submerged aquatic plant, because of the structure of it and its dense monocultures that it forms, it was fairly easy to delineate the patches in the imagery so we could get measurements of the patch sizes. So I'll just show you some of those images. Here you can see the patches at Hogback Island. Here it is at Long Island, where it was first detected. And here it is at a marina in Rio Vista. In all these cases, you can then see that there's a lot of boat traffic going through and chopping up this plant, which creates the risk of spread by fragmentation. Here it is in Elk Slough, which is near Cortland, California. So this is an area where we've documented large amounts of this stuff, especially around the Cortland Road Bridge. So you can see those olivey colored areas all along both sides of the margins. And in the course of doing the imagery to measure these patches, we also detected some new patches, including extensions further north than what we had previously detected. We also found it in Sherman Lake in large patches, including up to 8 patches. So here it is zoomed in on some of the very largest ones. And unlike some other areas, it grows in these big circular patches. And they're expanding outward, kind of like bacteria on a petri dish. And this is one of the very largest known patches in the Bay Delta.
measuring over 90 meters in diameter. And as with Elk Slough, we also found some new patches in the process of doing this imagery, including the one you see on the far left, which was previously undetected. So as I mentioned, we measured these patches. And so you can see the different sites arranged north to south. And the patches vary quite a lot in size, with the smallest one being in Old Town, Sacramento, which you can see here in the shade of Joe's Crab Shack, all the way up to the ones I was just talking about, Elk Slough and Sherman Lake, which each have over 12,000 meters squared of area. And so that's nearly 3 hectares across the system, which as a more useful reference, that's about 5.5 football fields of dense monocultures of ribbonweed. So we're still learning a lot about this species. We don't know a lot about the growth rates, but there is that one particularly large patch that I mentioned in Sherman Lake, which you can see from remote sensing data, from satellite imagery. So you can look at the size of the patch through time and get an idea of how fast it's growing. So for example, it increased 53% since 2020. It's growing pretty fast. So in summary, ribbonweed tolerates a broad range of conditions found in the Delta. There's about 3 hectares that we know about of dense monocultures in the system, including areas with a lot of boat traffic, which creates risk of spread. And so it's considered likely to cause various types of environmental and economic harm based on what it's done in other places it's become established. And Boating and Waterways is exploring ways of controlling this species starting next year. And just for folks, if you see this in Delta or really anywhere in California, please report it to CDFA. They have a Report a Pest hotline, and they also have this website that's a Report a Pest, and it's got all kinds of great information on it. Thank you.
And our next speaker will be Jenny Wiggins with the U.S. Fish and Wildlife Service.
Good afternoon, everyone. Today I'll be talking to you a little bit about what the Lodi Fish and Wildlife Office, in partnership with Kramer Fish Sciences, a little bit about the work that we've been doing with eDNA and using it as a guided tool in the fight against invasive species. So first, a few facts about the target species for this project. So loach are native to Northeast Asia, and 4 species are actually farmed for food in China. They're nocturnal benthic fish, making them difficult to detect in most systems, and they tolerate extreme environmental conditions, often burrowing in the substrate during dry down events. They also have a long lifespan and high fecundity, which means that they are capable of increasing their abundance and distribution rapidly. So loach were first observed in the upper reaches of the San Joaquin River with both large scale and pond loach collected by various projects. There were a total of 26 individuals collected between 2014 and 2021, eventually spanning 120 river miles downstream, with the majority of those observations occurring in 2021 alone. So here is the approximate location here's the approximate location in the San Luis National Wildlife Refuge, where a few individuals were collected in 2019. This refuge was actually chosen for the current study because because of its access to the main stem of the San Joaquin and the presence of large floodplain habitat, which is thought to be preferred habitat for loach. So in 2022, we conducted a 3-week pilot study to investigate the range and abundance of loach on the refuge. We also wanted to compare eDNA detection to a traditional gear type. So in this case, minnow traps. And simultaneously, we also wanted to validate the use of eDNA as a species-specific detection method. So through the study, we learned that baited minnow traps were great for catching loach, as we were able to trap and remove a total of 18 loaches from 9 canal sites. However, we found that without the additional information from eDNA, much more effort would be required to actually remove all the loaches from the system. So we needed then to shift from random trapping methods to a more targeted approach using eDNA as a guide. So we also concluded that minnow traps agreed with eDNA results approximately 82% of the time. And then lastly, we determined that we needed to tailor our eDNA detection methods to the local environment. And so takeaways from the pilot study then informed our 2023 study, which consisted of 2 phases. Phase 1 used a calibration study, a live card study actually, to calibrate eDNA detection to the local environment. And phase 2 focused on loach removal. And so for this portion, randomly chosen canal sites were sampled for eDNA, and then those filters were sent to Kramer Fish Sciences to be analyzed for loach eDNA and used in the trapping the following week. Sites with positive eDNA detection were then trapped using 10 baited minnow traps per site, and traps were spaced a total of 260 meters. This distance was actually informed using the data from the calibration study. Sites where loach were trapped were then resampled for eDNA the following week to analyze for changes in concentration. And so here, figure A is an example of the heat maps that were generated after every eDNA collection effort. And white circles here indicate where no loach were collected. Black circles indicate Black circles indicate sampled sites. The color gradient ranges from purple to yellow, with purple being lower concentration and yellow being higher concentration. And so then figure B shows those same sites, but notice that the same areas that were higher in concentration were also the same sites where we caught more loach. And so over the 3-month course of this study, we were able to remove a total of 658 loach using these guided methods. And so this figure depicts loach CPUE for the 2023 study. And the takeaway is that the dramatic shift we see in catch after early May. And so we found no significant correlations between environmental parameters and CPUE. So perhaps our trapping methods were effective. And with that, I'd like to say thank you.
And next we have Anita Arenas from the California State University, Long Beach.
Hi, thank you guys for being here. So as you guys may know, wetlands are very important because they provide a lot of human services, but in California alone, we have lost over 90% of our wetlands, and this is primarily due to human activities. And the ones that do remain, they have been degraded by stressors such as invasive species. Now, especially if these invasive species are plant species, they tend to be a lot different compared to our native vegetation, such as being a lot taller, bushier, and may even have a showy flower. They can also outcompete our native plant species and change decomposition, pollination, trophic interactions, nitrogen cycling, and productivity. Now, the non-native plant species I'm going to be talking about today is Iris pseudacorus, or yellow-flagged iris, and it gets its name from its bright showy flower, as you guys could be able to see here in this picture. It is originated from North Africa and Europe, but it was introduced to the United States back in 1771 as an ornamental plant. And it tends to flower during the months of May through July and is able to spread very easily due to its buoyancy of the seeds. So if we want to get a better image of the distribution of iris, the image on the left-hand side, which is a map of California, and the arrows in yellow shows the distribution of iris. But if you want to specifically look within the Delta system, which is the map on the right-hand side, all of those points show where iris has been located. And since its discovery in the deltas in the 1980s, it has spread very dramatically within a salinity gradient. So going from a freshwater system all the way to a marine system. And so one of our collaborators, Brenda Garral from UC Davis, was very interested in both the phenology and the growth of the iris in the delta system. Specifically within these 3 locations, which Buckley Cove and Brandon Island were more of a freshwater system, and Carquinez Strait, which is more marine system. And so she was able to find that the iris was less affected by the increase of salinity and were exposed to deeper inundations of water along the estuary gradient than those in the native range, so like in Spain. But if we look a little bit further down south, specifically in San Diego and Los Peñasquitos Lagoon, we've also found that iris has been seen within a salinity gradient as well. So management, specifically state parks, were very interested in the impacts of the iris in both the freshwater system and the brackish water system, looking at the pollinators specifically. And so what we wanted to see is we wanted to see the pollinations in both iris and non-iris canopies. So any non-iris canopies consisted of any vegetation that was not iris. And so we were predicting that we're going to have a higher pollinations in the iris canopies due to its bright fluorescent flowers. And so we observed the pollinators in both of those habitats and plant vegetation for a total of 160 hours in 15-minute periods in a 1 meter quadrat. And during those observations, we documented each of the pollinators, the time that they spent in the quadrats, its behavior, and the total numbers of flowers that they visited. And so what we found is that there was a significant difference in both the plants type and habitats, in which we had a higher number of pollinators in the iris freshwater sites. I also want to mention that we did not have any pollinators in the non-iris freshwater site. And so we also wanted to see what pollinators we saw within of these habitats and plants. And so what we did is that we did a non-metric MDS, and where we saw that we had a higher number of the honeybees in the freshwater iris canopies and more of the bumblebees in the iris canopies. So pretty much so far this study is showing us that iris is affecting the pollinators, specifically where we're getting a higher number of pollinators in the freshwater sites in the iris canopies. And this is very important because it's giving us a better understanding of the impacts of the pollinators, not just in Los Peñasquitos Lagoon, but it's also giving us a better understanding of how it could also potentially be affecting the pollinators within the Delta system as well. And when we're trying to look within the management perspectives and possible eradication of this particular species, it's giving us an indication that we should prioritize a freshwater site first compared to the brackish site as well. And this study is also very useful, not just in California, but it could also be very useful in other areas within the United States that is thinking about management and eradication of this particular species. So with that, I would really like to thank you guys for listening to this talk. Definitely want to thank all of my funding sources for this project and the CSUB Wetland Lab with their help with this project. Thank you.
And our next speaker will be Doug Johnson from the California Invasive Plant Council.
Thanks, Elizabeth. So I am here to talk about some analytical tools for protecting California's biodiversity from invasive plants. These are tools produced by Calypsy. So there are 3 tools I'd like to talk about today: the Calypsy Inventory, Cal Weed Mapper, and Weed Cut. The Calypsy Inventory is a database of plants, basically, and we'll talk about that briefly, but it helps land managers understand the plants themselves, their impacts, how to control them. Cal Weed Mapper, as its name says, maps the distribution of plants across the state, and that helps us set strategic priorities at the landscape scale. And Weed Cut is a database of ways to control specific plants in specific situations. So first, the Calypsy Inventory. It's a list of some 300 plant species. Actually, Elizabeth is one of the people who has done the lion's share of the work on it in the past, and it lives on. They're either classified as invasive or they've been determined to be a high risk of becoming invasive in the future. And you can get from this table, you can get ID cards, you can link directly to the Cal Weed Mapper or CWM map for that species, etc. But most importantly, if you click on the plant's name, and you can filter it by Jepson regions, habitat types. But if you, this is obviously too small to read, but if you click on it, you get the plant profile for the given species. And that is kind of your one-stop shop that links to all the Calypsy newsletter articles that have talked about that plant over the years, all the Calypsy Symposium presentations that have talked about that plant over the years, and then it links to CalFlora, to CalPhotos, to other websites, sometimes UC Davis control information, etc. So this is a really good way to get information on any plant we list. Moving on to Cal Weed Mapper, this is a statewide mapping data set that relies a lot on CalFlora. Cynthia is going to speak in a moment, for data on where plants are in the state. So this includes those same 300 or so species that are in our inventory. And it also includes data that's not in CalFlora, especially expert opinion data about where a given plant exists. And we use this to help regional partners design landscape level prioritization, so designing projects. And we really hope that it'll come in handy in the future for meeting the global biodiversity framework's goal of a 50% reduction in introductions or spread of invasive species by 2030. We'd like to see a goal set for California, and we'll be working towards that. Cal Weed Mapper also lets you, so that last screen showed selecting a specific plant, seeing where it is. You can also select a region. It could be a county, it could be a national park. In this case, it's a eco-region, USDA Central Valley eco-region, and it will list all the plants found in that eco-region, kind of categorized by how much of them there are. So if there's just a few instances, then it may be an eradication target. If there's a lot of it, maybe it's a containment target. If it's not there, but it's found within 50 miles, maybe it's a surveillance target, be on the lookout for it. So lastly, the weed cut tool so the Cal Weed Mapper tool is funded mostly by the Forest Service. This tool is funded mostly by the California Department of Pesticide Regulation. You can see 8 of, I believe, 21 different non-chemical tools shown here. So this is a database housed at the University of California that contains best management practices for all these tools. But it also has a decision support function where you can tell it what plant you have or what types of plants you want to control and your situation. You're on a slope, or what kind of substrate do you have, or how much of it is there. And this will prioritize which management techniques are going to work best for your situation. We're currently working on phase 2, which will integrate herbicides and chemical methods into this. So that's the whirlwind tour of the tools. I would say there also are a lot of other resources on the CalIPSI website, from reports to best management practices to training videos and more, aimed to help us all be as effective as possible in controlling invasive plants and protecting biodiversity. Thanks.
And next we have Martha Volkoff from the California Department of Fish and Wildlife.
I come bearing good news. As of yesterday, well, let me start. You're all interested in invasive species, appreciate the importance of responding to invasive species, but if you're a natural resource professional or in academia, you may be aware that there aren't a lot of resources to respond to a new detection. So I've been in invasive species in the Department of Fish and Wildlife's invasive species program since 2008. And over those 15 years, the day I walked in the door into that program, on our agenda was rapid response funding, enabled to quickly deploy resources in the event of detections. We work really hard on prevention, but we know that there are breaches in prevention programs or just entire absences in prevention efforts. And so having funding set aside in order to respond quickly, because while we don't know what the next invasive species might be, we have some good ideas, but where and when and what, we don't know. So having some money set aside in order to respond, or for a pot of money for others to respond to, would be very advantageous in our efforts to contain any detections and potentially eradicate. So working for the government, we're sort of at the whims of the budgets and the finances of the state. And so you can't always, the potential for that is you sort of have to go with the flow and hope an opportunity arises. And so that day has come. Yesterday, the California Invasive Species Council California approved the recommendation of the California Invasive Species Advisory Committee to set aside funding for a rapid response program. And yesterday they approved our application and process. So the SISAC has been working over the last year to develop this pot of money set it aside, and then the next step was develop the process. So we will, in fingers crossed, the near future, have $860,000. The application period will be on an ongoing basis. The constraints on the funding are short-term. The purpose is seed money to respond in case of a detection. So it's not going to fund an entire program or project, but the goal is to enable an immediate response. Applicants or recipients of this funding, in order to make that rapid, need to be a government agency. Otherwise, if just given the contracting processes of the state and getting money into hands, if it were not another government agency, there would be some longer administrative process that would make it not rapid. So that's a constraint for minimum requests of $10,000 up to $100,000 per project, and to be completed within a year of awarding the grants. And so this opportunity will be on a continuous application basis. If there's funding available, you can submit applications and SISAC will review those and make decisions on those. Second opportunity, also this year, so 2023 has been a significant year for rapid response funding at the national level. They also have developed a rapid response fund, sort of a pilot program. That first granting of money occurred, the call for proposals was in August of this year. They had an outpouring of need, and these two programs do wonders for demonstrating this need, which I think will promote the continuation and the support of this in a longer term. So they are in process of selecting those projects, and those should be announced soon. But they just also announced another $960,000, and this is nationwide, a call for additional applications that they'll consider. And that closes again, well, it closes officially July 15th, and or until they grant out all the money. So that's all I have. Thank you.
And next we have Cynthia Powell from CalFlora.
Thank you, Elizabeth. Thank you all for being here. Following up on the theme from this morning, which I'm going to call redefining success. Doesn't have to be eradication. We could call it, you know, a threshold for control. Whatever your goals are, having data-driven or data for information to drive your decisions about what your goals could be as you define or redefine success. CalFlora is a database that for weeds, for the Delta, for anywhere in the state of California, is a way to kind of assess what's going on, keep track over time of history stacks showing change over time. Unfortunately, not for non-plants, so the green crabs, the nutria, the loach, sorry, but for plants. We are a nonprofit wild plant database. Here is our team, and Pete Fry was going to be here today. You'll see him at other weed events. He's part of our weed team here at CalFlora. I've been trying to decide what the best word is to define what CalFlora does on a big picture, and maybe you all can help me in the Q&A session here. I've been using the word aggregate. We aggregate data from thousands of sources, including EDD maps, iNaturalist, county parks, state parks,
Or do we compile it? Some people might resonate better with that word. Some people might like assemble, collate, gather. Doug used the word one-stop shop. Maybe I should throw that on this list. So CalFlora is a one-stop shop for finding where are these weeds in the Delta? Where have they been in the past, but now they're eradicated, or are they controlled? Anyway, what you know about the plants should be reflected in CalFlora in order to have a complete picture of what's going on. This is way too much text to read. You're not expected to read it. These are just maybe several dozen of the thousands of sources CalFlora has for where the data is coming in from. All those blue dots you see on the screen. I'll show you the screen in a minute. Anita's presentation had 2 CalFlora distribution maps. I saw another one this morning. All those dots showing where the species grow. Those are coming from one of these thousands of sources. So from this aggregated one-stop shop data set called CalFlora, which non-native plants currently grow in the Sacramento-San Joaquin River Delta? I used what grows here on CalFlora and just generated a list of Calypsi listed and also not Calypsi listed, so just non-native plants that are growing right now in the Delta. So there's over 350, this must be a greater than sign that turned into a funny different arrow, greater than 350 non-native species currently in CalFlora for that area. And of those, 120 of Doug's 300-ish list inventory are Calypsi listed. Just to focus on one of our favorites, you know, lightning talk, I gotta just pick one. I picked alligator weed. On this map, you'll see clusters of blue dots reflecting where the alligator weed is in the state. The bloom wheel, some photos. If we were able to scroll down on these photos, we would see Gina's fingernails, very nicely done with some more alligator weed for scale. Thank you, Gina. And this is the kind of, and then you have a link to Calypsi at the top. So you have the scientific name, the author or authors of the scientific name, the common names. In this case, there's 2. It's a perennial herb, it's not native, and then a link to Calypsi, its rating is high on the Calypsi list. And then if you go below the fold, which was too big for my screenshot, there's a lot more information about this species. Without you in this crowd and without these data, doing the research that we need to do to be able to control or eradicate or somehow be successful, or at least try and be successful in handling these weeds would be, I'm not gonna say impossible, but more difficult. And so that's why coming from, you know, this aggregated data set reflected in these maps is really important. Now, if you want to dig in even farther, CalFlora has a software called Weed Manager that you can use if you want to track your treatment, if you're using chemicals, if you're doing some of the other non-chemical methods that Doug was displaying on his slide. You can track all of that using Weed Manager, which is a section of CalFlora. But also just, if you find a weed, add it to CalFlora and it'll show up on this map. Thank you.
And once again, we have Nick Rasmussen from Department of Water Resources.
All right, so this is a particularly nasty invasive aquatic plant that has not yet been found in the Delta, but has been found in places close enough I wanted to get it on people's radar. So as the name suggests, the plant looks like a floating head of lettuce. It's about a foot in diameter. The leaves have wavy margins, and the leaves appear ribbed because of their strong parallel veins. And they feel velvety to the touch because of deep, intense pubescence on the leaves. And the leaves are connected directly to the plant without any sort of leaf stalk. And because it floats around, the water just has these long, trailing, feathery roots. It prefers still or slow-moving fresh water. It doesn't tolerate very much salinity. And because it's a tropical plant, it also doesn't tolerate freezing of the foliage. However, the seeds can survive for long periods of time at temperatures below freezing. So the plants can rebound after a cold snap from the seed bank, meaning that this plant functions as a perennial in warm places and as an annual in cold ones. As with many of our invasive aquatic plants, this is transported and sold by the aquarium and pond supplier trade. Here's just one example of something that's currently on Amazon, where you can buy these for a dozen of these for less than $1 a piece and have them delivered to Sacramento in a few days. So new introductions are likely the result of people improperly disposing of these cultivated plants, such as dumping of an aquarium. And so once introduced, the species can spread very rapidly through vegetative growth. And it also produces high numbers of highly viable seeds. And so in an interconnected system like the Delta, plants and seeds can move around fairly quickly to new areas. This is considered one of the very worst weeds in the world, not just aquatic weeds, but worst weeds. And you can see from this photo why that is. This species can rapidly cover entire bodies of water with tremendous amounts of biomass, which can result in the clogging and damaging of water conveyance infrastructure for water deliveries and flood control. It can hinder all kinds of water recreation, such as swimming, fishing, and boating. The high amount of foliage can result in a lot of water loss through evapotranspiration. By slowing the flow of water, it can create breeding habitat for disease-vectoring mosquitoes. And by depleting oxygen levels, it can kill off native fish. So this weed is now found across all continents except for Antarctica, spanning 40 countries and 23 US states, including California. So here you can see on this CalFlora map, there are a number of populations in Southern California where the species has been present for many years. But the impetus for this talk is that there are populations that are starting to show up in Central and Northern California. Specifically, it is in the San Joaquin River near Fresno, which is so far from the Delta, but is hydrologically connected to the Delta, so it could eventually make it to the Delta if it isn't controlled before it gets there. It's also been found by the USDA multiple times in the Russian River in recent years near Heldsburg, which is not hydrologically connected to the Delta, but it's not hard to imagine a boater accidentally transporting plants on their boat to a place like Rio Vista and then getting it in the Delta. I think these two examples exemplify the theme of the symposium today of connectivity. So where do we stand on preparation for the species showing up? In 2004, CDFA assigned a pest rating of B. That's their second highest threat level. I would like to see them reevaluate it because I know that they have revamped their pest rating process since then. And as you saw, the species is in other parts of the state than it was when this rating was first made. Last year, the Division of Boating and Waterways, who does control of aquatic weeds in the Delta, added the species to their watch list, which is great to have those people on the lookout for it. And that was also part of the reason that this species was on my radar in the first place. And as of just a couple of weeks ago, my department, the Department of Water Resources, requested that California Fish and Wildlife conduct a risk assessment for water lettuce. This is important because a risk assessment is required before Boating and Waterways can start conducting control of any of the species that are in the Delta. And this is kind of a new thing, trying a proactive risk assessment, with the hope that having this proactively done means that if this species does show up in the Delta, Boating and Waterways can start taking action fairly quickly, which increases the chance of eradication rather than long-term management. And again, as with my other talk, I just want to leave you with the information for the CDFA reported pest system. They've got both a hotline and a website with a lot of great information if you do see this species in the Delta or really anywhere in California. Thank you.
And our final speaker in this session is Thomas Jensen from the California Department of Fish and Wildlife.
Hello, everyone. My name is Thomas. I'm the field lead for our CFW Invasive Water Snake Eradication Project. A little bit about this project. We conduct trapping for 2 populations of invasive water snakes in California, likely introduced from pet owner releases. The goal of the project is eradication of both populations. We have one in Roseville and one in Folsom. And if Nerudia expand their range, they will likely outcompete the federally threatened giant garter snake, as well as have negative impacts on other native species and the potential to get into the Delta. Some of our trapping details here, Nerodia are highly aquatic snakes that feed on fish and amphibians. So we trap them using these floating, self-baiting aquatic minnow and crayfish traps. We float them with water bottles, and minnows and tadpoles and whatnot swim in that attract the snakes to them. So you can see an example on the picture there. We deploy these traps from May to October during the snake active breeding season, and they're checked daily to remove any captured snakes and release non-targeted bycatch. The picture on the top is from Roseville, and that's a simple site to trap. It's a single channel, 2 or 3 feet wide. So pretty straightforward, whereas our trapping location in Folsom is a little bit more complex. It's sort of a braided channel that's in an urban greenbelt, and the accessibility is a little bit more difficult there, which presents its own difficulties. So our progress report, since trapping in Roseville in 2015, we have removed 114 northern water snakes, the Nerodia Cipodon. And since trapping in Folsom since 2017, we've removed 172 Florida banded water snakes, the Nerodia fasciata pictoventris. So for the past 4 years, we've had 0 captures in Roseville, so well on our way to confirming eradication of that population. And as you can see from our graph here with our Folsom population, we've kind of had fluctuating capture totals, and this is due to increased trapping effort in certain areas in the Folsom and accessing new areas at that Folsom site. Okay. And once the snakes are captured and removed, they're euthanized at one of our CDFW offices. We collect snake measurements, snake fungal disease swabs, and parasite samples if present from each snake. Then the carcasses are accessioned to the UC Davis Wildlife Museum. Some have also been donated to California Academy of Sciences for their collection. So looking forward to the future, hoping to confirm eradication in Roseville and increase in focus efforts on our Folsom population to contain and eradicate that population. So thank you. Oh, not yet.
I'll let Thomas take a seat, and then we do have some time available for question and answer. And as with the other sessions, we'll be alternating between questions from the room and any online. So just a couple of reminders. If you're asking a question, Cesar will be coming around with a microphone, and please state your name and affiliation and also who the question is for. And for those online, if you could also type in who your question is for. You're asking questions in chat. And for the speakers, please turn on the microphone when you're answering a question with a little button on it, and turn it off when you're finished. And so, yeah, go ahead.
Hi, this is Tobias, and I'm with Friends of Lake Folsom and Manitoba. We're gonna just get that mic on second.
There we go. Need it for online, unfortunately. Okay, now try.
Okay. Crystal Tobias, I'm Friends of Lakes Folsom and Natoma. I live in Folsom. I heard about the water snake in Folsom, like in the early 2000s, and didn't realize that there was an active program to eradicate it. But I'm just curious, is that Willow Creek, the waterway? Okay, so that's right by my house. I've never really seen the snake there, but I will certainly keep my eyes open for it. Thank you, good job.
Alrighty, we have a question from our online audience. This is from Karen McDowell, and the question is mostly for Martha. Great news about the funding from the ANS Task Force and other funds. And Karen's wondering, is the $860,000 per year for rapid response, or does it apply? And does it apply to all invasive species, terrestrial and aquatic?
Yes, it applies to all species, terrestrial or aquatic. That money was deposited into the Invasive Species Fund in California, so it is unlike the potentially federal funding, not limited to aquatics. And there was a part A to that question that I glossed over. Oh, good question. No, it was a one-time, so into that fund, a couple of years ago, there was 5 million put in there. Of that, that's where that $680,000 was pulled from. It's looking like we may have lean budget years ahead. So that was a one-time allocation. We continue to advocate for additional funding into the invasive species account. Something you should be aware of that's upcoming, hopefully in the near future, early 2024, is a summit on invasive species, where we encourage all of your participation and voices to be heard for expressing the invasive species issue and needs. And so if you sign up for mailing lists, our Department of Fish and Wildlife Invasive Species Program has a mailing list. I know we all get too many emails, so I know what I'm asking here of you, but if you're interested in receiving information, please subscribe to our mailing list, and then the California Invasive Species Advisory Committee and the State Council, Invasive Species Council, has a mailing list also where you can subscribe to receive information on upcoming meetings and opportunities to have your voice heard in the need. And neither of those email lists have an enormous amount of email traffic, so you won't be bombarded with info.
I'd like to quickly follow up. Given that CalIPSI has been the primary advocate for all of this, the summit has happened once before. It's at the Capitol building. It happened in 2018, and after that was when the bill that Assemblymember Grayson passed, 2470, he said, which created that Invasive Species Council, created the fund. That allowed us to then advocate for money to go into that fund, which has resulted in this. As agency employees, which I imagine many of you are, you can't really advocate for your legislators, but going to a summit at the Capitol, that's why it's there, is meant to educate legislators on this issue. So it's a really great opportunity to be able to, within the confines of your job, to be able to influence what happens in terms of policy and funding.
Hi, Lisa Wanger, Delta Independent Science Board. This is for Jenny Wiggins. I really appreciated that use of eDNA. That was very exciting. But I'm sorry, I apologize if I missed this in your talk, but I wanted to understand, in the end, did the eDNA help you target, or did you have data to compare whether random was just as good?
Yeah, that's a good question. So yes, we definitely say that using eDNA first as a guided tool was definitely helpful in where to set the traps. In the pilot study that we did, we just tracked randomly, and only a subset of those sites were tested for eDNA. And so essentially we found that the methods between trapping and pairing that with eDNA agreed 82% of the time.
I have a question. Quite a few of you presented invasive species that I can find at my nursery where they sell pond plants. And so I'm wondering, have you ever seen a successful public education campaign to help the public understand why we shouldn't be buying these plants and putting them in our garden ponds?
So, so yeah, I feel Nick's pain about, like, can't we just get these things out of the market? So this is not an aquatic solution, and others may know from Habitatitude or other programs in the past, but from terrestrial gardening, the Plant Right program has been around for about 20 years, and that is not a quick fix, but it's basically built relationships between agencies, universities, environmental organizations like ourselves, and the nursery trade. And so that's identified key plants that everyone can agree are problematic and put them on a list, voluntary list, encourage nurseries not to sell them, encourage growers not to grow them, encourage consumers not to use them. So that's one approach. I would say with some of these things like the water lettuce, it seems like a really ripe example for encouraging aquarium owners to be really responsible. It's pretty difficult. I think Australia was brought up as an example where they have more of a green list. You have to make sure it's safe as opposed to a red list, like only the things that we know are bad can't come in. It's pretty unlikely that's going to happen in the US. I mean, commerce is king, but I think that there is a lot that can be done with consumer responsibility and store commercial responsibility too. You can get some benefit by being a green industry.
And I would add to that the Master Gardeners program through more agricultural, ornamental, but if you can prevent them from being in the store to begin with, I think that's even more powerful than the interpersonal connection with Master Gardeners. And that aside, there's all the illicit, illegal movement, even if it is restricted in the state of California. So again, Habitatitude, I don't know if all of you are familiar, a U.S. Fish and Wildlife Service campaign targeting pet ownership. And Cesar could probably answer more questions if you had questions about Habitatitude.
Hi, my name is Barrow Maris, and I'm at the Department of Water Resources. I was just curious for the Rapid Response Fund, if any agencies or entities are applying for the use of these funds, since it is a one-time allocation, is there any mechanism by which applicants could, in their application, specify that they're going to repay the funds so that this rapid response fund could function more like a revolving fund?
Speaking only for the state rapid response funding, I think we would be happy to hear any suggestion that might be an opportunity that would help extend the funding to other needs.
Hi, Amy Williams, Yolo County RCD. A couple of questions for Doug. I really appreciate the Calipsy pools. Do you know what the timeline is for phase 2 of the weed cut that includes the rest of the pools?
I should. I mean, we're in the middle of a 3-year project, so I'm going to say probably 1.5 years till that's out, but it may be sooner.
I'm also curious what the definition is for like how surveillance is defined. Is it specific or is it specific to different species?
In Cal Weed Mapper, it's a very generic definition of anything that's not in the region of concern, but is within 50 miles. So sometimes, you might be within 20 miles excuse me, of the bay, and Spartina comes up as a surveillance target, but you're up on Mount Diablo, so you know that's not accurate. But it's a very generic. You have to interpret it, but it's within 50 miles.
Rachel Wigginton from the Delta Conservancy. I had a question for Anita. You had one site, I think it was the freshwater no iris site, where you didn't observe any pollinators, and I was just surprised by that, and I was curious if you had a hypothesis for why that might be.
Yeah, so one of the major reasons why I think we didn't see much pollinators within the freshwater site is because we really don't have any vegetation that has actual flowers in the freshwater site. It's mostly willows that are within that area. But when we did start our observations and research of the iris back in 2017, there was definitely a lot more of the native vegetation there. And iris has definitely outcompeted a lot of the native vegetation that used to be there and used to flower. So that definitely has an effect on why we're not seeing a lot of the pollinators within the freshwater site.
More questions online? No? Okay. We have time for maybe a couple more questions in the room.
I wasn't sure, in terms of the yellow iris, of yellow flag iris, do you think it could be a positive, situation for native pollinators? Has anyone ever brought that up to you?
Yeah, I do get that asked a lot. Yeah, I think it is nice that we're getting a lot of the pollinators in that area, but the thing is that we're getting more of the pollinators going towards the iris and not the actual native plants. So that really does hinder, what we're seeing within that area. And we are definitely seeing like that decrease in like the native vegetation, like it's in like the freshwater site where it's definitely outcompeting a lot of the natives in that area.
Ask my colleague Cynthia Powell, since, as you made the point, everybody, a lot of people, not everybody necessarily uses CalFlora. How the heck can they help support CalFlora?
Oh, good question, Doug. You can support CalFlora in many ways financially. You can get a subscription if you work for an agency that uses CalFlora for your work. You can get a CalFlora subscription that way. If you want to use Weed Manager to track your work when you're treating weeds, you can support CalFlora that way because that's also a subscription. And you can support CalFlora by contributing your own observations, your photos, your knowledge. You need, if you want to do some small amount or large amount of volunteering, we're always looking for volunteers to help curate the pages. All 8,500 plant species in the state of California, that's all species that grow wild, have a taxon page that's that alligator weed with the map and the blue dots and the names and all that. So we need curators for those pages. Staff can't possibly cover all that. You're always full of good ideas, Doug. What else do you think I should be saying right now?
Well, I think that covers it, but just the acknowledgement where you're talking about connectivity and the need for landscape level planning, you can't do that without data. And an organization like CalFlora is really a backbone organization. We're similar. If we go to try to get a grant, we don't have something really bright and shiny to show for it. We just help everybody else get their work done. So anyway, if your agency is part of the California Ecosystem Studies Unit, or not sure if that's the exact term, the CESU, that's a way that agencies can fund universities and nonprofits in doing things. So maybe there's a way, eventually, we can dream of the day where backbone organizations that really do help the landscape level connected work happen aren't scrambling.
Thanks for bringing that up.
Okay, and I think with that, we will go ahead and, sorry, Caesar, do you have one other thing you want to say or question?
For Nick, with regard to the water lettuce, where it's occurring now, and what eradication efforts are underway, and have those shown success?
Yeah, I don't know too much. This has just been on my radar because folks in other areas have been telling me about it. I believe the USDA folks, where they found it in the Rush River, they were careful to remove all the material that they found, but I don't think that they have done extensive surveys because that's not what they were out there doing. I think they might have been doing yellow flag iris or water primrose or something. But yeah, I don't think they know exhaustively where all it's been found, but when they have found it, they've removed it. And I do know that it's been present in Southern California for a long time, and they have tried all kinds of things, maybe even including biocontrol. I don't know that they've been all that successful, but I don't know a lot of the details. So yeah, the longer I've worked on aquatic weeds in the Delta, the more convinced I am that prevention is really like the only solution that we have at our disposal. So yeah, hence the reason for trying to get the new weeds out there in the public view as much as possible so people can report them.
I think we're going to have to end it there and let people go to break, but certainly, hopefully the speakers will still be around at break if anyone wants to speak to them individually. So maybe let's give one more round of applause for our speaker. And we have break until 2:45.
So I'm talking about harmful algal blooms. Harmful algal blooms can form in marine, estuarine, freshwater environments. In marine environments, they're usually formed by dinoflagellates and diatoms. And in freshwater environments, in the freshwater portions of estuaries, cyanobacteria are the species that are going to cause the harmful algal blooms. And so I'm going to focus my talk today on cyanobacteria. Now, cyanobacteria are actually one of the oldest organisms on Earth, and so they're native to a lot of environments all over the world. But I'm here today because they're also somewhat invasive in that they're expanding in geographical extent and severity in water bodies worldwide. So a quick overview of cyanobacteria. They're a bacteria, but they photosynthesize. And so their photosynthesis is actually responsible for the early evolution of oxygen in our atmosphere. They can tolerate a wide variety of environmental extremes. So you can find them in hot springs, you can find them in freezing environments, but they're most likely to be found in warm, slow-moving waters that are rich in nutrients. So cyanobacteria have a number of advantages that allow them to outcompete other phytoplankton species. They can survive under unfavorable conditions, so they can survive and they actually thrive in really warm temperatures. So 28 degrees Celsius is like a perfect temperature for them to grow. Desiccation, they can completely dry out. And when that area becomes re-wetted and there's enough nutrients, they can rebloom in a water body and they can dry out for many years. And they're also able to survive transport. So there's evidence that cyanobacteria can go through the turbines of hydroelectric plants and survive. They also are buoyant. So this allows them to move up and down in the water column. And this is advantageous because they can come up to the surface and harvest the sunlight and shade out other phytoplankton species. Then if the sunlight's too intense, they're able to move down slightly in the water column to avoid that extreme sunlight. They have anchinates. These are large, thick-walled cells containing reserved materials. Those can settle out to the bottom of the water column. And in the summertime, when they usually bloom, they can re-enter the water column. And then some species have heterocysts, and this allows them to fix nitrogen from the atmosphere. So one of the reasons that cyanobacteria harmful algal blooms are considered harmful is because they can produce a variety of different toxins. The most common of the toxins worldwide is a liver toxin called microcystin, and it's a namesake to the cyanobacteria genera Microcystis, which I'm going to be talking about a lot in my talk today. So microcystin poses a serious human health issue, but it's most commonly due to chronic exposure to the toxin. It's not typically an acute exposure situation where that it can harm harm. However, dialysis is one example that some people in South America had dialysis with microcystin contaminated water, and it killed about 100 people from that. But typically, it's a chronic exposure, and that's either through drinking the water, swimming in it, and like gulping the water as you're swimming in it, or if you're eating shellfish that can accumulate the toxin, or vegetables that are grown with the contaminated water. So I'm going to be focusing on the delta here. But hotspots of cyanobacteria harmful algal blooms, we call them CHABs, they form in coastal ecosystems worldwide. This is actually an area where we're seeing them become more invasive, if you will. They were more thought to be in freshwater areas, but we're seeing them in coastal environments a lot more frequently. This is due to various factors, but largely due to global climate change, which is freshening portions of the coastal environments, either through large storm events or flashy precipitation events. And then also these flashy precipitation events are causing excess nutrients to enter the water, and then also warming the water temperature. to make it more advantaged for cyanobacteria to grow. They've been in the Delta since at least 1999. Cyanobacteria themselves have been in the Delta for even longer, but blooms started to be observed in 1999. And certain areas, increasingly marinas and terminal sloughs, so dead-end channels in the Delta, are being recognized as CHAB hotspots. And what I mean by a hotspot is an area of dense cyanobacteria growth. These hotspots can then be transient in nature, and they can move downstream and pollute other parts of the Delta or other coastal ecosystems. Here in this picture in the Delta is the southern and central portion of the Delta, which is where we see the most severe cyanobacteria blooms. So one area that has anecdotally been identified as a CHAB hotspot is the Stockton area, which you can see in the red dot on my map. Most years, it develops dense cyanobacteria blooms. However, not present in 2023. That was an extreme wet year, as you all know, but it was still a surprise to me because it is pretty hydrologically isolated from the San Joaquin River. To exemplify how bad the harmful algal blooms can be in Stockton, we've measured microcystin over 1,000 micrograms per liter. The state of California has identified a danger level of microcystin of 20 micrograms per liter. So you can see that that's very high. So I wanted to show you some pictures of what cyanobacteria, and I'm really going to focus on microcystis in my talk because that to date has been the most problematic species that we see in the Delta. So on the left is the Stockton waterfront, and you can see that it forms this really bright green kind of oily scum. And then on the top picture is a close-up of that same scum. Then down is Mildred Island in the middle. And what I wanted you to see here is that there's actually some other cyanobacteria mixed in this picture, but the large like flakes that you can see, those are microcystis clumps. And so it looks different than the hotspot areas. And then on the far right is the Stockton Deepwater Ship Channel. And I'm gonna have several slides on this location later in my talk. But once again, this is a deeper area than the Stockton waterfront, more tidal movement, and you see these lettuce-like flakes of microcystis as opposed to dense colonies. So there's knowledge gaps on cyanobacteria that I'm going to address in my talk. There's really little research on these cyanobacteria hotspots, even though they're anecdotally identified as the worst areas for cyanobacteria in the Delta. So questions are, does the majority of cyanobacteria seed stock originate in these areas? And I have a diagram on the right that shows what this seed stock does. So it's up in the water column, the cyanobacteria are growing really well, and it cools down in the winter, temperatures are less optimal, or they've used up all the nutrients, they settle down onto the floor and then of the water column, and then once the water warms up again, they re-enter, and we have that cycle happen over and over again. And so we think these seed stock areas are very important for the Delta because they're inoculating the blooms. And so our hypothesis is that the hotspot areas are going to have more seed stock than more distal locations. We also wanted to address, do CHABs develop and grow throughout multiple areas within the Delta, or are they just growing in the hotspots and then being transported downstream? And then do hotspots have different conditions than areas immediately downstream? This is where I'm going to show a few slides on the Stockton area. So we have 3 years with different methodologies. So 2021 is a dry year. We looked at 8 sites across the Delta, and we collected sediment, and this was to evaluate the seed stock. We collected it in April, and so this is when the amount of seed stock that would be available to inoculate the summertime bloom. And then we also collected it in November. So this is at the end of the bloom season, and this is when we would expect the maximum amount of seed stock to be on the sediment floor. We use qPCR and DNA analysis to measure the seed stock in the sediment and to identify the different microcystis strains that could be present, and collected water samples in addition to the sediment from June to October, also using the qPCR and DNA analysis. In 2022, we collected sediment also in April at 8 sites, and then did a focus study on the Stockton area. So 11 sampling events in the Stockton channel, once a month, May, October, November. and twice a month, June through September. So this is like the primary bloom season in the Delta is typically identified as the June through November period. We looked at water quality parameters at every meter, and then we collected surface water and looked at nutrients, cyanotoxins, and then microscopy to identify what species were present. And then we also looked at sediment and pore waters because we wanted to see if there was potential for internal nutrient loading. 2023, that was our wet year. Had also had 8 sampling events twice a month from July to October. Only had one site this year, the terminus of the Stockton Channel, and that's where we saw those most severe blooms. It's also called McLeod Lake because it is lacustrine in nature. It is tidally influenced to see sea lions down there, but it really operates more like a lake than other portions of the Delta. We also collected the water quality every meter and then surface water for nutrients, cyanotoxins, and microscopy. So I'm going to go into a couple slides that show the results from our seed stock. So on the left panel here, I also had November 2020. So on the left panel, we have November 2020 and April, May 2021. And on the right panel, we have November 2021. So once again, when we'd expect maximum seed stock, and then April 2022. And that is the seed stock that is left over after winter. And so we're assuming these cells are either flushed away with high flows or that they're dying due to abiotic and biotic processes. So one of the takeaways here is the yellow bars are November. You see, the wording is incorrect, but we have much higher seed stock in November than April. And this is what we would expect. This is when the maximum amount of cells should be settling out of the water column. On the right-hand panel, it was really obvious that those greenish blue bars were highest in April to inoculate the blooms in Discovery Bay and Stockton waterfront. And indeed, in the summer of 2022, we had very severe blooms at both of those locations. Those blue bars are, because of the scale on the left-hand side, you can't see them as well, but in 2021 at Windmill Cove, we also had quite a bit of seed stock in April. So this figure has a lot going on, but I'm going to clue you into a few things. This is a network analysis. And so we used the DNA that we extracted from the sediment to look at how many Microcystis strains are in the Delta. We found 36 distinct Microcystis strains out there. And so what we were able to do is develop a molecular fingerprint. And we were able to use a network analysis to see how different sites matched up to one another. And that allowed us to see if there was potential for transport between locations. So the first picture or circle that I want to show you is this is a connection that we found between Buckley Cove and Windmill Cove. So this is just downstream of the Stockton region. The molecular fingerprints matched up with Old River and then Mildred Island and Frank's Tract. So this is suggesting that cells at Windmill Cove and Buckley are moving downstream and likely the same Microcystis colonies as we observed in Frank's Tract and Mildred Island. We saw something similar in Stockton at the McLeod Lake site and the downstream deep water ship channel. And then finally, we also saw a similar pattern at Discovery Bay in Indian Slough. So Indian Slough is just downstream of Discovery Bay. And depending if the pumps are on or not, it will pull the water either north or south in Old River. So now I'm just going to spend a few minutes on the Stockton area where we did a focused hotspot study. So this area is the Stockton Channel. It's very important economically to the city of Stockton because 30 cargo vessels pass through here monthly. They unload their goods at the port of Stockton, and then they turn around at the turning basin, which is site 4 on my map. The blue polygon is the Stockton waterfront, and number 8 is McLeod Lake. So the Stockton waterfront and then number 8 is where we see the most severe blooms. On the lower scale here, it's how far each of our 8 study sites are from the San Joaquin River. And so McLeod Lake, which is number 8, is over 3.5 kilometers from the San Joaquin River, which just shows how far away it is from its influence, especially during a dry year. So those 5 orange sites were our primary sampling sites. And this is a principal component analysis that we did looking at all of the data that we collected. So moving from left at the red dot, the San Joaquin River near Calaveras, we're moving from west to east. So McCloud Lake, the purple dot is our most eastern site. And you can see with that purple ellipse, how the McCloud Lake site really separates out from the other locations. So this whole entire stretch is about 5 miles long. McCloud Lake is the shallowest and it had the most severe blooms. And that site lined up also with these factors such as high chlorophyll, high phycocyanin, high pH, and high dissolved oxygen, which are all associated with cyanobacteria blooms. The Turning Basin and Morelli Boat Ramp, which is on the western side of Stockton waterfront, they're in the middle there. There was a dense bloom at Morelli Boat Ramp, but it was less severe than McCloud Lake. And then the yellow and red ellipses, those had much less microcystis than all of the other sites, and those really separate out from the McCloud Lake site. Principal component 2 explained much of the nutrients and the temperature, and that's because nutrients and temperature were high across all of our sites. So I just wanted to show you some other pictures of what that principal component analysis looked like. So on the left, I have chlorophyll. Once again, moving from left to right, we have water depth on the y-axis, and you can see McCloud Lake is substantially shallower than the deep water ship channel, and that's where you have that hot spot of chlorophyll or microcystis. These pictures on the right, on the far right-hand side is McCloud Lake. Once again, you see these dense green blooms, and then on the left, the deep water ship channel, which is about 12 meters deep, you have the flakes of microcystis, and the water looks brown. I also wanted to show you the microcystin. So this was the only toxin that we detected during 2022. Highest toxins at McCloud Lake, 270 micrograms per liter, exceeding the danger threshold. Dense blooms at Morelli Boat Ramp, but maximum toxin concentrations were low, below the warning threshold. And in areas where those flakes of microcystis dominated, toxin concentrations were much lower. In 2023, we had no observable cyanobacteria at the McCloud Lake site, which was very interesting. Really low counts of microcystis, and we only detected microcystin on 1 out of 8 sampling dates of 0.17 micrograms per liter. So why? I looked at the nutrients, and the red dots are 2022, the blue triangles are 2023. We have total nitrogen on the top, total phosphorus on the bottom. Anything above those black lines would be considered non-limiting nutrients to cyanobacteria. And you can see that the nutrients were quite a bit higher in 2022, but they also were fairly high in 2023. If you look at the San Joaquin River flow just downstream of our study site, I thought it was really interesting how 2022, we basically had negative flows there. So it's true, we weren't getting much influence in the McCloud Lake from the San Joaquin River, but you saw a huge influence on the San Joaquin River, most likely at the McCloud Lake site in 2023 when you had such high flows. So in conclusion, we had for the seed stock, we had a large depletion of overwintering microcystis cells at each study site. The microcystis cells are highest in hotspots, but cells are also in other places. Genetic data confirms there's a connection between hotspots and blooms in downstream locations. For the Stockton hotspot, we had a gradient of the most severe cyanobacteria at McCloud Lake and then decreased as you moved west. Toxin concentrations outside of McCloud Lake were low, and mitigation efforts should focus in McCloud Lake, but I don't have time to talk about that today. I ran out of time. Thank you. And the Central Valley Water Board was instrumental along with all these other partners.
Okay, I don't think we have time for questions. And unfortunately, Ellen has to head out to another engagement. So maybe try and follow up with her if you have questions. Next up, we have Betsy Wells.
All right, so I won't touch the mouse and I won't touch anything but the one button, the right hand button?
Yeah.
Okay, great. We're gonna make it through the rest of the day, guys. Thank you all for staying into the afternoon. We're in the home stretch, and I'm going to be talking today about jellyfish and clams. This work is actually just got accepted for publication. It's part of a suite of papers about drought and drought synthesis put together by the by IUP's drought synthesis team. And this was like everyone's been saying about all their work today, it was a team effort. I worked with Rosie, Laura, and Christina to put this all together. And it's been a pleasure to be doing this. But so this was, like I said, it's mostly, it started as a drought paper. But as you guys know, when you start looking at just one thing, it always expands out to a little bit more. Luckily, I'm in the afternoon, so most of you guys have said a lot of my introduction for me, which is that the San Francisco estuary has a lot of non-native species. Thank you guys. Some of our favorites out there, and that these guys can have a very large impact on the food webs on all different levels and in multiple complex ways. And I'm going to talk about some of those complex ways today, I hope. The other part that whenever people are looking at the San Francisco estuary is what's happening with the physical drivers. And in this case, like I said, we're looking at drought, which can have a really big effect on food webs. This chart that you see on the right-hand side, that's actually from one of the other papers in this drought paper suite that's coming out early next year in San Francisco Estuary and Watershed Sciences. And I'm really sorry the type is too small to see, but basically the top graph is chlorophyll, what happens in a drought, and the lower graph is zooplankton, what happens in a drought. And the red and orange means that there's actually more of it in the South Delta, and the blue means that there's less of it out in Suisun Bay. And this was a really great introduction to some of the ideas I'm talking about here, which is that drought can have effects that vary over time and space. When you say, is drought good for the food web? Yes, also no. And in addition to the sort of effects that they have, the abundances, distribution in an estuary with a drought or a wet year, as we've seen this past year, can alter really dramatically. A dry year, things sort of contract upstream, and in a wet year, everything sort of gets blown out downstream. And that's something that we see a lot. And so when we're talking about in the estuary, where in the estuary matters a lot. And of course, climate change is going to make everything harder because we're projected to have more extreme years, more frequently, with bigger effects as we go forward. So taking water years are different. Climate change is going to make them more different. Invasive species change things. This is a whole bunch of moving parts to try to understand when we're talking about the food web like it's one thing. So today, the study species that we chose to look at for this drought effort were 2 species of clams and a species of hydrozoan jellyfish. Propagule has been around since we think the 1940s. It's up in the freshwater portions of the estuary. The Corbula has a really high tolerance for almost anything, but you mostly find it in the sort of low to mid salinity portions of the estuary, sort of from confluence to Suisun Marsh and down into Suisun Bay. Both of these guys, for the purposes of this talk, we're going to say they mostly eat phytoplankton. That's not completely true, but for this talk, that's what we're going to concentrate on. Meodius, this is a Black Sea jellyfish, sort of in the low salinity zone as well. We think it showed up in the 1950s because people were recording seeing jellyfish that, in retrospect, we think were probably that because there aren't any native hydrozoans around here. Now there's 3, by the way. This one is from the Black Sea. It mostly eats zooplankton. Again, not just zooplankton, but mostly zooplankton. And I just want to say a lot of the work on Meodius has been done by folks at UC Davis and the Durand and Moyle labs. So I'm standing on the shoulders of local jellyfish giants here. The other thing to keep in mind as we're going through here, jellyfish float, clams don't. It's true. And with that means, and we're going to talk into what that means in terms of not just space, but time a little bit later on. So we had some hypotheses. The first one is basically what happens with abundance during drought for these 3 things. What happens to their distributions? Is salinity the thing that we think is really driving these during the drought, as opposed to absolute flow, temperature, turbidity, et cetera, et cetera? And what does this mean for the food web? We're kind of we thought simple questions to set ourselves. This is where I bang the drum about data, which is to say, Rosie and Laura put together a bunch of different data sets to look at jellyfish abundances here. And this is the work of literally hundreds of people, from starting in, you see that picture from 1959 from CDFW, using from Summer Toenet. How many people have put these data sets together? Lots. This is also where I banged the drum for open data, because these were all accessible and we could just use them. So thank you for all of those people who I'm not listing because I don't have enough slides, who put these data sets together. The third thing I want to say about this data, we're measuring jellyfish. These are all fish surveys. This is bycatch. None of these methods have been, for the toenets or the Suisun Marsh fish survey, have been evaluated for how well they catch jellyfish. So just keep that in mind. No one's sampling jellyfish, quad jellyfish right now that I know of. Clam data comes from a couple of DWR survey efforts, one that's happened either monthly or quarterly since 1978, again, long-term monitoring data, and another one that was a more spatially intensive but limited time-wise survey. This is my patch. This is what I do most days. And again, I just want to point out, clams are where you leave them. Clams, you come back to where clams are, because once they settle, they're staying there. Jellyfish are moving. This is just a look at where we were pretty much from the Benicia Bridge for the clams. We're going all the way out to San Pablo, up into as far up the delta as we could take boats. So that's not even all the clam survey states, by the way, but that was just the most recent map I had available. There were a lot of clams. So to jump right into the data, Meiotis varies across time and space. If you take that away and that's your take home, great. You find it more in the confluence, Suisun Marsh and Suisun Bay, and it follows the water. It gets up to the confluence in the drier years in yellow. It gets down more to Suisun Bay in the wetter years in purple. Fantastic. This is an easy story to follow. It makes total sense. If you look at it with models and ask which of the things explain it the best, I'm not showing you any models, don't worry. But salinity does explain it pretty well. The purple years are the wet years, and the y-axis here is the distance from the Golden Gate. So as you have a wet year, it gets closer down to the Golden Gate. In a dry year, it moves further away. Really intuitive, makes sense. Clams. Two clams doing two different things. Propagule is a freshwater clam, and it kind of noodles around in the North Delta, South Central Delta, and sometimes the confluence in wet years. But then you find Potamacorbula, which is more the brackish water, low salinity clam. And that one seems to like the dry years more. You see more of them in dry years or following dry years, and less of them in wet years when they kind of get flushed out to San Pablo. Mind you, it's not the actual clams that are being flushed out. It's that when they're reproducing, they're either reproducing less, and their young, who are planktonic for about 2 or 3 weeks, are going out further downstream. So there's a lag effect here. So this is what I'm talking about. Clams don't float, but their larvae, at least Potamacorbula larvae, do move, but then there's a time lag before you see them showing up in next year's population. Potamacorbula has been doing weird things since 2000. If you look, this is through time, starting in about 1986, which is when Potamacorbula first started showing up. Just take a look at what happens in 2000. It goes from sort of like low numbers to more and more and more. And one of the more interesting results that we found was if you look at pre and post 2000 in purple, that's the pre-2000. And it responds like, it responds kind of like the jellyfish with a 1-year lag, that after a wet year, you find them further downstream. Post 2000, they are less responsive to salinity. It's not that they're somehow like resistant to salinity changes. It's just that there's so many of them in Suisun Bay that even if the outer edges of the population slosh up and downstream, the centroid kind of stays where it is a lot more. Why is this happening? I don't know. You guys tell me. But this is happening, and we're seeing more and more of them. Okay, great. Jellyfish are moving with the water. Clams are moving with the water, but with a lag. What does this mean for their impact on the food web? So this was us looking at the filtration. This is potential eating. How much of the water column, the y-axis here is water column turnover. How many times a day is the water column being turned over by jellyfish in orange or clams in green? And this is where I have a big asterisk to remind me to tell you guys, we didn't base this on Meiotis filtration numbers because we don't have those. Meiotis is a hydrozoan. The closest we could get was Aurelia arida, which is a scyphozoan. It's the moon jelly. We don't have this basic information, so we're doing the best we can, but don't quote these numbers because at best, this is an order of magnitude for how much they're doing. And the short answer is Clams are there a lot of the time. The means are not that much different from the maxima. They're there, no matter what their water year, they're hanging on. Okay, so what if you take this number, this filtration number, and then you say, okay, for the years that we sampled, how much did they actually eat? What was their realized impact on the food web? Oh, that's a different graph. The clams are still out there. And this is in grams of carbon eaten. So the clams are out there. They're eating, it looks like a whole bunch in the South Central Delta because there's more phytoplankton there to be eaten. But then you look at those big orange bars. Those are all the jellyfish maxima eating. And what's happening here is that jellyfish bloom. They come out for a short period of time. They're eating higher on the trophic scale, so more carbon than the clams are. And they are, and when they're eating, they're probably showing up mostly when there's a lot of zooplankton to be eaten. So they show up, they clean up, they're gone. These guys are really only here in the warm months. They kind of retreat back down to their polyp stage, which we still haven't seen in a while, as far as I know, in the colder months. So what this means is that you have different regions doing different things. Like you look at Suisun Marsh, wet year, tons of clam grazing. Sorry, wet year, tons of jellyfish grazing. Dry year, tons of clam grazing. Suisun Marsh cannot catch a break. And because there's that time lag built in for the clams, let's say you have a dry year and then you have a wet year. Well, in the dry year, the clams are starting to settle up near Suisun Marsh because remember, they're moving upstream with the wet year, with the dry year, with the salinity moving up, and they're settling. And so the next year when it's wet, they're still already there. They're still filtering, but now the jellyfish are there. And you get the 1-2 punch on two different levels of the trophic ladder, and you can have these compounding effects of these invasive species, depending on what's happening with what water year and what's happening in sequence. Droughts are already kind of stressful. This is not something that we need, is having multiple trophic effects from invaders. So Meiotius, you mostly see it around Suisun Marsh. It follows salinity. They can have massive but short-lived effects. Clams are there all the time. There's a year lag. Something weird happened in 2000, and Botamocorvule has been booming ever since. And that you can have, and that they're still in Suisun Bay in large numbers that I don't really understand why. But beyond just the data findings, when we were putting this together, I started thinking about the theme of this workshop, which was connections. There's so many connections between abiotic and biotic, and between biotic, that aren't necessarily apparent. In the food web, like I said, clams eat phytoplankton, jellyfish eat zooplankton, and that's not all. Those are the major links, but we know we're not looking at some of what we think are the smaller drivers. We know things are connected regionally. We know they're connected on different timescales. And then I was thinking on the human level, the data sources, like putting this together, the synthesis effort. This was interagency, open science, people working together to make this happen. But we don't always know about the data sets. I think it was last week we found out there was an entire jellyfish data set from the enhanced deltas and melt monitoring we didn't know about. There's going to be a poster next year's IP. Go look for that. Talking to Christy Bowles today from CDFW. Turns out she's monitoring jellyfish too. I didn't know that. So we're still missing data. There's still gaps. And like I said, there's gaps in the natural history that we still don't know about. That as much as this is one of the most studied estuaries, and we know so much, and we're synthesizing so much of our data really well, there's still stuff that we're like, oh, we're still missing some of the stuff, especially some of the newer invaders. So thank you guys for listening, and thank you guys for your attention. And I just want to point out, we actually saw a double rainbow one day when we were out.
Yeah, we have a couple minutes for questions.
Thank you. This is me. This isn't online. I just want to say thanks, Betsy. This is like devastating, but also really good for us to know. And so I'm wondering if you looked at zooplankton abundance maybe as a variable in some of these models. I'm just curious if like we would see more grazing from jellyfish or clams if there's more food, which would lead us to believe that like food supplementation wouldn't therefore be that effective because it could just get gobbled up pretty quickly.
This is where I say I'm neither a jellyfish nor a zooplankton biologist. So, I mean, I think as with any question of food production, its effect on jellyfish, it is both possibly a driver, but you're also seeing the effect of grazing post jellyfish. Like when you go out and sample, what you're seeing is essentially the leftovers left on the platter. I think it's Robert Schroeder's PhD thesis was looking at what would bring on the blooms and how much they were eating at different times of year and during different seasons. And that jellyfish effect on zooplankton production and how that feeds into the jellyfish life cycle. I know it's complicated, and I do think that they are overlapping in timing. But beyond that, I think you've probably got something there, but I don't have the answers for that right now.
And because I forgot, this is Lauren Damon with the Delta Conservancy. Also, there's a question online.
Yeah.
Betsy, this is Tiffany Brown.
Yeah.
Betsy, could the shift in clam densities after 2000 be related to larger climatic oscillations, such as the PDO, which had a big shift in 1998 to 1999?
It's possible, because it's been 23 years since 25 years since that last big PDO. I would want someone who knows more about larger climactic events to weigh in on how much that affected the estuary and how much we can expect that to feed in. I suspect folks like Jim Claren and Jan Thompson, who do a lot more on climate and hydrology as it relates to these guys, might be better sources for that. We had a more limited data source than we want, and we didn't have enough data to have multiple events like that. Sorry.
Thank you.
All right, thank you.
Can you see your slides now, Evan?
I can, yeah.
Perfect.
Excellent.
So just say next when you need me to advance the slide.
Okay, I sure will. And I took out a number of animations, so I'm going to be saying next a lot. So I'm going to talk about connectivity from a somewhat different angle, and that is the interaction between species, and specifically with respect to hybridization between Wakasagi and Delta smelt, concerning the potential vulnerability to Delta smelt. And I know there are a lot of people, I'll just say in advance, that have interest in this from the management and the regulatory perspectives. I'm not going to speak towards policy, but I'm going to be talking about information we've learned or need to know to set up better, more informed decision making from a regulatory and management perspective. So next slide, please. So I know this is a pretty broad audience, and so I'm going to start just with some basics on hybrids and hybridization. So hybridization being the event, basically, and hybrids being the offspring, should any be viable. And next slide. And one of the things is that hybridization is pretty common. It's pervasive in the plant world. It's pretty abundant in a number of animal species, and fish are particularly prone to it. In one of the classic papers on freshwater fishes, predominantly was from the 1950s, Carl Hubbs just showed how prevalent it is among fishes. And it's something that can occur under a variety of environmental conditions. It can be of natural occurrence, that is between native forms, that's just part of their evolution and ecological history, to hybridization that is anthropogenic, typically associated with environment, habitat change or disturbance. And the outcomes can vary a lot. They can range from inconsequential to complete admixture of species. And, where we tend to be most concerned is when this involves non-native species hybridizing with natives. But even in the case of native species, hybridization between them can be of concern. For example, in altered systems where species may become rare. In those cases, it may be more likely to hybridize with a native species, or that disturbance could just disrupt a natural historical phenomenon between the species, but then it becomes problematic. But I'm going to focus predominantly on, well, entirely on non-native and native species hybridization. So next slide, please. And I'm going to talk about 3 of the osmerids in the Delta, in the San Francisco estuary, the native Delta smelt and longfin smelt, and then the introduced Wakasagi. Next slide. So we know that hybridization between the 2 natives, Delta smelt and longfin smelt, occurs. It's rare and apparently inconsequential. It's not surprising because they're pretty distantly related in the family. And next, similarly, next, with Wakasagi and longfin smelt, there are no records of it. We don't know of it happening. It could occur, but it's similar to Delta smelt. It may be likely to be inconsequential, not a guarantee, but it's not been detected yet. Next slide. But between Wakasagi and Delta smelt, next slide, it's complicated. And it's complicated, it potentially could be complicated biologically, but that may not translate into it being complicated from a management and regulatory perspective. And we need to know more about it before we'll know how complicated that may be on the more on the management policy side. Next slide. So I want to step back and just talk about Wakasagi. The next couple of slides will be Wakasagi just in the Delta, and then transitioning to talking about it with hybridization with Delta smelt. So next slide. In the Delta, as many of you may know, Wakasagi were introduced to California reservoirs in 1959. Next slide. And this is a species that comes from the Western Pacific, from Japan. Next slide. And it's distantly related to Delta smelt within the genus. It was introduced from Lake Suwa, Japan. Our recent genetic data are very consistent with that, showing that the form here does in fact align with the population from Lake Suwa. And it was documented as early as 1990, possibly was in the Delta earlier than that it gained entry to the Delta. But by 1998, it was broadly distributed within the Delta. Uncommon, but broadly distributed occurrences. And additionally, in more recent years, there's been evidence that it spawns locally in the Delta. And next slide. In recent years, there's also been an uptick in abundance of Delta smelt. of Wakasagi. As Delta smelt, the solid black line that's declining, as Delta smelt has declined, Wakasagi has recently increased. And next slide. So with that in mind, we can talk about.
I'm going to ask you to pause for just one moment. We're having some technical difficulties in the room. I apologize. So it's
no worries. That's fine. I'll take a drink of water.
But Evan, could you speak again just to make sure we can still hear you?
Yeah.
Right. Is that the correct slide we're seeing?
That is, yes.
Lovely. Thanks, everyone. All right, we're back.
Great. So Wakasagi and Delta smelt hybrids have been known from the Delta for quite a long time. They were first detected in 1994, and more recent studies, particularly this one from Benjamin et al. 2018, has found that they're not only the Delta smelt and Wakasagi hybridized, but they're predominantly limited to the first generation of hybrids. That's just hybrids between the 2 parent species. So next slide. What's interesting is that the catches in the wild are asymmetric. That is, all of the F1 hybrids have had a Wakasagi dam, so mother from the hybrids. Next slide. There also have been some back crosses detected. That is a hybrid that has mated with the parental species and produced offspring. In those cases too, have only a Wakasagi mother. Now there's really limited data on this, but among these, about 20% are back crosses. So that's not a trivial number, but again, this is limited data. And the next slide. But what we have not seen in the wild is a hybrid that involves a delta smelt mother. And it's not real clear why this is. It is consistent with some inherent incompatibility between the two species. They're fairly distantly related within the genus. It wouldn't be surprising. And this has happened before in other cases. So it's a reasonable assumption that that could be an influence. But there are some other things that potentially could be explanatory as well, potentially in combination with incompatibility. So next slide. And what else is happening is that I mentioned that Wakasagi had increased recently, but delta smelt, the population in the wild, has collapsed. And starting in 2021, experimental release of cultured delta smelt began. So there's a lot of uncertainty going forward with questions about the ability to rebuild the population in the wild of delta smelt. So next slide. So to mention, there's a lot of uncertainty here. And next slide. And with that uncertainty come challenges. One of them is we have some gaps to fill. I mentioned the asymmetry between hybridization, but it's largely unexplored. We don't know the cause of that. In addition, that we're having increased and perhaps increasing difficulty with IDing larval osmerids, larval smelt to species. Now that may not be because of hybridization. It could be, for example, that we have just a better representation of Wakasagi diversity, morphological diversity. But we don't know right now, in part because larval smelt are preserved in formalin, and that's proved to be a challenge for genetic analysis. So we have very little information, essentially none at this point, on the incidence of hybrids among larvae. And also there's some potential drivers that we need to consider. One, I mentioned experimental release of delta smelt, but which are potentially naive fish. They come from culture. They've been in culture for quite a long time. But also with respect to this asymmetry, there are a couple of aspects of Wakasagi biology that could help explain that as well. For example, Wakasagi reproduce earlier than delta smelt. So there's some overlap. So potentially Wakasagi gravid females are available as delta smelt males are becoming available to the system. And similarly, Wakasagi are just vastly more fecund than delta smelt. So it could be a numbers game. That's another possibility, that we're just more likely to sample hybrids that have a Wakasagi mother. And this has led to lingering questions about the nature of hybridization between these two species, and in turn, from the management and the regulatory standpoints, it's hard to know how to grapple with it when there's not enough information at this point. And as I point out to the right here, there are a lot of agencies and institutions, researchers, and the hatchery that are very interested in these questions, but we need more information. So next slide. So that gets to what risk are we really talking about when we're talking about hybrids? Next slide. Oh, next slide. So I mentioned there's a lot of uncertainty about Wakasagi, but now that we have experimental release of cultured delta smelt and moving towards a supplementation program, this is really a question within the context of these two programs. And what we're trying to do is produce delta smelt in the hatchery, release delta smelt to the wild, have them recruit in the wild, and build the population back up to a large enough level that we can capture wild origin delta smelt again and return them to the hatchery to complete the cycle for sound genetic management. And we want to release only delta smelt and return only delta smelt, not any influence from Wakasagi in terms of their genes. So next slide. So that presents us with potentially a twofold risk, and that is we have genetically managed production. We don't want to have the risk of Wakasagi genes infiltrating the hatchery population, the refuge, or the supplementation production there for this very vulnerable species. And although we have less control in the wild, there's a concern about hybridization, particularly as we're releasing delta smelt, and as Wakasagi, at least for the last several years, have become more abundant. But even if hybrids are inviable, or largely so, if these fish are willing to mate together, there's potentially a recruitment risk. So that you can think of it as a tax on recruitment, that if these fish, if delta smelt are wasting reproductive resources to hybridization, that puts downward pressure on our ability to have the population of delta smelt grow in the wild, to grow in abundance so that we can return fish in historical numbers back to the hatchery. So these aren't certain yet, but these are the two types of risks that really are we're focused on at this point as important ones with respect to the hybridization between these species. Next slide. So what do? Well, we're not catching many fish in the wild at this point, but we can learn things about spawning, for example, to get a better idea of how these fish interact together. And I'm going to show two slides here. Both of them have posters. I don't want you to look at all the details. If you're interested, you can go back and look at that. But they're really just to show you how much work we put into understanding spawning of delta smelt, and then second, how we've leveraged that to start investigating hybridization. So next slide. Our work with spawning behavior has been led by Gene Tsai at FCCL, the hatchery for delta smelt. And through this work, we've learned a lot about delta smelt spawning, when they spawn, how they spawn behaviorally, where they spawn, the habitats they spawn in. And through these methods and the information we've gained, we've been able to leverage that to next slide, to apply it to understanding hybridization between delta smelt and Wakasagi. So we're interested from two sides of this: the pre-zygotic, that is the events leading up to hybridization, and then post-zygotic, what happens after species mate together. And this is important because we're really investigating what are the barriers or facilitators, the influences potentially on hybridization between these species. And remember, if you look at the upper right, I mentioned that there's this asymmetry. Well, we want to know, why might that be the case in the wild? And what we find from our experiments, next slide, that behaviorally and in terms of egg or larval viability, we see both crosses. So fish are willing to mate together. Both species mate with the other. And their eggs are viable, and the larvae are viable up to 10 days post-hatch when we had terminated the experiment. So there's a mismatch here between what we see in the wild and what we can create in the lab. And so under certain conditions, there are no apparent barriers to hybridization in either direction between these species. Now, that's only up to in a small window, but it does help inform our understanding of risk with these species. So next slide. And it comes back to genetics and recruitment risk. Next slide. And you might ask, what now? What do you do with that? Next slide. Well, we have several studies in progress now. We're conducting additional spawning experiments where we give fish a choice between spawning with the same species or spawning with the other that is available at the same time. you'd think that might be straightforward, but it's not always the case. Preference isn't always so obvious. And we also are doing complementary embryo viability experiments in conditions of salinity and temperature that are more representative of the range of conditions that these fish would encounter in the wild during the spawning season. We're working towards new genetic methods to identify larval delta smelt. And that will benefit us not just in terms of assessing hybridization, but we're in terms of IDing larvae and with respect to take, making sure we're getting good IDs. We've had a lot of difficulty, as I mentioned before, identifying fish morphologically. So we have a complementary study to look, and this is with CDFW and Fish and Wildlife Service, to look at morphological IDs of delta smelt so that we can move forward with a much better understanding of larval smelt population abundance and hybridization potentially. And we're continuing our efforts on modeling and monitoring of cultured and supplemented stocks. And we, for instance, have been talking to Will Smith in our office about whether Wakasagi may be a useful covariate for delta smelt recruit. So next slide. And I just want to sum up with the contributions we've made here. And that is we have moved from an abstract set of risks to reducing the uncertainties and moving towards a concrete assessment of the risk that we're carrying with delta smelt. And we've done that through leveraging our knowledge we've already gained from wild populations and prior experiments, both in terms of hybrids and other studies not related to hybridization. And from that, we've been able to move towards lab experiments and other studies that refine our understanding of delta smelt by Wakasagi hybridization and its consequences. And in turn, this will have several benefits. We're moving towards more informed management and regulatory decisions. It will help with informing delta smelt culture and monitoring programs, and ultimately our desire to have an attempt to have an effective supplementation program for delta smelt. And next slide. And I have a lot of people to thank here from Davis and the Fish and Wildlife Service and the Bureau and many more that have been instrumental to this effort. So thank you.
Perfect. Hey, good afternoon, everyone, and welcome to the last talk of the day. My name is Rachel Hutchinson, and like Cesar just said, I work for the Tahoe National Forest. I used to work for UC Davis, and that is where I did the bulk of this research, and also when I was at University of Idaho. So I was actually really excited to be able to come down and talk to you today because a lot of this work I did over a decade ago, and I did it for just about a decade. So it's kind of exciting to me to be able to come back and think about the perspective that I've gained in this last decade about the work that I did. Sometimes it feels like it was yesterday, and sometimes it feels like a really long time ago. So I'm going to try really hard to stay focused on sort of the management implications of a lot of the work that we did. This study went on from, I'm going to talk about work that happened from 2002 all the way until 2013. And what that means, we did a lot of different experiments during that time to try to learn as much as we could about Lepidium latifolium at the Cosumnes River Preserve. And we focused on floodplain, riparian environments, and more upland grassland areas. So that's what you're going to hear about today. So the two primary management related questions that I'm going to try to talk to you about today is how does flooding or that connectivity or the restoration of that connectivity affect the population growth of Lepidium, the efficacy of treatment, and then that native species regeneration or how the plant community responds after you do those treatments to those areas. And then how well do these herbicide treatments that we tried actually work in these two very different plant communities, riparian floodplain and grassland. So the study location, the Cosumnes River Preserve, I don't know how many of you are familiar with it, but it is a lovely place that I spent a lot of time in a long time ago. And we focused on Lepidium latifolium, which is a perennial species, Brassicaceae family. It reproduces via seed and propagule. It can float through water and regenerate. And one of the things that we were trying to do here is this study was sort of instigated because there were some experimental levee breach restoration efforts, which at the time were experimental. This was in the late 90s. And as a result of those experiments, and I can't point, I thought I was gonna be able to point, but in the area that looks kind of like a triangle, that's where those experimental levee breaches occurred. We had perennial pepperweed coming in within the first couple of years to that newly restored, burgeoning, very exciting riparian forest and wetland community that was previously a tomato field. So that instigated these two different studies that I'm going to talk to you about today. One is a population growth study where we were looking at how, it was really like we were counting lepidium, how much lepidium was there, how big were the patches, and then the other was an herbicide treatment study where we were looking at how can we get rid of this lepidium that is growing. And let's see, sorry, I have notes because again, it's been 10 years. So these two studies, I think they were not necessarily intended to be linked together, but in the end, the observational study allowed us to have a lot of insights into the sort of growth and the trajectory of perennial pepperweed in the locations where we were also doing this herbicide treatment study, and it helped us to develop a little bit more of a holistic approach in terms of what our management recommendations were when we were done with the project. So to jump right into the population study we conducted on the triangle floodplain, this area here that I have on the map, in between 2002 and 2007. And so what we did, we went and we visited every single perennial pepperweed patch that we could find, and that was if it was one stem or 10,000. We walked the perimeter of it, we GPSed it, we counted all the stems, we estimated cover, and that was back when, it was like a giant backpack Kimball GPS with those giant batteries and the antenna. You guys were living in that time, you know what I'm talking about. Hopefully all those batteries got recycled properly. And what we were trying to do was really understand how is the species growing and what was going to be the impact on this, again, this newly restored floodplain. So we repeated the study annually, and we really quickly realized that in really wet years, because of the connectivity and the restored sort of process on the floodplain, some of the patches, they just disappeared, they were eradicated. And that was totally in line with some of the research that was happening at the same time that said, if you inundate perennial pepperweed for a couple of weeks, it'll magically disappear, which we don't, that's not a good management tool in a lot of places because you can't just put everything underwater. But we were kind of excited by that because, oh, well, maybe this is a way to manage pepperweed. However, what we were seeing is that when you ended up in another dry year, you started seeing the perennial pepperweed start to grow again. And one of the things that we were able to do as a result of this work, because we were so obsessively going after every single patch, is we were also able to determine whether or not the growth and the shrinkage of these patches was spatially dependent. So what we were able to find was that actually there were very specific areas where we saw perennial pepperweed grow really fast or shrink really fast as a result of either a wet year or a dry year. And what I would say is that it was that the growth and the shrinkage was most prevalent in areas where there were levee breaches, so where there was sort of in the direct flooding zone, or in areas that were just slightly lower than in other areas, maybe like up against a levee where flooding would be happening for a little bit longer inundation, or they would have a longer inundation period. So that decline in spends that we were seeing, it would knock back the species. And if you were the land manager, and we were trying to think like a land manager at the time, and I was a researcher, now I'm a land manager, we were sort of like, oh, this would give you a leg up, right? You give a wet year, it knocks back Lepidium latifolium, and all of a sudden you have less perennial pepperweed to treat. The unfortunate thing, of course, is then you have a dry year and it pops back up again. However, it is possible that this is actually sort of like a natural control mechanism where you no longer have to worry about the exponential growth of this species in this area where this disturbance process of flooding has been restored. So, in dry years, the species grows, and then in a wet year, it'll dip down, and then another dry year, it'll come back up again. And if you don't have other tools at your disposal, like herbicides or tarping or anything else you might want to do to perennial pepperweed to try to make it go away, you can sort of lean into the fact that the next time there's a wet year, the populations will be somewhat moderated, or the growth will be somewhat moderated. So that's something that we have really thought about, and we really pushed when we were talking to the land managers at the summit about how they might manage those environments. However, we also have things like levees, right? And we have things like upland grassland areas. They don't have that same disturbance process. They don't have that same connectivity and that opportunity for inundation. And so that's sort of where the herbicide study comes into play, to try to understand what's the interaction between flooding, the interaction between herbicides, and then the interaction of like when you don't have flooding at your disposal to help. Okay, so this study was conducted between 2005 and 2013, and there were a few questions we were asking. The primary question was, which herbicide was more effective? Was it glyphosate or chlorsulfuron? And then the second was, what did the recovery of the vegetation community look like post-pepperweed? We also wanted to understand if there was a difference in treatment efficacy or in vegetation community response after herbicide in either grassland or riparian floodplain communities. So, and this is a slide that shows the map of the 4 different treatment locations, and at each one of these locations, we had 4 replicates of each of our treatment types. And these are all of our treatment types. So we had an invaded control, where it was just perennial pepperweed. It was in a 3 meter by 3 meter plot, 9 meters squared. We had a mow control plot where we mowed it and then left it alone. We had a mow chlorsulfuron plot, and we had a mow glyphosate plot, and then we had a non-invaded control where we were just looking at the reference community to better understand what the vegetation was doing. So what we found was that, let's see, what did we find? We found that both chlorsulfuron and glyphosate were super effective at killing perennial pepperweed. Chlorsulfuron was a little bit better in the long-term outlook, and but what we also found, you can see this in here somewhere, is that in the control invaded plots, in what years in the riparian floodplain area, oh, sorry, I'm not supposed to touch it, sorry, in the riparian floodplain plots, which is on your right there, stems decreased, similar to what we saw in the population study. So again, we had a little bit of trouble at first trying to understand, was herbicide actually almost just as effective as just flooding, or is it the herbicide that worked really well, or was it the combination of the two? So we spent a lot of time talking about that and working through that. One of the more important things that we were focused on was what the plant community response was after. And one of the important things that we found is really focusing on what that reference community was. We also had a seed bank experiment that went on at the same time to understand what was already present in the ground and available to regenerate after we would spray. And then later on, we also did a hydrochloric study where we looked at plants coming onto the floodplain in the floodwater to see if there was sufficient material in that as well. Generally speaking, our floodplain environments had a higher native diversity, lower non-native diversity that was not significant, and our grassland communities had higher non-native composition and less native species. And so this, in terms of what was our expected outcome after we finished our removal of perennial pepperweed, we really leaned into this in order to understand what was going to happen after. What did happen after was in areas where we removed the perennial pepperweed, right? And then in our riparian floodplain environments, we saw an increase in native plants. We saw that peak about 5 years after we sprayed the herbicide. And then, but in our grassland communities, we saw it be reinvaded by non-native grasses, which is exactly what the reference community was. So it wasn't a surprise. It was just a little bit disappointing. One of the things we were most worried about was the propagule pressure that was going to exist on an annual basis every time it would flood in our riparian environments. And so the way that we tried to understand that was by looking at turnover through time. And what we saw was that, of course, riparian floodplain areas, they were turning over a lot every single year. So we were seeing different plants in one year from the next year, and where the grassland was pretty static. This was, we were able to directly correlate this to elevation, which could be a proxy for how much flooding these places were experiencing. And this helped us to really understand that when there were two things that were going on in the riparian floodplain. One is when you had a big flood year, you ended up with lower perennial pepperweed, and it also was an opportunity for the native plant community to come back. And so in those years, it wasn't just the perennial pepperweed died back, but it was also the natives had a better possibility of recolonizing because those sort of abiotic conditions were right for them to reproduce, and they were able to sort of outcompete the non-native community. And I think, let's see, in conclusion, I tried to keep this as short as possible, knowing it was the last talk of the day. We did find that wet years, they present this sort of enormous opportunity if you're a land manager, right? You have this decline in perennial pepperweed and riparian floodplains, you have an increase in native species recruitment, and what it could provide is an opportunity for you to think about how to best manage this species. That isn't necessarily true for your more upland environments, and obviously your levees are going to be a big source of new populations. And I think that what we landed on as management recommendations are use what you have in terms of that process, in terms of that restored disturbance regime, but don't forget that it's not solving all of your problems, and you probably have to go after more active mechanisms for controlling pepperweed long term. Okay, with that.
All right, thanks everyone for staying here through the entire symposium. I really appreciate your presence today. We're throwing a little audience participation at you at the very end of the day. So if you want to pull out your phones, you can take a picture of this QR code, or use the QR code to open a survey that will allow you to give feedback, and then we will look at it live together. So while you all are pulling up that survey, I just want to take a moment to thank all the contributors for today. We couldn't have done this without your participation, so thank you so much for your time and energy preparing your talks. They were all really great. I know I personally learned a lot. For those of you who are interested, we are going to have a no-host social hour at Lucid Winery after this. It's less than a 10-minute walk from here. The address is on the whiteboard in the back. All right, so now that you have that pulled up, I am going to show you the results. All right, so our first question that we were interested in hearing about . Right, here we go. All right, our first question here is, what part of the symposium did you find most useful? We already have 14 responses, so great talks. I agree. Thank you so much. People are saying the whole symposium was useful. Social science talk, I agree. That was really exciting. Rochelle Tanner and Brad Henderson getting special shout outs. Everyone from the whole room to facilitate the networking. I agree. So thanks so much again, Ted, for those sandwiches. It's always nice to talk to people over lunch. Hearing a whole variety of topics, bringing together a wide range of presenters. The first session gets a shout out there, and the second session. The rapid talks. So it seems like people really enjoyed the whole program, which is really great to see. All of the biological technicians for the floodplain, invasive species management. The discussion around tools for controlling invasive species, and the wide variety of talks. I do feel like that's sort of reflected in the answers we're seeing here, that we did get a wide variety of speakers on this topic of connectivity. And the talks, session 3, all right, so all the sessions are getting some special love, which I really appreciate. All right. So now for the opposite side of the coin there, so this is a little dual question. So are there topics or concepts related to connectivity that you wish we had highlighted? So things you thought you might hear or wanted to hear about that you didn't hear about. And then a just general request that if you have feedback for how we can improve the symposium in the future, we would love to hear it. We covered it all. And this will stay open. All right, here we go. More information about what was happening with climate change. That's a really interesting point. I do feel like we had a couple talks that hinted at climate change impacts. Betsy's comes to mind for me, but I agree there was, you know, not as much of a climate change bent as there might have been. Love the social science component. It's so important, agree with that. But maybe we could have even talked about that more. Another vote that we could have talked about climate change more and sea level rise, so sort of related global change. So it's a really good feedback. Interested in more engagement with the community. So maybe thinking again about those social science aspects and how we might be able to bring in community members, which we did have a couple in the audience today, both in person and online, which was really exciting. More just basic management practices. So maybe we're trying to work with speakers to get them to talk about their management recommendations, which almost everyone did, which I truly appreciate, but maybe we need even more of a focus on that in the future. Oh, bullfrogs. So, you know, maybe we didn't have any, you know, maybe some more species and taxa diversity is what that's calling for. All right. So would like to see some more technical talks and success stories. So some requests there for a little bit of a positive bent to things. Oh, thank you all so much for that feedback. It'll be open for a little while, so you'll be able, if you think of something, you know, not on your drive home, but maybe when you get home, you can try to put it in there, and we will definitely be pulling down these results and thinking about it for the next biennial symposium, which will be in 2025. So with that, we are closing out a few minutes early, and I really want to thank everyone again for being here and hope that we see a few of you all over at Lucid to chat a little bit more.